then an exciting panel, and then some information about new drugs that actually are in patients after we heard yesterday about all of the things that might get into patients. So I'm going to start out and introduce my very good friend and colleague, collaborator, Dan Lowenstein, who is the professor and vice chair of neurology at UCSF, right around the corner, who is going to talk to us about new directions in epilepsy research and get us really excited about what's coming down the pike.
Thanks, Jackie, and good morning. And I'm going to launch right in to make sure that we stay on time. So Jackie gave me what struck me as a pretty tall order about 6 months ago. She said, basically, review all of the exciting things in epilepsy research in 30 minutes. So that's what I'm going to try to do. Prepare yourself. 6 quick stories, an average, I think, of 5 minutes each. And here's what they are. I'm going to talk about seizure laser tag, genomics rules, the ultimate model system, fishing for drugs, interneurons to the rescue, and thinking big about a project, the Human Epilepsy Project. My apologies to the bench scientists in the room, as well as the pure lay audience. I've done my best to try to come up with something of a balance to make each of these topics understandable to some degree, okay? You can tell me if that works in a moment. Okay, so let's talk about seizure laser tag. This refers to a really spectacular report from John Huguenard and Jean Paz down at Stanford last year, in which they essentially showed that using a particular form of light on a particular type of channel can interrupt seizures in an in vivo model. So let me walk you through this. They used a very nice focal stroke model in which there's a thrombosis of the cerebral cortex in the animal. And John's group and others have known for a long time that there are important connections between the cortex and the thalamus. And the idea here was to see whether the thalamus, how it might be involved in a focal stroke model. And they demonstrated that was clearly the case. Here you see a control neuron versus one. This is a thalamic neuron in the injured animal. So even though the damage was out in the neocortex, they demonstrated that the neurons in the thalamus actually get injured, and they have abnormal activity. This is a control neuron in the thalamus, and this is an injured one showing spontaneous high-frequency spiking activity. So they asked the question, is there a way that we might be able to modify the activity of these thalamic neurons and alter the epilepsy behavior in the animal? Now, to understand what they did, we need to go through very quickly exactly what's going on with optogenetics. So probably everyone's heard the term. Everyone knows it was one of the scientific breakthroughs over the last couple of years, certainly in neuroscience. But just to make sure that you understand how this works, here's the 6 steps that are involved. You need to actually get an opsin, which is a very special type of protein, which is sensitive to particular wavelengths of light. And you put that behind a promoter that will put the opsin in the proper cell that you're interested in. You can package that into a virus, inject it into the animal, and hopefully the opsin is going to find its target. Now, once you've done that, you need to have a particular type of light wave, in this case a laser light beam, which when it reaches that opsin will open up the channel and modify the behavior of that particular cell. Now, just to demonstrate what this looks like, the opsins actually are sort of universally found in nature. They're light-sensitive molecules, and in this case, this is of some flagellated organism. You take out the DNA, you insert it, as I just said, say with virus packaging. It finds its way into a particular cell type, and then that cell expresses the opsin along the surface of the cell, and because it modifies the flow of ions in or out of the cell, you can actually, in a very targeted way, change the behavior of that individual neuron. So once you've done that, then you can do a whole variety of experiments, physiology and so forth, on that modified cell type. So that's what John and his group did. They created the stroke in 1-month-old animals. They then placed an inhibitory opsin in those targeted to those thalamic neurons that we just saw, implanted the laser device and an EEG into the cortex, and then did chronic recordings and stimulation and monitored the animals. So that's the experimental setup. So here's what they found. So when the animals would have their spontaneous seizures, which was part of the stroke model, and they turned on the laser beam that was focused on those thalamic neurons that would cause inhibition of those abnormally excitable cells, the seizure stopped. And that's shown here. So here's the onset of the seizures by EEG, the laser, and then interruption of the seizure. And then they went on to actually create a closed loop system. Now that they demonstrated that turning on the laser could abort the seizures, well, what about creating a system where the EEG, when it picks up the onset of a seizure, automatically turns on the light? And that's what they did. So here's the EEG recording system. They have a processor that reads the seizure onset. It turns on the laser, and then they can determine whether or not it works. And they demonstrated that, in fact, it does. Here in this left-hand panel, you see the onset of the seizure. It's detected. The light's turned on. The seizure never actually occurs. That's compared to the detection with no light, in which the seizure does occur. So a beautiful demonstration of a very, very sophisticated, refined way of targeting individual neurons and with a closed-loop system aborting a seizure. Okay, second topic, genomics rules. So this is a really nice figure from Ingo Helbig, one of our colleagues in some work that's going on in Epi4K. This is a figure that he put together for a review that we published together, but this is all Ingo's ideas. So this is basically an overview of the history of epilepsy genetics. It all began in 1995 with the paper by Sam Berkovic and colleagues describing the first true epilepsy gene, a mutation in the nicotinic acetylcholine receptor. And as you can see, this was discovered in part because of a very informative, remarkable family in which epilepsy clearly runs through the family. And so with that discovery over the next number of years, most of the epilepsy genes that were found, in fact, turned out to be ion channels. So we could refer, Ingo refers to that time, up until 2000, as the channelopathy era. Well, then we had about actually over 100 candidate gene studies that essentially all negative results. So this was using genome-wide association studies with, unfortunately, not sufficiently large enough numbers of patients. And so Ingo refers to that as the dark ages. In more recent years, we've had a whole host of studies that have been looking at so-called copy number variants in the genome. So these are variations in our genomes which are either additions or deletions or duplications. And some very important work from a variety of investigators around the world have demonstrated that these so-called CNVs are in fact important. And so we can refer to that as the CNV revolution. But really where we're at now, because of the extraordinary advances that have been made with sequencing technology, we're really at the next generation sequencing bubble. Just one example is the work that came out of Epi4K that built on the work that was done by the Epilepsy Genome Project. And this is a paper describing de novo mutations in patients with epileptic encephalopathies. In fact, in this work, there were 264 individuals with either infantile spasms or Lennox-Gastaut syndrome that had exome sequencing along with their parents. That's 800 exomes. And in that work, a whole number of de novo mutations were discovered. Some were previously known, some were new, and in fact, there's even a longer list of de novo mutations that are likely causative in this population, but we really just need more numbers to be certain about that. The thinking here is at this point, we probably can explain about 15% of the genetic basis of all epileptic encephalopathies that don't have an obvious, say, structural or metabolic cause. Now, I know that Steve White gave you an overview of a number of things going on, and he mentioned the Epilepsy Genetics Initiative. I just wanted to touch on that a little bit more because it really, this is a fantastic idea that really David Goldstein came up with at Duke, and that's been embraced by those of us in Epi4K and now Cure. And the idea here is that the clinical sequencing that is being sent more and more and more to the companies out there is really relying on the in-house understanding of an individual company. So that the company is relying on the information that they get from the literature or their own analyses. But really, we're in the era of epilepsy genomics, not just epilepsy genetics, genomics. We need as many genomes as we possibly can to really understand what the landscape is for all forms of epilepsy. So the idea here is, why not create a data repository that includes not only the exomes and the genomes that are being produced in research labs, such as the work going on in Epi4K, but also brings together the information that's being generated by a growing number of clinical testing labs. So this really is the basis of the idea of the Epilepsy Genetics Initiative, and I'm going to just show you a video.
Imagine having the ability to unlock the genetic secrets of epilepsy. Just think of what this would mean for the 65 million people currently living with this debilitating disease and the doctors who struggle to find a way to stop their seizures. Knowing what causes epilepsy and what treatment might work best seems almost like an unattainable dream. But thanks to modern advances in genetic research, this dream is close to becoming a reality. By examining the changes in a person's genes, researchers have been able to identify altered genes that lead to some people's epilepsy. With about 20,000 genes in each of us, this is no easy task. However, by using genetics to diagnose and treat each person individually, we could create a world where we tailor treatments to each individual person with epilepsy. Unfortunately, current gene testing is limited because studying gene data is challenging, and analysis sometimes needs to be repeated again and again over time before we can identify a cause. Now, that's where we come in. The Epilepsy Genetics Initiative, or EGI, is a project of Cure dedicated to advancing epilepsy genetics. Cure has created a place where people with epilepsy, with the help of their doctors, can house their genetic data to have it analyzed and reanalyzed to help find the cause of their own epilepsy and help advance science as it seeks to uncover new causes of epilepsy that have yet to be discovered. Researchers will be able to study the genes which will be confidentially collected in EGI and build a database that has never existed before. Just imagine what this will mean for epilepsy research, therapy development, determining which treatments work best for which people, and discovering why some people with epilepsy suffer from coexisting conditions like depression and learning and memory challenges. By bringing together people with epilepsy, medical professionals, and scientists, EGI will help to improve the diagnosis and treatment of epilepsy in a way that has never been done before. Find out more at www.cureepilepsy.org/EGI.
So as we were developing this idea, one of my concerns was that the companies might not be that interested or willing to deposit such information into a common repository. And in fact, we've discovered just the opposite. And so this has been a beautiful experience, actually, in collaboration across all domains. Just a couple of other details about this. The idea now is with a repository that's accumulating thousands and thousands of exomes and eventually genomes over the next number of years, that groups can reanalyze the data on some regular basis so that we can take advantage of essentially the numbers, because it's all in the numbers. Those results will be communicated back to the referring physician and their patients, and then the data will be made available to all qualified groups. So whether it's an academic group, a company, other investigators, that's what the repository is meant for. And finally, before I go to the next topic, I just want to mention Ingo, who I referred to just a couple of minutes ago, has been a remarkable blogger. And the International League Against Epilepsy, the Genetics Commission, has actually adopted his blog. And if you really want to go to the source of all exciting news related to epilepsy genetics, go to epilepsygenetics.net, and I'm sure you're going to be entertained. Okay, next topic, the ultimate model system, U. So everyone's heard about inducible pluripotential stem cells. I thought I would just walk us through more precisely what that means. So the idea here is that you have an individual, in this case the patient or ourselves, and by taking a skin biopsy, literally a biopsy of skin from the forearm, you can dissociate those cells, grow out fibroblasts, and with specific molecules, you can drive those skin cells to be inducible pluripotential stem cells. They're stem cells, meaning that they have the capacity now to go from their very embryonic, if you will, condition to be induced into different cell fates, to be made into essentially any of the cell types in one's body. When you have that type of IPS cell, or inducible pluripotential stem cells, there's two big directions you can go with that. First, you can actually, in the case of a patient who has a gene mutation, you can modify that mutation, you can correct it in the IPS cell, then drive it back to a healthy cell state, and theoretically transplant it back into the patient. And this is something that has already been done with blood-borne oncologic disorders. On the other hand, you can take these iPSC cells and drive them into a differentiated state, into a certain cell fate, and then use that cell type to screen for compounds that might be useful in a particular disease state, and then find disease-specific drugs and then try it in the patient. So that's the general idea of the power of iPSC cells. And many of you have, I'm sure most of you have heard of the fantastic work that's been going on in this area, led in large part by Jack Parrott and his colleagues at the University of Michigan. So Jack had the brilliant idea of deriving these iPSC cells from patients with Dravet syndrome, knowing, of course, that in many of those patients, the gene mutation is in the sodium channel subunit. And by creating that cell type from an individual patient, not only did you have the specific mutation, but you had it on the specific and truly unique genetic background of that patient, which is very important. And by using, again, specific factors to drive the fate of those cells, he could make, for example, as shown in this slide, neurons. Now, by studying those neurons, he and others have been able to show that there are clearly abnormal characteristics of the firing properties of neurons derived from patients with Dravet. That's not a big surprise, but this was a very clear, elegant demonstration of that, and it's actually made significant progress to understanding the underlying biology of Dravet syndrome. And as Jack is fond of saying, this is really human seizures, and this is like a human's cells in a dish, which is pretty amazing. Now, Jack also had the very clever idea of asking, well, what about the association between Dravet syndrome and SUDEP, which of course is, clearly one of the one of many tragic aspects of this form of epilepsy and epilepsy in general. And knowing that these various disorders of ion channels can predispose to SUDEP, they asked the question, well, what's the connection between the ion channel mutation in Dravet and the behavior of heart cells in those patients? So again, step back for a moment. He has iPSC cells. They can be driven to whatever fate one's capable of. So why not drive them instead of to neurons into cardiac myocytes? And that's in fact what they did. This is an example of what they look like. And by creating these myocytes, they could study them in the dish. And here are some videos of the behavior of these cells. So in the upper left-hand panel, these are control stem cells. And they grow, they differentiate, they replicate, and actually they create a syncytium. They actually create essentially a cardiac tissue in the dish, and they beat. And if you just watch the behavior of these cells, that is a beating piece of heart tissue. Now let's compare that to three cultures of myocytes taken from three different patients with Dravet syndrome. Each of them has a different mutation, although they all involve the sodium channel subunit. And the question is, are any of them associated with abnormal behavior of the cardiac cells? And I'll show you all three, and you can take a look and tell me whether any of them look abnormal. Here we go. There we go. So if you imagine taking the pulse of those additional cultures, any of them look abnormal? Actually, one of them looks abnormal, and it's the upper right-hand panel. It's actually having an arrhythmia. And so here's a really dramatic demonstration of the power of iPSC cells in being able to advance our understanding of the phenotype here related to cardiac function in Dravet patients. Okay, here we go again. Next topic, fishing for drugs. So this is a spectacular paper that came out of Scott Barabant. Scott, you're here somewhere? Yes, there he is. Okay, so many of you have probably heard about this. This is arguably one of the most important papers over the last year and a half to two. And this is a remarkable study in which the zebrafish was used as a model system for screening for drugs. So you probably have seen pictures of zebrafish. The work that Scott did actually involved this big guy here, although he's really little. This is the larva of this. This is why they're called zebrafish. They've got these stripes. That's the larva. And what Scott and his group have done is they've been able to, because these organisms are so relatively easy to manipulate genetically, they actually placed an SCN1A, a Dravet mutation, into these zebrafish and created essentially a Dravet zebrafish. And you can study these actually in little tiny wells, 96 well plates, and you can watch the behavior of the fish. And if you take a look in the upper right-hand corner, you're going to see the control animal here and a Dravet mutant here. And you'll notice that there's a heck of a lot more behavior going on the right-hand side compared to the left. That control animal is alive, by the way. You'll see it move every once in a while. But the mutated zebrafish is actually having seizures. And this was, you can hold these fish in place and actually do extracellular recordings and observe directly the seizure activity coming from this animal's brain. And the beauty of this system is that because you can study one fish per well, using a tracking system, you can, in an automated way, you could actually compare the behavior, the swimming behavior of, in this case, control fish versus those that had the mutation. And so Scott used this system to first test whether known anti-epileptic drugs have an impact on this model, and in fact, they do in a selective way and in a predictable way. So that was a proof of principle that the system actually could be effective in assaying for the ability of different compounds to control the behavior of the animals. And then they carried out a screen of a library of molecules, and here's the readout. And you'll notice that the y-axis is velocity, percent change from baseline. So using the seizure model, what we want to see is a decrease in the movement of the animals. And so anything that's down low is a possible hit. Of course, if it's too low right at the baseline, that means the animal's not moving at all, which means it could be dead. So you have to screen for that. But in fact, the red dots show a significant decrease from baseline of compounds that were tolerated by the animal. And lo and behold, the drug that really stood out was clemizole. Clemizole was a drug that was approved by the FDA in the mid part of the 20th century. It's an antihistamine that was planned to be used as a anti-bronchospasm agent. It never really got regular use. Scott, you probably know the details as to why, but he is really moving forward now to see whether this could be brought to use in humans. When he tested clemizole in the individual fish, he discovered the following: that is, the untreated animals would have seizures. Clemizole aborted them essentially completely, and this was not due to just paralyzing the animals, because using a drug that was a neuromuscular blocker, in fact, did not have an effect on the seizure activity. So really a fantastic discovery. Okay, Interneurons to the rescue. Actually, this is another Baraban lab work. So here the idea is, could you come up with a way of just placing cells directly into the brain, into an epileptic region? The idea is you identify where the zone is, where the seizures are coming from. Then you transplant the cells into that area, hope that they integrate and control that focal area of network dysfunction. Well, the question is, which cells? I mean, the brain is a complicated thing. Here's a cross-section of the brain, and there's 10 to the 10th cells there to choose from. How do you come up with a cell that's going to be of benefit? Well, everyone knows that, for example, the neocortex has not only does it have a lot of cells, it has many, many, many different cell types, some of which are excitatory, some of which are inhibitory. Probably it would be ideal to, in the case of epilepsy, to come up with purified inhibitory cells that would make sense for quieting an excitatory network. And in fact, a lot has been discovered over the last decade and more about how it is that the inhibitory interneurons make their way into the mature neocortex. And in fact, many of them come from a region deep inside the developing brain called the ganglionic eminence. The ganglionic eminence gives rise to these primordial cells that are destined to travel throughout the neocortex, find their location, and develop into inhibitory interneurons. So Scott and colleagues took advantage of this understanding of basic fundamental developmental neurobiology and asked the question, could we isolate the source of these inhibitory neurons and see if they develop in a way that we could transplant them into a seizure focus? And that's in fact what they accomplished. So here's what they did. They took the medial ganglionic eminence, which is the source of GABAergic interneurons, and tested whether or not they could migrate into a host brain. And then once they established that, tested whether or not they actually have mature firing properties and whether they integrate into the host circuitry. And the lower panel is part of the work that they did to demonstrate those last two properties. In fact, the cells do migrate, they do have mature firing properties, and they do integrate. This is what they look like if you take these medial ganglionic eminence cells and transplant them into the hippocampus. In the case of a very young animal, they'll migrate up to 5 millimeters. In a P60 animal, which is a very mature rodent, they'll actually migrate 2 millimeters. And these can migrate in the cortex, in the hippocampus, and the amygdala. So with this setup now, Scott and his colleagues had the ability to take isolated inhibitory neurons and place them into a seizure focus. And this is the work that they did. When they placed it into a genetic form of epilepsy, in this case, a potassium channel mutant, which had spontaneous seizures, and injected them at P2, they saw a 96% seizure reduction. This is in an animal that has a general dysfunction of the brain, of the cerebral cortex, due to a potassium channel mutation. But I think even more spectacular was using these cells in an acquired form of epilepsy. So they used the pilocarpine model of temporal lobe epilepsy. They did an adult transplant. Remember, that's at P60, where they only migrate a couple of millimeters. And they, in fact, found a 92% seizure reduction, specifically when the cells were transplanted into the hippocampus, not, for example, into the amygdala. And some of the comorbidities that are associated with this model were also reversed. Okay, last topic, and this one's really quick. Let's think big, the Human Epilepsy Project. This is a project that Jackie, Ruben Kuznecki, and a whole number of other people around the country and the world are working on. We had this idea about, God knows what, Jackie, 4 years ago. So this is a project that we believe deep in our hearts and our minds is really, really important. So important that even though we had some difficulty getting funding from NINDS at the beginning, I should have said at the beginning of my talk, actually everything that I presented couldn't have happened without NINDS. All the investigators that I've mentioned have been supported in major ways from funding from the NIH. With the Human Epilepsy Project, we really turned to industry supporters to help get us going, and NIH actually has been really helpful in giving us ideas as to how to continue this work. But the basic idea of the Human Epilepsy Project is we need to do something about the fact that we do not have any bioassay, we do not have any biomarker for disease activity in people with epilepsy. We have patients who come and want to know whether their epilepsy is getting worse, whether it's stable, or whether it's gone away, and we essentially have no way of knowing that answer. And until we do, the suffering that is associated with epilepsy and its comorbidities is never going to go away. So the Human Epilepsy Project is basically that, it's built around that whole idea. And so what we're doing, we've created a prospective observational cohort study. We're attempting to enroll between 500 and 600 people with recently diagnosed focal epilepsy, and this is a prospective study, so they're going to be followed for a minimum of 3 years. And what we're doing is collecting a lot of information about these individuals, both in terms of their clinical semiology, their imaging characteristics, their EEG characteristics, blood for DNA and serum for proteomics, and urine for metabolomics, as well as really detailed information on neuropsychiatric comorbidities. And the idea is that by having enough patients over time and following them carefully, we will indeed be able to discover the biomarkers that we so sorely need. So the real goal is to create an open repository of detailed clinical information and these samples for future studies. We need help, by the way, for this. We're underfunded, so if there's anyone out there who's interested in participating in this great endeavor, I can see Jackie smiling, I'm sure. We'll crowdfund it. There you go. So there you go. That was a quick foray. I tried to highlight seizure laser tag, the ability to target individual neurons and stop seizures. We're in the era of epilepsy genomics, and the Epilepsy Genetics Initiative is a cool idea to expand that. I've told you about inducible pluripotential stem cells and the insights they're providing, the amazing zebrafish, transplanting interneurons into targeted areas and aborting focal seizures, and finally, the Human Epilepsy Project. So thanks so much for your attention.
This has been an incredibly wild ride through science, and because Dan isn't going to be here for that long, he has to go back and work on these amazing things. If there are any questions, we can take them now. He will be here for the rest of the morning, but it was that clear. Okay, so thank you very much, Dan. That was incredibly informative. And now for something completely different. So next, I want to invite up Mark Evanstad, who is the CEO of Upshur Smith, who, as we have heard and will hear, is very involved in finding new therapies for epilepsy. And he's going to talk to us about the, improving delivery of therapy in patients who need them, an industry perspective. And I have to personally thank him because I asked him to come do this talk about 6 months ago, and he is a very busy man, and he has, made it his, mission to come here. So thank you very much, Mark, in advance.
Good morning, everyone. Actually, I'd like to thank Jackie for
most important thing that you said was getting medicines to patients in a way that's affordable to them, because we have all suffered through having medications that we think can help people, and suddenly they finally get approved and on the market, and then we find out that nobody can take them because there are those systems of no. So anything that anybody can do in that regard, we very much applaud. Does anybody have any questions for Mark before he leaves? I don't know if somebody is getting up to go out or go to the microphone. Go out. Okay. No questions? Mark, thank you so much. Now I'm going to hand the mic back over to Roger Porter.
I'd be grateful if our panelists would come up on my right-hand side, please, so that the webcast works better. And I will introduce you as you are coming up. This is a terrific panel, one that I worked very hard, I have to say, to get together, and I'm very proud of it. It's the role of corporate and government partnerships in developing new epilepsy products. We have Santiago Arroyo from the Vice President and Head of Clinical Research and Chief Medical Officer of Pharma Therapeutics from Worldwide Research and Development from Pfizer. We have Roy Twyman, MD, Vice President, Global Head of CNS Development from Janssen. Both of these gentlemen are tasked with what does Big Pharma look for in a new drug for epilepsy. Then we have Dr. Michael Gold, Vice President, Global Therapeutic Area, CNS from UCB. What does a medium-sized epilepsy-oriented company look for a new drug for epilepsy? And finally, because drugs isn't everything, drugs aren't everything, we have Frank Fisher, the CEO of NeuroPace. What does the device industry look for when considering developing a new product in the epilepsy space? So I have invited each of you to have 5 or 10 minutes to tell us what you think right off the top before we initiate the actual panel discussion. And I suggest you come up here at the microphone, it'd probably be easier. So Santiago?
Sure, thank you very much, Roger. It's a pleasure to be here, and it's a pleasure to see so many known faces for so many years. So let me start, I think I have here this, by a disclaimer, of course. Everything that I'm going to say is my opinion, and just my opinion is not Pfizer's opinion. And I have a vested interest in Pfizer's well-being. So let me start with, when you think about what do we need for a new drug to be successful in big pharma, we need efficacy. We need a drug that is differentiated, and the best differentiation is efficacy. We want something that has superior efficacy that will really make a difference. There are other characteristics that we will look for, like having improved safety, tolerability, absence of dizziness, of somnolence, better delivery of effects, like no titration, better scheduling, and broader spectrum. And of course, it would be ideal to have a disease-modifying drug, although obviously we are very far from that. But that will make a real difference. Not only I think that efficacy is important, I think that mostly neurologists across the pond, I mean here and in Europe, believe that efficacy is the main thing. Safety appears as a secondary, tolerability appears as secondary, basically because many or most of our antiepileptic drugs are already relatively well tolerated, and efficacy is the major feature that is missing. So this is a cumulative distribution plot, and for those that are less familiar with it, on the x-axis is the improvement measured by change in seizure frequency, percent change in seizure frequency from baseline, and you have on the left side, you have a reduction in seizures, on the right side, a worsening in seizures, and in the y-axis you have the percentage of patients. So, and this is one of the Lyrica pivotal trials. And you see the percent improvement, the 50% improvement, is about 45% or so. And this is very typical. The effects of pregabalin in this case are very typical of all of the major antiepileptic drugs. what we get is about 50% of patients improving about 40% of the time, and we have very few patients improving 100%, very few seizure-free patients. So what we want is to basically raise that line and have no seizure worsening, have a very substantial amount of patients that have a 50% response, and have many, many subjects, and I put here like 50%, that are seizure-free, because seizure freedom is what really makes a difference. So how can we do that? And there is not a clear answer to that, but I will think that it would be naive to think that one drug is going to be the magic bullet that is going to change epilepsy forever. Epilepsy is a very heterogeneous disorder, and no one drug will make that happen. So the only way to go around that is to try to understand who are the responders within the population of patients that you are going to treat. In order to get to the responders, you need a biomarker, and probably one of the best potential biomarkers is genetic variants. So how to get to that from analyzing common variants in an heterogeneous disorder is pretty complex, and there has been some efforts done in the past with 1,000 patients here, 500 patients there, 3,000 patients, but really you need tens of thousands of patients in order to get variants that will make a difference. And once you identify those variants, you can probably get to understanding their functionality and the directionality, and finally understand what will be the pharmacology of a potential druggable target. So we are far from there, but I am happy to see that there is already something in the epilepsy field that is starting, and hopefully we'll get to reach to those 20, 30, 40,000 patients that have good phenotyping and good genotyping done. The other possibility, instead of go to the common variants, is to go to the rare variants, to go to subjects or to patients that have a rare genetic disorder, and as Dan was saying before, get tissue, or in this case, blood from these patients, and do induce pluripotent embryonic cells, and from that differentiate those cells into neurons, and basically have the ability to analyze the properties of those neurons in a dish. Dan was saying before that you need a biopsy. There are some labs, our lab, that we can do that from blood. So you don't need anymore a skin biopsy. From blood, you can get to the iPS cell, you can get to the cortical neuron. So basically, today you have a screening platform that will allow us to understand the phenotypic information from that cell, the genomics information on the other side, and hopefully be able either to find a genomic marker or to understand the pharmacology of new targets. I'm happy to say that Pfizer, Interneurons, and the Epilepsy Foundation of America are currently in the process of finalizing an agreement, and we will be working with children of Dravet syndrome, trying to basically do what I have just mentioned here in this slide. So I'm very hopeful that we will make a lot of progress, not just for these children, but for many other people with epilepsy. So what is this is my last slide, by the way. Sorry for this. So what is the vision at the end of the day? I mean, where do we would like to be? I mean, something that is interesting to see is that many drugs for epilepsy work across different disorders. They work, valproic acid works in epilepsy, it works in migraine, it works in bipolar, pregabalin the same. Many of our compounds work across different disorders. So would it be great, right? If there were some variants that were associated with the function of some channels or receptors that could be common across different disorders, and that will define those responders to specific compounds. Could we evolve like oncology has evolved? That right now, epidermal growth factor mutations are treated, not specific cancers. You treat the mutation, you don't treat the cancer. So if we were to reach that to that level, it will probably be very possible to have a high efficacy in a few patients across different disorders. It will be cost effective. Thank you.
Thank you very much. Roy?
Great. Thank you, Roger, for having me, and thank you, Jackie, for inviting me. A disclaimer or two also. My title's not in CNS development. We don't have a CNS development group anymore. Our organizations organize ourselves into disease areas, and I'm in fact in charge of the Alzheimer's disease area at Janssen. And that being said, so Janssen's entirely neurological focus is in Alzheimer's disease and neurodegenerative conditions, and epilepsy is not a priority. And so it's been a while since I've actually thought about epilepsy itself, so I've jotted some notes down here, and much of this actually follows our thinking about any opportunity as we get into potentially developing a drug product itself. And let me first start saying that you know, the world of drug development and the pharmaceutical business of yesterday is not going to be the world of tomorrow. And so the industry is now challenged with what the future is going to be like. And that challenge is that we need to be innovative. And so the history of Me Too type drugs and things are not going to be really reimbursed you know, substantially for large companies to be able to realize the profits and necessary return on investments from that. So the future of therapies tomorrow need to be innovative. And we heard a lot of fantastic things yesterday, actually, being innovative in the field of epilepsy. But when one thinks about the special timelines and challenges within the drug business, these timelines are quite long. So what's innovative today needs to be innovative when your product hits the market 5, maybe 10 years in the future. And in fact, it needs to be remaining innovative as it returns on the investment over the lifetime of that drug over the next 5 or 10 years as well. And so you need to be very, very forward-thinking in your innovation as going forward here. So the biggest troll, if you want to call that, is Father Time. Because Father Time keeps on pushing that innovation bar higher and higher and higher. And so we need to beat that innovation bar by the time this product reaches the market, and it also needs to be innovative for the life of that product itself. And so how do we think about that? So first and foremost is the unmet need, right? You know, and can we access the market itself? And so clearly in epilepsy there's an unmet need, and there is access to the market as well. And we all know, and we've heard this, is the refractory population needs better drugs, they need higher efficacious drugs. Epileptogenesis has been touched upon here. This would be a fantastic entry into this field as well. Santiago just talked about, and there's going to be some overlap between what I'll touch upon and what Santiago touched upon, is targeted medicine. This is the future. Is we're going to go after stratified populations and targeting therapies to specific patients with diagnoses or diagnostic approaches and biomarkers, as Dan Lowenstein pointed out. It's going to be really playing a big role in this in the future itself. And so this is another great opportunity in epilepsy. And also there's the opportunity of improving the current therapies. We know that 60–70% of subject or patients are adequately treated with their drugs today, but you know, the drugs are producing some interference in their lifestyle and things. And so there is an opportunity perhaps improving the overall risk-benefit profile itself. So that's number one, is the unmet need. The other one is that as we think about this innovation hurdle that we have, it has to be differentiable. The opportunity to differentiate must be there, and we must know that we have an opportunity to get to that. And this is a big challenge specifically for epilepsy, as we don't have really predictive mechanisms to identify, do we have a potentially useful drug that will truly differentiate in the future itself. And so this has been proven. This field has been extraordinarily successful in introducing new drugs for patients with epilepsy. There may be, what, I don't know, 20, it's been a while since I've been in epilepsy, 20 drugs now. You know, can we show that any of these drugs are clearly differentiated from any of these other drugs? No. You know, we have multiple mechanisms and things, but no clear differentiation. So we need to figure out how to bring forward new drugs that are truly differentiated, that beats that innovation hurdle, so one can get actually a return on investment in the future, as I pointed out. The other thing, the number 3 thing that we bring in is, where is the science? Is the science substantial? Is it really advancing? Will it provide us that innovation opportunity through the science? And so Dan gave a fantastic talk. This is really exciting, seeing where the science in epilepsy is going, because this is where a lot of the other fields have really taken the bull by the horns. They've moved away from animal models. Animal models have been good in sort of testing some pharmacology and things, but the animal disease models have been very inadequate. And until we get the iPSC cells or other things coming forward here, animal models have not been all that useful except for some pharmacology itself. What we need in fields are things like molecular targets, and so Dan touched on some of those, and it's really exciting to see that perhaps the new wave of genetics, maybe copy number variations, things will add to this itself, and so this is extremely exciting to hear that for epilepsy. I'll give you an example. I said I was in the field of Alzheimer's now. The Alzheimer's has a couple, it's a proteinopathy basically. One of the key proteins, amyloid, has over 230 mutations pointing to amyloid as causal to the disease, which involves 2 gene targets there. 230 mutations establishing causality. And so we have a great molecular targets not only in amyloid, but also in the tau protein, where the science, and again, this exciting science says that there's something interesting about tau protein where it seems to be acting like a prion protein and spreading. So it provides a new target for pharmaceutical intervention. So that is an example of exciting science that drives industry into going after specific disease states itself. The other one is also in the sciences, can we translate it, or do we have translatable models? And this is one of the other things is that Dan pointed out, was, again, applauding the efforts here, and I've heard about some of the remote monitoring approaches from yesterday. Translatable models that allows us early decision making, such that we can actually identify what is potentially promising or what is potentially a dead dog and we need to kill it, early. So these early proof of concept approaches need to be established for epilepsy as well. And if there is a path for that, that makes it an attractive opportunity for investment into that type of disease area. The fourth thing is there a path for development? And so that's one of the positive things about epilepsy is there's a clear path for development for this. But we also heard some exciting news from Dr. Katz yesterday that perhaps there are new ways of thinking about enabling development in epilepsy itself, and this is actually going to potentially help pique the interest of industry with regard to development in epilepsy as well. So I applaud that quite a bit. And then the fifth area is what about the competitive landscape? In epilepsy, this is a problem. There's been a lot of drugs that's entered the market. There's a lot of generic drugs there. And so what does that mean from the standpoint of standard of care? The standard of care allows many, many treatment opportunities for patients where physicians have the choice of a multitude of drugs. And so the driving force, the economic driving force for this is that to try the cheaper drugs first, because there's really no difference with them. We haven't established differentiation, is to try the new drugs first before actually getting to a new product. And so unless the new product clearly has, as I said, that differentiation and providing that additional value, and it's going to be principally an efficacy value, you're going to really have a very difficult time competing for price in this highly genericized market itself. And then lastly is business opportunity. Is that, as we look at this long timelines for development, and like I said, it takes a long time to recover the investment itself, the product needs to be protected. I see a lot of things that look very, very interesting, and unless there's a long enough lifetime to protect that product that is intellectual property, patent life, you're not going to have the opportunity to get that return of investment because you can't protect it unless there's some other novel way of approaching that provides protection for that product, and that's extremely important for us. I think that covers most of the key points here. So again, I think one of the things that really excited me yesterday was Roger's talk, actually, and that in fact there is a wave of innovation that's happening in epilepsy. And actually, from my standpoint, as one of the folks in our company, the leaders in our company that actually decided that epilepsy was not an attractive opportunity, I actually start to see a window opening here, and actually I'm quite excited, so thank you for that.
Well, thank you. Dr. Gold?
All right. So first of all, thank you to the organizers for inviting me. I'm standing in for our Chief Medical Officer, Iris Friedrich-Lowe, and she sends her regrets that she could not be here. And for some of you who know Roy or me, you have to appreciate the irony that there's a world-class epileptologist learning about Alzheimer's disease, and I'm a lapsed behavior neurologist who cut his teeth in Alzheimer's disease and now treading water in epilepsy. So, just a disclosure. So the UCB context, and for those of you, I don't know if the slides are going to be made available to you, that hyperlink will take you to this issue of this Nature review, which we sponsored. And in it, there's some very interesting articles about epilepsy, cutting-edge science, but I want to call your attention to a little bit on the front matter, and there's an email there for you, okay? And that email there is actually a humble and sincere outreach from us in UCB to any of you out here in the community, that if you have an idea that you want to share with us, please do so. We don't know what we don't know, and we're certainly open to the notion that there are people out there with good ideas in epilepsy that could help patients. So please feel free to reach out to us. And Henrik, 1-800-HENRIK is our, for those you know, Henrik Littgaard is going to be sort of the point person. You can see the UCB context, and it's a bit of a shameless plug for the company. We are committed to patients with epilepsy, persons with epilepsy, families that deal with it. Keppra is the engine that keeps on giving in 90 countries. Vimpat is doing its work. Brivaracetam is completing its phase 3 development. And Joe D'Souza, who's a colleague of mine, will provide you a little bit of context there as well. We're working with a broad range of companies to work on different aspects of helping patients and persons with families with epilepsy to deal with this disease. And again, we have this broad background in terms of expertise. I think folks have talked about this, so I'm not going to read the slide to you, but the idea here is obviously it's a genericized market with many available drugs and a real problem with the number of drugs that are in early development and in phase 2. If you actually talk to regulators, we're not talking just in the US, but across the globe, they're really not terribly excited about Me Too's. Reformulations are not terribly exciting to them. Regulators are also interested in innovative therapies. And if you talk to payers, they're also not terribly excited about epilepsy. And I'm going to take a moment here and say, I don't agree with the perspective that it's a culture of no. We've sat with these payers, and to the T, they're all interested in providing access to drugs that are meaningful to their patients and to the people who are paying for this. The problem, and I'll get into it a little bit, is they don't understand epilepsy. They have really no good understanding of what the problems. They see a bunch of approved medications, they see a lot of generics, and they think the market is satisfied. And we just actually had a group of these payers, both US and Europe, at a meeting outside of Brussels. And when you ask them about what do they understand about other parts of epilepsy, the different etiologies, how it's classified, what's unsatisfied about the market, they have no clue. And so it's easy to say no when you don't understand. And so what I would really challenge this group to do is, they are not your enemy. They really are not your enemy. They just don't understand. And we have a huge opportunity to actually educate them and bring them into the fold to help them understand why access to certain medications may be much, much more important. So the context from a patient perspective, we spend a lot of time talking to patients, interacting with patients and their families to try to figure out what they want. And I don't think it's unique to epilepsy patients. What they want is a cure. Either cure me or prevent me, and if you can't do that, then help me manage the symptoms as well as you can. We understand that getting a diagnosis that's timely and correct is difficult for many patients, okay? Shuttle around between different neurologists. Some syndromes are not that simple to figure out, okay? Getting put on the right drug at the first time is a crapshoot. So some patients get moved around from one drug to the other. We think we have an understanding in terms of rational drug selection, but I would challenge to say that not many physicians really understand how to pick the best drug for the first go around, okay? And if we talk about issues of adherence, one of the major reasons that patients with epilepsy stop taking drug or not compliant is a side effect profile. And there's this notion that somehow poor adherence is an irrational thing. And I keep on pushing about my colleagues that sometimes non-adherence is an absolutely rational decision when you take a trade-off between, let's say, a seizure that happens once a year and not being able to focus on your work, or getting, not able to find your way back home because your attention or memory are impaired, that somehow that trade-off is not necessarily a bad one. And there still, as some of you have noticed in that video that we showed before, there is still substantial stigma. So in terms of the scientific context, I mean, I'm preaching to the choir, guys. It's a complex collection of diseases, syndromes, complex genetics. There's interactions between the environment, aging. We know that bimodal distribution. The technology is improving in terms of diagnosis and treatment. There are preclinical models. The iPSCs have everybody excited. It's not just an epilepsy, but I can see and I can hear the vibe here. The notion of being able to take somebody's cells and have a model of the disease in a dish is electrifying, no doubt about it, okay? And from our perspective, although epilepsy is an ancient and historic disease, it still requires specialized treatment and expertise to diagnose it correctly. And in many places in the world, it's not that they lack, there is no expertise, there is no access, okay? So when we talk about the issue of emerging of science, so one of the things that we're struggling with is how to actually get the right patients on the right drugs. And what you see here is a whole identification of things that are coming to bear in the area of epilepsy, inflammatory pathways, transcriptional issues, changes in receptor expression, blood-brain barrier issues as well. And those have translated recently into a couple of new areas. And the talk that you heard yesterday from the folks from Sage about refractory status is one example where it's a fairly new population, okay? The idea of super refractory status is fairly new. High mortality, high morbidity, and clearly need for treatments. But it's driven by the fact that there is now new data about the issue of the downregulation of the GABA receptor. And so if you know that there are now families of compounds that address receptors that are not downregulated, you now have an indicator to a specific patient population for whom a drug is particularly indicated. And if we want to call it precision medicine, let's call it precision medicine. But that's a bit of a driver, is to match a drug with a known pharmacology or specific pharmacology to patient populations in whom that pathology is driven by that particular problem. And as you heard yesterday, there are studies already ongoing in refractory status. Same thing for neuroinflammation. There's emerging evidence of the role of neuroinflammation in temporal lobe epilepsy, neurofibromatosis, NMS. Cytokine profiles are emerging. There are now druggable targets, and there are exploratory studies that were being done, especially with IL-1B and TNF. So if you can now fragment the patient populations, okay, there are markers of inflammation in these particular patients, and you have particular targets that you can go after, now you're segmenting the population, and you have a better match of a patient population with a particular approach. What is not likely to work anymore, okay? We are not likely to develop a drug just because of a novel mechanism of action. I think Roy eloquently made the point that differentiation is a problem, and we don't have a marker of why somebody is likely to respond to mechanism A versus mechanism B. So while I applaud, and I can't tell you how happy we are to see people pushing the envelope in new mechanisms, just because it is a new mechanism is not going to be sufficient to actually invest in developing it, okay? Just because it works in every animal model is the same rationale. Just because it works in a broad spectrum, you still haven't handed us, in pharma, a rationale for differentiation, okay? We talked about the issue of precedent and mechanism of action with a little extra. This is particularly difficult because it's, differentiation or superiority when you have a little bit of a tweak in formulation gets very, very difficult to prove. And there's this kind of notion that if you get a drug, you just get it out there, And in a period of a couple of years with 10 to 15,000 patient years of exposure, you'll figure out what makes it special. And I recognize that this has happened more than once. Levetiracetam was a great one, and so was lamotrigine, a drug that went out there with no a priori understanding of what was going to differentiate it, but over time and over use, clinicians found its niche and people really started to understand the value. Again, Roy said it, that model is not likely to work. And so we have to be much more creative and think about how can we actually get time and space to allow a novel mechanism to be used in the wild, okay, to be able to really figure out, does it have a particular space? And here's one of the places where I would say, here's an opportunity to talk to payers, okay? You can argue one of two models. You can say, you know what? Let's get it out there. It's not a matter of an approval. You can get it approved, but you can talk to a regulator and say, you know what, give me 3 to 5 years at a discounted price. Make sure that we have adequate access. And then with some hypothesis-driven studies or observational studies, if with X amount of exposure we find a differentiating signal, then you'll agree to give me a bump in the price to actually get a decent return on investment, okay? You could conversely get pushback and say, we'll give you a premium price for 3 to 5 years. But if by that time you don't find a signal, we're going to discount your price. But either way, and it's not, these are not exhaustive. I sincerely believe that there are models out there that would allow payers and sponsors and patient advocacy organizations to find a middle ground to allow medications with novel mechanisms the time and space to be explored in the real world. And so really what's the hope from UCB's perspective in that mid-sized pharma is that we're hoping that by matching patients with the right drugs, that effect sizes will be larger, that it'll be easier and our sample sizes will be smaller because you're actually matching patients to their pathology. Clinical utility will be easier to demonstrate to regulators and payers, to patients, to their families, to their physicians, okay? Benefit risk issues will be improved, so trade-off in terms of better effect and maybe the side effect profile will be more manageable. You won't have to be struggling every day with differentiation against the multitude of generics. Health economics will be easier to demonstrate. And we hope that if you have this kind of match in the pathology with a drug, that it'll actually drive patients to be diagnosed earlier and more appropriately because you'll now actually have something that is specifically targeted for them. And so what's missing is really this issue of complementing models of diagnosis with a mechanistic classification. So we talk about in some other areas whether it's an inflammatory or a metabolic or this or that. So maybe one of the challenges here in the epilepsy community is to figure out, can we start to think about a mechanistic classification? We need to understand how these disorders or syndromes evolve with time and age and comorbidities, okay? And we need to also talk about the fact that there are regional differences in expertise. So treatment of epilepsy in Africa or Latin America, some areas, it's going to be very, very different than it is in New York City or Philadelphia. And I think we have to be cognizant of that. And it's really development commoditization of clinical trial designs. And this is maybe something that's close to dear to many of the hearts. We need to have clinical trial designs that are commoditized and standardized so we're collecting the same data. Cross-compound comparisons are facilitated. We can train, and this is a lot of what Transcelerate is doing maybe in other areas, is standardization of training of raters, sites, CRFs, so we actually cut the cost of clinical development and actually emphasize the ability to recruit patients faster, better, and cleaner data. So I think there's a lot of work for us to do. And just to kind of conclude for me, plenty of unmet medical need, lots of issues here. Okay, I think there is an informational, educational gap that we need to work with in terms of the payers. Okay, a build and come approach doesn't work. We have to be able to come up with, hypothesis-driven, scientifically grounded reasons why we think a particular molecule is going to differentiate. Okay, we have to approach this on a global basis because if you actually look at burden of disease estimates in terms of epilepsy, the real problem is in emerging economies and developing economies. And I think, and this is something I've said, I think it's the best molecule wins, all right? We need to work together to give the best molecule the chance to win out there for the patients that need it best. And I think that's it for me. Thank you.
Thank you very much.
Good morning. How's everybody doing? All right. Glad to see everybody's awake. Terrific. So I was just thinking, coming up here, first of all, I want to thank Roger and Jackie for inviting me to participate in this panel. It's nice to have medical device representation amongst this august group. I was just thinking that looking at the titles, or the various titles of the talks, we had what does big pharma think about, investing in an epilepsy product, and then the second topic was what does big pharma think about investing in an epilepsy product, and a medium-sized pharma investing in an epilepsy product. If this meeting were held 10 years ago, we would have had 50 more grades of pharma all the way down to teeny-weeny pharma, and then, oh, by the way, maybe there's a medical device that might have a piece of action here. And it reminded me of my first AES meeting in December of 2000. And I walked in, and the main purpose was to go through the scientific exhibits and whatever, and there was also, of course, the exhibit floor. And in the exhibit floor, there was absolutely nothing but pharma products, and Cyberonics had this tiny little room off to the side where they were throwing some posters up. And the reason I go through this is that in that period of time, medical devices and the medical device industry really has had much more of an effect and ability to bring therapies to the epilepsy community. And we're very proud to be a part of that. When I think about the device industry and the question that was posed, which was, what does the device industry look for in making an investment in an epilepsy product? If you think about two categories for the device industry, I'm sure they've never broken themselves up this way, but think of it as either there's an established medical device company, in other words, they're in business, they're actually selling a product or multiple products, or there's the startup community. So if you're an established medical device company, there are two reasons that you would invest in an epilepsy product. The first reason is that you are already in the epilepsy business. You're selling products to the epilepsy community. Therefore, you're selling products to an epileptologist, a neurosurgeon, or a neurologist, again, to treat epilepsy. So what you would look to do under that set of circumstances is to broaden your product line. And we heard an example of that yesterday in the Cyberonics presentation, where the description was made of the new seizure alerting device that they've got. And basically, they're going to go and broaden their product line with that product. So that's an example of that kind of reason why a device company would invest in a product for epilepsy. The second reason would be that the device company has a product or technology that they are selling to another community, another disease community or series of customers. And that product, with little or no modification, could be utilized to treat epilepsy. And we had another example of that yesterday with the folks from Visual Aids, for example. That product was first developed to ablate tumors, brain tumors, and they saw the opportunity. In fact, Joyce Kramer had a big piece from the Epilepsy Therapy Project of encouraging them to apply that technology to epilepsy treatment, and now it's being used, very well, and I'm sure will be used much more broadly. So that's an example of somebody that has an existing technology. We could put other companies in that category, but existing technology to apply. Those are really the only two reasons that an established medical device company is going to invest in epilepsy. If someone is going to invest in epilepsy that doesn't meet those two criteria, it's going to come out of the startup community. And when you look at what is it that interests venture capital to invest in a company, it becomes not epilepsy specific. It is what makes the best investment for the venture capital community to make. And so how does that decision get made? If they're going to invest in a medical device, there clearly has to be a large unmet clinical need. The company has to have a proprietary or unique approach that is going to solve the problem. Basically, it has to have intellectual property that will protect that approach. There have to be no freedom to operate issues that exist that would cause that type of an obstacle. They have to have the kind of scientific and technical capabilities that can demonstrate to the venture community that, in fact, they've got an opportunity to be successful with this venture. They've got to have a business plan that looks like it makes sense from the vantage point of how much investment is going to be required over what period of time to be able to get what kind of returns, and is this going to be, from a venture capital perspective, the best investment that that firm can make for a potential return to its investors. So they are very disease agnostic when it comes down to that type of decision. It has to be something that will stand on its own to be able to be the firm that gets that kind of investment, or the company rather, that gets that kind of investment as contrasted to everyone else. And then the other kinds of factors that come in there, in addition to the scientific and technical capabilities, what's the management team going to consist of? What's the regulatory path that you have to go through? What's the approximate length of time it'll take to get all of these kinds of things done? And then where's reimbursement? You know, is there going to be an opportunity for these products to get paid for? So when I think about NeuroPace and think about NeuroPace in the list of criteria that I just went through here, I was sitting there thinking that, my God, you know, NeuroPace was absolutely meant to be, because if you think about all the things that had to come together to allow it to exist, it was meant to be. And let me just take you a few down memory lane with that one. So now, 17 years ago, basically, I was approached by an individual that had developed intellectual property for responsive neurostimulation. And the idea was to treat neurologic disorders, epilepsy being the first, but the intellectual property, the patents had already been filed, and the concept was there. And to me, coming out of the cardiac world, I said, my God, you know, this really makes sense. Here you have an episodic disease, you know, the idea of being able to monitor brain activity and then deliver stimulation when necessary. This really makes sense. Let's look a little more. Large unmet clinical need, large market opportunity, order of magnitude, 400,000 to 500,000 people in the US that would have partial onset seizures that were not controlled with 1 or 2 seizure foci. Basically, the other kinds of criteria that you begin to look at, you say, well, gee, you know, this looks good. Where could we get some initial seed capital? And it just happened that at that point in time, I had just, I'd been a CEO of a cardiac defibrillator company. It had been acquired by St. Jude Medical. I was doing a short stint at Kleiner Perkins as a CEO in residence. I brought it to them. They said, yeah, this sounds like a good idea. So you sort of had that entree that allowed that to occur. And then we said, you know, we've got a lot of interesting stuff here, but we really don't have anybody that knows anything about epilepsy. So what am I going to do about that? Well, okay, we're on Sand Hill Road, which is in Menlo Park, and what's the nearest institution to around here? Gee, that's Stanford. Let me see if I can get a list of people that are, you know, who's running that epilepsy center over there? And I found out it's, by God, it's somebody named Martha Morell. So I call up Martha Morell and say, hey, you know, I've got this idea for something, and would you mind if I came over and had a little conversation with you? And so I'll never forget that day. I walked into her office and told her what we were thinking about doing, and she just looked at me. And I don't know if Marty's out here today. I know she was here earlier, but and she just looked at me, and I'm thinking, my God, what did I say that was so stupid that I'm getting? And all of a sudden, she slaps her hand on the desk and she goes, "We've been treating epilepsy patients for 20 years. Why didn't we think of this?" So, you know, we had one of the most wonderful leaders in the whole community that was right next door and then interested in working with us. And then it didn't end there, because she had already accepted the job to go to Columbia and run the epilepsy center there. And it turned out that 2 of the founders of the company, because we had to do proof of principle work, were located right in that upper New Jersey area, and the work that got done was in her lab with general purpose equipment that let us take through the early stages of financing to then be able to have enough of proof of principle to be able and then raise a venture round. And so that's what started our path, and then we developed an external version of our product that we tested in patients with an IDE that were undergoing intracranial monitoring. We then had, in parallel, we were developing the implantable unit. We conducted then 2 clinical trials, a feasibility trial, a pivotal trial, then went through the rest of the regulatory process. And 16 years after our first funding, we actually got approval of our first product and are just beginning to commercialize. And I would submit to you that having those particular events all come together and sequentially allow us to get to this point means that we were meant to be, that this wasn't something that just happened randomly. So with that, I'll just thank you very much for listening to me kind of go down memory lane a little bit, and look forward to participating in the panel.
Thank you very much.
So we're running a little behind, but I'm going to steal about 10 minutes from the break because we have such an extraordinary group here. I don't see anybody at the microphone, so maybe because we are tight on time, I will ask each of the panelists one question and let them expound, and then we'll call this particular session to a close. So starting with Santiago, how is it that we can get Pfizer back into the clinical arena of thinking about epilepsy? You have a long history with the companies you've acquired, at least, of being involved in epilepsy. Do you think that we need to adopt the Michael Gold model to get the next drug out?
We are very interested in ion channels. That's an area of strength in our research. So that brought the idea of epilepsy. And maybe the second point is that I am at Pfizer, and I am very interested in epilepsy. So it will always be important for me to be there.
One of the most compelling reasons to have you here, and thank you for coming. Roy, your company is no longer in epilepsy. If we had a differentiated compound, which has been the byword for this morning, if we had a differentiated compound that looked really good, would it entice Janssen to rethink this process?
Yeah, Roger, thank you. It's something I neglected to talk about. And so the perspective I provided was soup to nuts, right? And so do you invest in the discovery work and try to advance this forward, build an entire infrastructure that supports an epilepsy discovery and then early development, late development program. However, I would say every company here remains opportunistic, meaning that if something looks really, really good and attractive, of course there's going to be an interest in bringing that in. And for us, and I'll just set the bar for us where that is, and that relies not at NME, not at first in human, but at proof of concept. And that proof of concept, as I pointed out, was a proof of differentiation. I think right now it would be very hard to convince Janssen to invest in an epilepsy development program that is a commitment to phase 3 development without evidence of proof of differentiation. And so let me just leave it there. Then.
Okay. So, Mike, you've made the most provocative statements about how we might approach payers. I'd like to know, since I wouldn't know a payer if one walked in the back door.
That was obvious when you wouldn't pick a checkup last night.
That's correct. Although my wallet is considerably lighter. How do we actually approach it's a two-pronged problem. First of all, we have a ton of drugs stuck in the valley of death, which is pre-IND, okay? And those are, in my opinion, non-differentiable, except from mechanism of action. And usually we just know it's novel. And that's not what you want. I know, like you're shaking your head. But let's assume that we could get some of those through that process and get them out on the marketplace with a novel mechanism of action and good data. How would we negotiate with the payers how would Big Pharma negotiate with the payers to decide whether or not at this stage, because we can't differentiate earlier, we can differentiate in the clinic. That's what we always do. You pointed out levetiracetam, but it's actually true for all the drugs. All the drugs have been differentiated really in the clinic, okay, from phenytoin all the way up to retigabine, which has stubbed its toe, obviously. So how are we going to make that differentiation, and how are we going to find the payers and make that work?
So you pick up the phone and you call Blue Cross and Blue Shield, or you call the VA or Medicare, and they will come talk to you because they're interested in figuring out how they provide better services to their customers. So the payers are not shy. Okay, WellPoint is out there, and if you talk to European agencies, payer agencies, they'll be happy to come talk to you and share their perspective because they're vested in doing the best they can with the budgets that they have. From a sponsor perspective, there are a couple of things that come to mind. One is the notion of sharing savings. Okay, so at GSK, there was what was called the Asheville Project, where they were using Avandia to try to improve the care of diabetes. And they entered into a contracted demonstration project with Asheville that said, we're going to provide the drug for free. And there was a program where there was a control group and intervention group and looked at compliance, adherence, doctor visits, complications, hospitalizations, et cetera, et cetera, et cetera. And the agreement was, or the idea was, that the intervention group would show a lower rate of complications, healthcare utilization costs, and that the sponsor could share in those savings. And there's nothing wrong with that same approach with a treatment for epilepsy. If you have a drug that in routine clinical use really reduces the number of breakthrough seizures, hospital admissions, episodes of status, that has a profile that allows patients with I'm sorry, I keep on referring to them, I'm a physician, so they're patients to me. I understand politically it's persons with epilepsy. So if they're able to stay at work longer, do better at school, there's a health economics outcome that can be measured and you can share in that savings. So there are models out there, but they are not simple. They require a long-term view. They require, commitments from multiple parties. And I think patient advocacy organizations are probably one of the most effective, loudest voices to actually get those parties to the table. Okay.
Thank you for bringing that up to us. I think it's a perspective that we hadn't appreciated in that context. And I think the Epilepsy Foundation has to give serious thought to this issue. So the last question of this panel is to Frank Fisher. And Frank, one of the things that it's a more scientific question than the business question, but one of the things that I've always been intrigued by from the NeuroPace standpoint is the potential to improve it with time. As you gather data. Could you expand on that? Because I think that that's one of the most exciting aspects of the NeuroPace RMS program.
There's no question, Roger, that that opportunity exists. For those of you that aren't familiar with our system, the device is implanted, actually replaces a little piece of the skull. Leads are placed near the epileptic focus. The brain activity electrical activity is constantly monitored. When detection parameters that are unique for that particular patient are triggered, there are segments of the electrocorticograph that led up to the triggering of the therapy and then a post period that's programmable in terms of length that gets stored and is available on what we call our patient data management system. So this information resides in our system. The physician can access this from any place that he or she has internet connectivity, and the data is there for analysis purposes. And very candidly, when we started this whole thing, we thought we knew a whole lot more than turns out that we actually knew. And so we have been learning during this period of time as well. But as more and more patient data becomes available and more analysis becomes available to us to perform, there's no doubt that we will learn much, much more, and it will be to the benefit of patients, not only in terms of how our system can be optimized for controlling seizures, but what opportunities there may be because of its ambulatory monitoring capability to also feed back information to the physician in terms of drug therapy and the effectiveness of a different drug. For example, we've seen instances where there's been a patient on a particular drug, this is now in the open label period, the physician made a change in that drug, and the proxy for actual seizures is the number of detections. And you can see this just go down in the mud with the response. Clearly that patient has been having a fabulous reaction to that particular drug. Now this is something that normally would take God knows how long, 6 to 12 months just out in the clinic with the patient coming back with their seizure log and the rest of that kind of stuff. But within weeks, you could see that kind of activity. So not only will we learn more about the optimization of our system by itself, but the partnership between the drug therapy and the device therapy and how that works together synergistically, that's a whole world yet to go.
Terrific. Listen, I'm grateful for this extraordinary panel. I greatly regret that we have to stop because of time, but for all of you. My colleagues are probably going to be here for at least a few more hours, so please avail yourselves of these phenomenal experts in their field and ask them questions individually. Thank you very much for coming.
So all of you are familiar with bank error in your favor, right? If you guys look at the break for today, it says 9:55 to 10:20, 15 minutes. I think that's a bank error in our favor. So that being the case, we're not going to worry too much about running a few minutes late. And I am going to once again invite Rajesh Ranganathan to come and talk to us about how the NINDS fits into making sure that we're developing the best new therapies.
Thank you, Jackie. I've lost my voice overnight. It was the party that Steve threw in his hotel room. You get a few drinks into him, and man, he can do a mean air guitar. Have you ever seen this? Anyway, so I'm not sure what's So I think there's a quote from some business leader somewhere that more business gets conducted during the informal sessions than during the formal sessions. So I'm almost tempted, given my voice, to yield the 10 minutes back so that the folks can actually interact during the break, if that's okay with the chair. All right, so let's just break.
You have 20 minutes. 20 minutes. 10:20 on the dot.
Welcome back, everybody. My name is Steve Schachter, and I have the privilege of being the host for the next session, which is called Clinical Stage Drugs. Yesterday, Roger Porter talked about the Valley of Death, and here we are now. We're going to be presenting 12 products that have walked through the shadow of the Valley of Death and, having feared no evil, are now in clinical stage. And so we're quite excited by that. The good news is we have 12 clinical stage products to talk about. Unfortunately, we only have 2 hours to do it. So I'm going to try to keep our speakers to their 10 minutes of allotted time. And hopefully they'll finish a bit earlier than that so we can have a question or two. So let's then proceed to our first presentation by Emil Kakas, who's CEO of Ultragenyx Pharmaceuticals, to talk about triheptanone.
Thank you, and I want to thank the organizers and Jackie for inviting us to talk about triheptanone today. I'm the CEO of Ultragenyx. It's a company that's just, oops. Can we put it in presentation mode? Green should push it forward, but it's pushing it out. What? I am. Well, all right, one more time. There we go. All right, we're building a rare disease company that focuses on therapeutics, both biologic and small molecule for rare diseases. We founded the company in April 2010, just up the way here across the Golden Gate in Marin County. We have 5 programs now already in phase 1, 2. 3 of them already generated positive phase 2 data. We've managed financing well. We've gotten actually through an IPO, and actually at the end of Q1 had $165 million in cash, which gave us the reserves to actually drive ahead a lot of development. So I can understand the terrible situation, some of the great molecules that were here yesterday in that valley of death, but you can get through. We managed to get there. We're now 84 employees in Novato. Our pipeline, just to touch on, consists of biologics, including a couple for connective tissue type disorders, Karen23 for XLH and recombinant human GUS for MPS 7. Those are not in the neurological space, but on the bottom there is the triheptanone program and salicylic acid extended release. The SAR program is for hereditary inclusion body myopathy, and some of you neurologists may have patients with HIBM, and that showed positive data, 48-week data in December, and presented earlier at AAN. The triheptanone program is the one I want to focus today. It's a substrate replacement therapy that is basically an engineered triglyceride that delivers energy into the mitochondria in a way that bypasses some of the defects and restores some of the dysfunction that occurs in mitochondrial and other disorders. The fatty acid oxidation defect we won't talk about today, but are diseases that have a defect in fatty acid oxidation that result in a series of problems, including muscle problems such as rhabdomyolysis, liver and heart problems. Now, I'll talk today a little bit more about triheptanone for glucose transporter type 1 deficiency or GLUT1 deficiency syndrome. It's, the substrate replacement concept here is in this disease is based on the fact that they are missing or inadequate levels of energy, and the idea is that this triheptanone has a way of replacing the missing substrate. So let's talk about GLUT1 first, and we'll talk about the mechanism of triheptanone. GLUT1 deficiency syndrome was originally thought to be an extremely rare disorder discovered by Dr. DeVivo, and over the last few years it's become clear that it is much more common than realized, with a rather pleiotropic set of phenotypes that are possible. Disease is all caused by a defect, an autosomal dominant defect, that is a 50% loss of the glucose transporter. If you have no transporter, you do not survive as an embryo, so all the patients have at least 50% of function. And when you don't have enough glucose, of course, the preferred substrate for the brain, you end up with seizures and epilepsy that's usually refractory or very difficult to treat with traditional AEDs. Patients often have cognitive developmental delay that can be progressive over time, and movement disorders commonly, a variety of different types of movement problems. All of this is basically due to the lack of glucose transport in the brain. In the PET scan there, just showing you some work of Dr. Pascal and Dr. DeVivo on showing the difference in glucose uptake in the brain of patients with GLUT1 versus normal. The prevalence now is thought to be 3,000 to 7,000, and this is coming from some surveys looked at idiopathic generalized epilepsy, where they sequenced patients consecutively and discovered perhaps about 1% of all idiopathic generalized epilepsy is in fact GLUT1 deficiency, undiagnosed. Another study looking at early onset opson seizures found that 10% of their clinic actually had GLUT1 deficiency syndrome as the cause of early onset opson seizures. There is a number of other phenotypes, and seizures being one of the common ones, but these patients have very complex seizure patterns. They can have multiple types of seizures in the same patient. Now, I'm not a neurologist. My father was a neurologist, so I grew up with neurology at home, but these patients have an interesting complex pattern of seizures. So the current standard of care for GLUT1 has been, there are really no currently approved specific drug therapies, and traditional anti-epileptic drugs are relatively ineffective in these patients. Many of them do take one or more AEDs, but they usually are not well controlled. But the ketogenic diet has been shown to be very effective in controlling seizures, and this involves a diet of 70–80% calories as fat. I think it's been talked about yesterday. And it induces an alternative energy source, the concept of delivering ketone bodies, although, as was discussed yesterday, we don't really know if that's the whole story, that's part of the story. Is there an effect on transmitters, neurotransmitters, or other things beyond energy? I think those are some pieces that are not known. But the concept is ketone bodies get across the blood-brain barrier, and so that block to glucose is bypassed. While it's been very good in seizures, it's clearly not been optimal in developmental delay or cognitive problems or in motor dysfunction, and therefore there's clearly room for improvement beyond that. And KD compliance can be difficult. It's an extreme diet, and there are other versions of modified Atkins and other things that people are looking at, but clearly it's a demanding. And there may be problems with a high-fat diet, and a number of complications of that diet can also be, have been described. So the question now, what is another way to do it? And triheptanone has an alternative strategy. Triheptanone is a triglyceride of C7 fatty acids. So these are odd-chain fatty acids. Your body has even-chain fatty acids, usually C16, C18. These are C7, so they're medium chain and they're odd. And that causes something very important in how they get metabolized in the body. What happens with triheptanone is once it gets into the gut and into the liver, it basically gets broken down into heptanoate. And the liver will further metabolize it into 4 and 5 carbon ketone bodies, regardless of whether you're on a ketogenic regimen or not. The medium chain will get broken down specifically, and so they make ketone bodies regardless of whether you're fed or fasted. The heptanoate will be released to the bloodstream as well as 4 and 5 carbon ketone bodies. The heptanoate itself actually can diffuse directly into the brain, and the ketone bodies also get across via the MCT1 transporter. So really, triheptanone provides 5 different energy substrates to the brain. And what's been studied in the mouse model of this is, in fact, that this can actually help improve the metabolism, both in restoring the function of the mitochondria, but in actually allowing gluconeogenesis in the brain as well, and I'll show you that in a moment. So when you look at how triheptanone gets metabolized, it basically gets transports across the mitochondrial membrane by diffusion itself. The medium chain enzymes then clip it into acetyl-CoA and propanil-CoA, two compounds that enter the TCA cycle. Now, acetyl-CoA is what you normally get from fat. But the unusual piece is that the odd chain leaves you with a 3-carbon unit, which allows you to recharge the Krebs cycle. The Krebs cycle gets deficient in hypoglycemic disorders because the gluconeogenesis process drains the Krebs cycle of intermediates, and the need for neurotransmitters will do that as well. And so providing the 3-carbon unit allows you to recharge the Krebs cycle, and as you can see there through the malate cycle, allows you to generate glucose in the brain or anywhere else. 1 molecule of triheptanone actually can be converted then to 2 molecules of glucose. Fat normally cannot be converted to net glucose, but triheptanone can. So the gluconeogenesis component, as well as recharging the Krebs cycle, are part of the mechanisms behind triheptanone. And I won't have time to go through all the details. This is a very nice article by Marilyn Valencia in 2013 that showed through a triple metabolic labeling that, in fact, the heptanoate both is going to the liver, getting converted to glucose, as well as other substrates, ketone bodies. And the ketone bodies, as well as heptanoate directly, are getting to the brain, and that gluconeogenesis is going on in the brain, including the production of glutamate and glutamine. And the glutamine production suggests that the glia are actually dominant in the metabolism, but there is clearly C13 glucose getting taken up by the neurons as well. Some very nice work showing how heptanoate might be able to fix the problems in GLUT1. Now, There's already been some data previously not published, but described in a few patients with GLUT1 that showed a significant improvement in seizures and cognitive function. Those anecdotals have not been published yet, but we're hoping to see them soon. We're conducting a randomized control study of GLUT1 patients. These are patients who are failing the ketogenic diet and cannot have breakthrough seizures. We're not asking people on ketogenic diet to be taken off. If they're on it, they're stable, they should stay on it. But if they can't do it and they're failing and they have seizures, they can enter the study. If they meet the seizure baseline rate of 4 per month, they're randomized then trypnoid or control oil. They're continued then for 8 weeks, and the seizure baseline rate is compared. They're also looking at cognitive function as well as motor function in the study, and then they go on to an open label. That study's up and operating. If you have patients that might be interested, please let us know. We're also supporting a number of investigator programs globally, looking at combination of trypnoid with ketogenic diet in GLUT1 patients, looking at motor dysfunction issues and the other symptoms and manifestations of GLUT1 deficiency syndrome. There's also some work going on in refractory epilepsy. So we're doing support for these programs. If you have other interesting ideas, we're certainly open to looking at those. One last piece, GLUT1 diagnosis. We think it's very much underdiagnosed, and not enough neurologists need to be paying attention to it. The diagnosis of low glucose in the CSF is certainly what Dr. DeVivo would recommend you do. But in addition, you need to be looking for and doing the sequencing, complete sequencing, and we're now establishing a free sequencing plan with medical neurogenics, which can help, and we're going to be rolling that out shortly. That will help us diagnose patients who have refractory seizures that have turned out to be GLUT1 and help better understand the GLUT1 population. And from the initiatives I've been hearing today of the epilepsy group, it seems like we would need to get connected up and make sure we're working closely with those existing initiatives on identifying, understanding GLUT1. That's it from me. Thank you for listening.
Thank you. Perhaps time for one question if somebody wants to approach a microphone. If not, given the complication of the cognitive delay, when, in your opinion, would be the ideal time to start therapy in a patient with this disorder?
It's definitely early. I mean, I think the data already Dr. DeVivo has with KD and treatment is early treatment is really essential. And this is why we need to be thinking about this in young children with new onset seizures, infants essentially. They need to be thinking about this diagnosis and getting on it because early treatment can have an important impact.
Okay, thank you. Thank you very much. Our next speaker is Dr. James Casella from Alexa Pharmaceuticals to talk to us about inhaled alprazolam.
Good afternoon. Thank you, everybody. And I apologize in advance because I have the throat thing that's going around. So, I'd like to thank the organizers and Jackie in particular for inviting us back this year to talk a little bit about our inhaled alprazolam program. We are looking at acute repetitive seizures. I think it's preaching to the choir here, but basically these are seizures or seizure clusters that occur in patients with epilepsy. They're not typical to their usual seizure pattern, and these are what you can be considered to be breakthrough seizures. What we've looked at in the literature is, and for treatment options, benzodiazepine seems to be the drug class of choice. There's only one therapy available today for the treatment of acute repetitive seizures, and that is with a rectal diazepam gel, Diastat. It is approved, but as you look at the package insert, you can see that there are features of this route administration and the drug itself that may be less than optimal for this patient population. One of the things that we've noticed in the literature is that there are a number of drugs under investigation for the treatment of acute repetitive seizures, all benzodiazepines, primarily through the intranasal route administration. Those drugs include diazepam, midazolam, clonazepam, lorazepam. So we're looking at alprazolam. But before we get to alprazolam, I'd like to give you a little bit of background on Alexa and what we do and what our technology is. So we've developed an inhalation drug delivery system called Staccato system. It's a very simple system that takes solid drug and delivers it to the deep lung. The purpose is not to treat the lung, it's to deliver drug into the bloodstream. So we've developed a system that is a single breath actuated system that delivers drug. It takes very little effort by the patient. It is very intuitive to use, and unlike other types of inhalers, there's no coordination required because you simply breathe through the device in order to deliver the drug. We take excipient-free drug. So in this case, it's pure alprazolam. We spray coat it onto what we call a heat package, and when the patient breathes through the device, aerosol is generated in situ. So we're transforming a solid drug into a fine aerosol particle. The reason we're doing this is we're creating aerosol particles that are optimal for deep lung delivery so that we can get that drug, those drug particles directly into the bloodstream via the lung. And this is via the deep lung. With that, we have characteristics with all the products that we have using the Staccato system where we have IV-like pharmacokinetics. We have rapid absorption into the bloodstream, and we have very high bioavailability of the dose that comes out. We actually have one product that is approved in the US and in Europe using the technology. It's called Adasuve, and it's an inhaled form of loxapine, which is used in treating acutely agitated patients who have schizophrenia or bipolar disorder. So it's a product that's used in an emergent care situation. Because the technology is really important here in delivery of the drug, I wanted to go into a few of the details of how it actually works. So if you look at what we see here on the screen, there are a couple of various components to it. There are some electronics and a battery in it. Mostly the electronics are involved in sensing the patient's breath. So when the patient is breathing through the device, there is a trigger in there that when it reaches a certain airflow, which is not much more about what we're doing right now, sitting at rest, it will cause a electrical circuit to actuate and start the reaction. The drug is coated on what we call a heat package. It's coated by basically spray coating the pure alprazolam on there. Solvents evaporate off and we're left with a solid drug film. And when the patient breathes through the device and actuates it, the heat package initiates a heating reaction. So we're getting the drug on the surface of that heat package up to about 350 degrees Celsius. It rapidly vaporizes that drug, forms the particles that we know are important, the size are important for getting into the deep lung, and those are pure alprazolam particles. If you look at these pictures, which are taken from some high-speed photography of the actual vaporization process, you'll see in the upper left-hand corner at time 0 before the patient has initiated the reaction, there's drug coated on the stainless steel substrate. 30 milliseconds after the patient has breathed through the device, you can see the cloud starting to form. That cloud are thousands of aerosol particles that are 1 to 3 microns in size. By 50 milliseconds, you can see that most of that drug has been vaporized off and is in the airstream that the patient has generated. And by 200 milliseconds into the process, that patient has totally vaporized that drug, and that drug is now in their deep lung. Because we have such rapid generation of the aerosol, and we have particles that are optimal for getting it into the deep lung, we have rapid delivery into the bloodstream. So this is data now from our phase 1 study and from our phase 2 study in non-epilepsy patients, where we show that the plasma time concentration over 2 different doses. The important point to note here is that when you look at the time scale, you're seeing that we have a rise to maximum concentration within 2 minutes. That is dose independent. So this is a hallmark of the technology. We not only see this with alprazolam, but we see this with all of our other compounds as well. So again, by generating these types of aerosol particles, we get the drug into the bloodstream very quickly. Just for a perspective, and just as a put it in relationship to the other drugs that are being developed for ARS, you can see down in the lower corner, the staccato delivery of alprazolam produces a maximum concentration within 2 minutes. Just to look at some of the other drugs given by intranasal administration, again, these are from literature papers, and we can provide the references if you need them, but they're all out there. Midazolam, lorazepam, diazepam, clonazepam, with the studies that are available in the literature, have a variety of time to maximum concentrations. As we noted earlier on our first slide, if you look at the rectal gel, of course, the time to maximum concentration in the bloodstream is an hour and a half. So for people that are having repetitive seizures, we think it's very important to have rapid onset of effect. And here we're clearly showing that we can achieve rapid concentrations in the bloodstream. We know from studies in phase 1 and in phase 2 with inhaled alprazolam that looking at pharmacodynamic measures such as sedation using visual analog scales, we see maximum effects within 2 minutes. So the PK seems to be following, or the PD seems to be following the PK. So just as a final slide on the update of where we are with staccato alprazolam for acute repetitive seizures, we have an active IND. We've studied we've had 3 different studies to date with about 100 subjects and patients studied, all showing that the drug is safe and well tolerated. We are planning, in the late stages of planning, a proof of concept study in epilepsy patients. We're going to use the intermittent photostimulation model to determine an active dose range of alprazolam. We're initially going to look at 3 different doses. We'll establish the photosensitivity range and then look at the suppression and abolishment of that effect with the various doses of inhaled alprazolam. And we expect to get that study started probably within the next quarter. And I'd like to take any questions that you might have.
Thank you very much. Any questions from the audience? Jim.
It wasn't clear if this device is intended to be a single dose or multiple dose product.
So the device that we have in development is a single dose product, as is our commercial product that we have with Adasuve. We do have a multiple dose product capability, and we have another product line, but our intention here with this would be a single dose product.
And it's hard to tell from the component parts. Would you anticipate that circuitry and component parts would represent a substantial cost?
There's costs associated with it,
Sure.
but electronics are very cheap these days. So I think we can be competitive with what's out there for current treatment, for sure.
Thank you.
Just, I'm trying to understand the sort of safety profile. So if you're getting Cmaxes and Tmaxes that look like IV, generally patients are dosed IV in a hospital setting because of the rapid sedation. So can you help me understand how somebody in an ambulatory setting would be using this and not put themselves at risk? Let's say they're driving or walking or doing something, operating, I don't know.
So obviously the concept here is that for acute repetitive seizures, we will be treating post-first seizure. So the patient will be using it. They've had a seizure, so they probably aren't going to be driving. One of the things that we're looking at and what we plan on doing with our dose ranging study is to look at the effect on the EEG pattern versus the sedative profile of alprazolam. I mean, one of the reasons why we chose alprazolam is that while all benzodiazepines are sedating, we see less sedation in healthy volunteers. We see less sedation we were in a panic population before. So that's one of the things that we really have to do in the first study is to look at the changes on the EEG pattern versus what we'll be measuring, which will be sedative parameters such as VAS scores and things like that.
Great, thank you.
I apologize, but we're going to have to cut it here, and you'll be available afterwards for further questions. Thank you.
Thanks.
Great. Our next speaker is Alice Mead, who is with GW Pharmaceuticals to talk to us about Epidiolex in pediatric epilepsy.
Good morning, and many thanks to Jackie and the other organizers for giving us a chance to update you on our progress. So I don't need to tell you that there's a great deal of public interest in the use of CBD in the treatment of childhood epilepsy. It seems like daily the newspapers are presenting the cases of desperate families that are trying to change their state laws or travel to Colorado to obtain preparations. Dr. Sanjay Gupta profiled a particularly dramatic case in his CNN program, and an excellent survey was published by Drs. Jacobson and Brenda Porter, and I recommend that to your reading. So there's a growing body of anecdotal evidence, but there are no rigorous clinical trials or clinical evidence, and that is what GW intends to provide to physicians and to patients through our development program. So we are not just a Sanjay Gupta opportunist. We have been thinking about CBD for a very long time. We were founded 16 years ago in the UK to develop a range of prescription medications that are derived from the cannabis plant. And way back then, we thought that CBD had significant therapeutic potential. So in fact, it forms half of the principal cannabinoids of our first product, Sativex. And because of the development of Sativex, we have amassed a large body of preclinical and clinical safety data. So that is already in place. About 7 years ago, we began more intensively to conduct preclinical research into CBD and other non-THC cannabinoids in a variety of medical conditions in a number of models. And in epilepsy, 2 product candidates emerged, CBD and CBDV. But actually, fast forward just a bit, US physicians urged us to take the CBD and develop a formulation for children and to conduct a US development program because they were increasingly concerned that their patients were being exposed to different vernacular preparations of uncertain quality and safety. So hence Epidiolex was born, which is more than 98% pure CBD in a liquid formulation. We are also pursuing the other candidate, CBDV, which is a structurally related molecule with a different side chain. It shares some mechanisms with CBDV, but it has different safety profiles and efficacy profiles in different mouse models. It shares, as I said, some mechanisms, but it hits different ion channels and different enzymes. So we have already completed a phase 1 in CBDV, and we intend to begin a phase 2 sometime next year. So the problem with most of the CBD products in the US are that the composition, the quality, the consistency, and the safety are unknown and quite questionable. And increasingly, there are reports that many of these products may be contaminated with heavy metals and pesticides and contain significant levels of THC. GW conducts all of its cultivation and manufacture under GMP in accordance with strict standard operating procedures and quality control processes. We conduct all of our cultivation by clones in organic medium in glass houses in the UK where every aspect of the cultivation cycle, which is 12 weeks, is computer controlled. We extract the active ingredients, and in the case of the CBD extract, we put that extract through additional purification processes to remove the THC. We remove the THC because we think that the very young brain is different from the adult brain. The very young brain, if constantly or chronically stimulated by THC, that may lead to untoward, unintended, and just a whole variety of consequences that we don't fully understand. But we do know that the very young brain is undergoing rapid neuronal development, and there's a growing body of evidence that THC can inhibit and distort that neuronal growth. There's also evidence that THC in some models and in some animal species is proconvulsant. So we remove the THC. We think cannabinoids offer an exciting new class of AEDs. The endocannabinoid receptor system, or ECS, is sometimes called a supermodulatory homeostatic mechanism that achieves balance in the body by a variety of mechanisms. Cannabinoids don't have a single target or a single mechanism of action. One possible target would be that the ECS can inhibit the release of excitatory neurotransmitters. And CBD modulates the ECS and other receptor systems and non-receptor mechanisms. Inflammation is important in the development of epilepsy. There are no medications that currently target the inflammatory processes, but CBD is a potent antioxidant and anti-inflammatory molecule. It also inhibits the release of pro-inflammatory cytokines. It's clear that ion channels play a role in the development of epilepsy, and CBD and other cannabinoids modulate the flow of cations and in particular intracellular levels of calcium. The GW program is moving quickly. We've secured orphan designation for Dravet syndrome and Lennox-Gastaut syndrome for Epidiolex, and just today we announced that we had secured fast-track designation for Epidiolex. We will also seek breakthrough designation and other accelerated pathways. We have two prongs to our program. One is the GW-sponsored placebo-controlled clinical trial program, and the other is a series of physician-sponsored INDs under FDA's expanded access regulations. Those began with individual patient INDs, then 5 intermediate-size INDs were approved for 125 patients. Now 300 patients have been approved in 12 sites to receive treatment with Epidiolex for a variety of epilepsy conditions. The first 2 of the intermediate-sized sites began in January, and we expect to release data at mid-year, which is almost here, so stay tuned for that. The GW-sponsored program is, as I said, moving well. We've opened an IND with FDA with a phase 2/3 protocol in Dravet, and we intend to begin the first part, which is a PK safety study in the second half of this year, with pivotal studies in Dravet and LGS to begin in early 2015. It should be noted that CBD is a Schedule I substance under the Federal Controlled Substances Act. It doesn't matter whether it comes from hemp, a laboratory, or a THC-containing cannabis strain. And so each research site must have a DEA Schedule I research registration. The expanded access program, the physician INDs, are also going well. There are over 600 weeks of exposure and a range of continuous exposure of up to 60 weeks. Only 1 patient has been withdrawn for lack of efficacy, and this slide gives you the range of epilepsy conditions that are being treated in those expanded access INDs. We think that this will generate a very interesting data set, which may guide us in future development programs, and we hope will be of interest to the scientific community. This just gives you an overview of the timelines of our program. As I said, we expect to begin the first part of a phase 2/3 protocol in the second half of this year in Dravet, with pivotal studies in Dravet and LGS to begin in early 2015. The expanded access INDs will continue until Epidiolex is approved by FDA, and we will be making data communications at regular intervals. Finally, CBDV, having finished phase 1, we will open an IND with FDA with a phase 2 protocol later this year and begin that phase 2 study in early 2015. Thank you very much for your attention.
Thank you. So we have 2 minutes for questions. First, Dr. Katz.
Yeah, do you anticipate any problems with enrollment in your controlled trials for Lennox-Gastaut and Dravet, given that you have expanded access programs and some of the patients in those programs have those conditions?
Some do, but there are a very large number that actually don't have Dravet or LGS. And as the new expanded access programs open, they're trying to reserve some of their LGS and Dravet for our clinical trial program. We collaborate, so you know they don't operate completely independently.
Any other questions? Can you tell us about the bioavailability of the liquid formulation?
Well, you know, bioavailability, I may not be the best person. I wish that Dr. Guy could be here, but he was detained in the UK, and I'm an attorney in a sea of scientists. But you know, the oral formulation is probably not as bioavailable as an inhaled or an IV formulation, but it's still for children. For chronic conditions, this would be better.
Any other questions? If not, thank you very much.
Thank you.
Our next speaker, Dr. Stephen Collins, CEO and President of Insero Health, to talk to us about huperzine.
Well, thank you very much. It's quite an honor to be here. It's nice to see such a large group of people with interest in epilepsy. Insero Health's mission is to develop drugs for conditions which don't currently have any effective therapies, and in particular, conditions which have catastrophic endpoints. So we have a philosophy of trying to help those who need the help the most, and the way we're trying to pull that forward is by taking traditional medicines found around the world and improving them. The benefit of this, of course, is many of these medications have hundreds of years of safety data. And as opposed to trying to develop medications on a new target where you unfortunately find numerous side effects for patients, we believe this will help decrease the chance for safety problems. So just briefly, I'll go to an overview of the company. Our focus, as I say, is in rare and catastrophic diseases, so-called orphan diseases. our first indication for our lead program will be in Dravet syndrome. The lead compound is a synthetic form of a medication which has been used in China for hundreds of years, primarily for the elderly to help cognition as well as in pain and inflammation. The company is a clinical stage company. This asset has been in phase 1 studies in adults with refractory CPS. And our next stage will be to develop a pediatric-friendly formulation which will be used for children with Dravet. We have a team that's developed multiple medications in this space before, and we have good IP and other protections. Clearly, what we'd like to do is move this along and partner it with a company that would be able to take it all the way forward. Just briefly about the company, the management team is the same team that developed in the United States vigabatrin for infantile spasms, clobazam for Lennox-Gastaut, Depacon, Depakote ER, et cetera, et cetera. Scientific Advisory Board has some unknown people like the guy to my left. Stephen Schachter is the chairman and the scientific founder of the company. That was a joke, Steve. Roger Porter, ex-president of AES, and obviously well known to this crowd, as well as Steve White from the ASP. Warren Lambert and Sam Reich are the 2 other board members in addition to myself. Well, the highlights, I'm not going to go through all of this. The bottom line is this molecule has been around in China for hundreds of years through the ASP program, and I'll show those data in a minute. It has shown tremendous efficacy. The potency is one of the most potent drugs the ASP has ever screened. As an example, it's 57 times more potent than Keppra in the 6 Hertz model. The differentiator here is that this drug aids in cognition, and as those of us who are either patients or physicians who work with patients with epilepsy know, the majority of the medications, in fact, are anything but cognitive enhancing. So we have a way of manufacturing this material from a completely synthetic method, and we have the intellectual protection based on that synthetic method. We've already demonstrated that we're able to manufacture up to 50 grams of this highly potent compound, and so we're poised, ready to make an extended release pediatric formulation, juvenile tox studies, and then a second IND, this time for Dravet. We hope that will happen in 2014. Excuse me, late 2014, early 2015. This is some of that, quote, ASP data, and what you can see in the green is that at the classic step of evaluation, 32 milliamps, INS-001, which is huperzine, is far more potent than Keppra. And in fact, Keppra has basically no efficacy at the more impressive stimulation of 44 milliamps, and yet INS-001 is still quite potent in a range which humans have actually experienced before, a dose range. So as I said before, our lead indication is Dravet. I hardly need to discuss Dravet with this audience. Clearly, huge unmet need, and while I hope that many of the therapies people are talking about here will help some children with Dravet, the unfortunate reality is it's highly unlikely any one medication will help all the kids with Dravet. So for a bunch of complicated science reasons I won't get into, we had a belief that huperzine would help in Dravet, and so with the help of Dr. Andrew Askai, the very generous help of Dr. Askai at Emory University, using a genetic model of SCN1A, that is Dravet, we were able to show that at very low doses, we were able to obtain 100% control of seizures. Now, it's a little tough to see, but all the stars at the bottom are animals that have no seizures. And so the majority of animals actually had no seizures in a dose range if you scale it up to humans, which would be quite practicable. So the summary is this is a clinical stage company with Harvard roots, ergo Ivy League. We have an experienced science and development team to pull it forward. We have a validated lead compound that is over 400 publications on the pharmacology, the preclinical safety, and hundreds of years of safe use in China. It is a novel mechanism, very clearly one that can be used for epilepsy, pain, and inflammation, but has not yet been. We have IP around manufacturing methods, as well as formulation, et cetera, as well as a pending orphan status. In summary, Insero Health is poised to take this asset into kids with Dravet as soon as we can. Thank you.
Thank you. We have time for a couple questions. from the audience?
Just want to send checks in, that's okay too.
Andy?
Steve, this isn't meant to be a nasty question, but I'm curious. Is there any evidence from the Chinese experience that kids with epileptic encephalopathies, or even Dravet's more specifically, have had a benefit?
No, it's a very good question. You know, the Chinese literature speaks in general terms about this, but there haven't actually been studies. We did our synthetic manufacturing with Wuji, one of the premier manufacturing companies in China that supplies for Glaxo, BMS, et cetera. And we have a Chinese partner that we're in diligence with to be able to potentially go forward in China. Anecdotally, we've spoken to some docs in China and they say, oh yeah, I've used it. The reason it probably is not getting used is one I didn't get into. This is very bioavailable. If you take one of the capsules now, you'll get a serum peak almost instantly. And at very high dose, you'll have nausea and may even vomit. That's why this needs to be an extended release. You could go to GNC and buy this stuff, but the reason you probably wouldn't use it in your child is that you'd have to be giving it every 2, 3, 4 hours. It's very rapidly cleared through the body, and that's probably why. Now, in cognition in China, it's used twice a day. It probably would be much better if it was an extended release formulation. Thanks.
Another question, please?
Can you speak to potential utility in seizure types outside of Dravet, and then conversely, your thoughts about your IP position in those seizure types?
So to take the last question first, we have a method of use patent around seizures and pain that's worldwide. To get to the other question, the ASP screen would indicate this should work in refractory complex partial seizures. It also works in the MES model that would help explain potentially to help progression of seizures, that is to say spread of an individual seizure. It does not work in PTZ and some others. So my guess is this is not going to be a drug like valproic acid in which you get effect on absence seizures, drop seizures, tonic seizures, etc. Drug-drug interactions, glad you asked. Probably not going to have significant interactions.
That was my next question.
Probably not going to have significant interactions with stiripentol, clobazam, et cetera. Can't exactly answer that right now. And so add-on trials in Dravet would obviously be adding on to those medications. Looks like it's going to be okay. Thank you.
Great. Thank you. Thank you, Steve.
Thanks, Steve.
Next presentation will be from Dr. Gail Farfel from Marinus Pharmaceuticals to talk to us about ganaxolone.
Thank you, and I'd like to thank the organizers for welcoming us back again this year. Let me make sure I can. Right. I'm going to focus on what's new with Marinus since the last time I presented 2 years ago. Most importantly, we have a new CEO, Chris Cashman, who I think I saw in the back somewhere, and a new CFO, Ed Smith, and all now located, we have a new office in downtown New Haven, Connecticut. We have, since the last time I presented, two of our patents in the US and Japan, Australia, New Zealand, a few other countries they're rolling out, have been issued for covering our nanoparticulate composition, which is very important, as you may remember from previous talks, in the bioavailability and managing the fed-fasted effect for this preparation to make it into a compound that could be approvable with the right studies. We also have issued our synthesis patent, which is also important, and so this constellation of patents will provide protection for ganaxolone until 2026 or later, which has really reinvigorated the company. For the remainder of the program, I'll talk mostly about our epilepsy development program. However, we also have proof of concept studies ongoing. We have one in a pediatric population, 6 to 17, in Fragile X syndrome. And then we have a proof of concept study in PTSD that is nearing completion. So it has been very gratifying throughout this meeting to hear the talk about GABA. There wasn't much talk about GABA for a few years, and I felt pretty alone. Especially the information about the interneuron therapy this morning was very exciting. How does ganaxolone work at GABA? It's part of the class of neurosteroids, the positive allosteric modulators, which are metabolites of cholesterol and by way of progesterone. What's important about allopregnanolone is that it is metabolized from progesterone and can be back converted to an agent that can be active at the nuclear progesterone receptor. So ganaxolone was synthesized originally by Consensus in the 1990s with a methyl group at the 3-carbon position, and that prevents the conversion to an entity that is active at nuclear progesterone receptors, and thus we are developing it for chronic use. So just to go a little bit into the chemistry of neurosteroids and these extrasynaptic GABA receptors, I think we've touched on them a few times already this meeting. The synaptic GABA receptors are the ones, the darker blue, and then the class of extrasynaptic receptors are the lighter blue that are postsynaptic outside the cleft, appear to differentiate in terms of their availability, particularly in certain seizure syndromes. And with the lipophilicity of allopregnanolone and by extension ganaxolone, there is a diffusion into the postsynaptic membrane that provides this constant low-level tonal modulation that provides a GABA signaling that is different mechanistically from the synaptic signaling. And so we believe is the basis for the difference, particularly with the benzodiazepines, in terms of the lack of observed tolerance that we have seen. In terms of development, ganaxolone, as many of you know, has been in development for a number of years now. We have 4 open INDs in both adult and pediatric indications. We've completed our preclinical IND-enabling and preclinical testing package with the only exception of the 2-year carcinogenicity studies. And what is most interesting about the drug is the clean safety profile that we see in this testing. Safety pharmacology, we don't see any end organ toxicity that's been identified. The genotox studies, similar. The reproductive toxicity studies were clean. There was no findings in embryo-fetal development, no postnatal findings, no fertility findings. And then the chronic toxicity that's been completed to date, the 1-year dog studies were also clean. So very, very encouraging profile for a drug that could be used to treat adult populations as well as pediatric populations. It is metabolized by CYP3A4. It's a common target that we have all managed, I think at one time in our careers or another. Ganaxolone in a midazolam probe study was shown not to be either an inducer or an inhibitor itself, but of course interacting with common CYP3A4 drugs, and the most obvious for an epilepsy meeting are carbamazepine and phenytoin. There would be changes there in ganaxolone levels. Now, the next 3 slides are data that I have presented previously. I want to remind you of what we have accomplished in the program. I am going to go through them quickly so that I can talk about our new study. And I understand they're not available on the app yet, but they will be up later today. There are 3 studies to discuss. A phase 2 study in drug-resistant partial onset seizures in adults. That was presented last time. 147 subjects, 98 were on ganaxolone at 1,500 milligrams a day. And there was a significant difference detected between the mean seizure frequency change in the ganaxolone patients relative to the placebo patients. The mean difference was almost 20%. The median difference was 16%. And then the data in the figure on the lower right represent the 50% responder rate. We did have an open label extension to this study, excuse me, and these data were presented at AES a few years back, and the findings from the open label extension supported these findings, and the mean changes in the group that went out to 26 weeks were approximately 25% improvement, with almost 40% improvement at the end of 1 year for those subjects who were still in the study. The safety profile from the study is shown in this slide. And I'm not going to try to work with the pointer, but dizziness, fatigue, and somnolence were the only adverse effects that were noted at an incidence over 5% that were higher in the ganaxolone group compared to the placebo group. Everything else in terms of AEs was actually the same or higher in the placebo-treated patients. Notable in this study and the open label extension and the program to date is that we haven't seen any findings on ECGs at all. the occasional individual, but nothing that indicates a trend, nothing that is population-wide, and nothing that's persistent. We haven't seen changes in hematology. We haven't seen changes in vital signs. The clinical picture really does support what we saw in the early pharmacology studies. These are data from a publication in Epilepsia from 2000 that many of you are probably familiar with. This was the pre-surgical model, so it is, in a sense, a proof of concept study of ganaxolone monotherapy. These were 52 subjects, half treated with ganaxolone, 1,875 milligrams a day, and half treated with placebo. And the Kaplan-Meier survival curve, the ganaxolone is the dark line on the top, excuse me, shows the subjects who survived the 8-day treatment period without meeting the criteria for discontinuation due to emergence of seizures at the end of the period. So with all that, it gets me to where we are now. And after talking with many of you after this meeting and at many other meetings, the question was, well, with a profile such as yours, would you get enhancement of efficacy if you ran a higher dose of ganaxolone? And so these are data from a PK study that we ran in the past year, looking at 3 dose levels, 1,200 milligrams a day, 1,600 milligrams a day, and 2,000 milligrams a day, given as divided dosing or BID. And subjects were inpatient, healthy, normal volunteers. They were dosed for 3 days at each level, and what you're looking at, the dots are the trough levels, and then the little spikes are the full PK curve at each of those dose levels. And so what we see is between the 1,200 milligram a day dose, this is with our BID capsule, so it's a new formulation. At 1,200 milligrams, going from 12 to 1,600, there is, it's a logarithmic graph, so it's a little bit hard to see, but there is essentially a conservation of the dose proportionality. But when you go from 1,600 milligrams a day to 1,800 milligrams a day, you do lose that proportionality. It's about a 25% increase with a concomitant increase in trough levels of 10%. So that gives us a sense with this formulation of where we need to look in order to increase the dose in a clinical study. The last slide is the design of the study is on clinicaltrials.gov. It's an ongoing trial. Cohort 1 is fully enrolled, and it is a comparison of 50 individuals, adults with partial onset seizure, drug resistant, that are dosed for 4 weeks at the 1,200 milligram level and then 4 weeks at the 1,800 milligram level. Those individuals are almost completed with their treatment, and the safety profile, looking at it from blinded reviews, does appear to be consistent at both dose levels with the safety that we have observed in other aspects of the program. And then we are currently also enrolling in the second cohort, which is a 12-week maintenance phase of the 1,800 milligram, which we hope when completed can be part of our registration package. So with that,
Great, thank you very much.
Take questions.
and 100 people randomized, again in an adequate and well-controlled trial design. Treatment was once a day. Titration was gentle, 2-week intervals, 50 milligrams, 100 milligrams, 150, and 200. We didn't force people to titrate up to 200. We let them settle out at the dose that was tolerable and seemed effective to them. And if they were intolerant at a dose, we would allow them to drop back one dose level. The first clue comes from the subject disposition data. 113 people randomized to YKP 3089, 109 to placebo. In terms of discontinuations, for any reason, from the double-blind trial, results between placebo and drug are the same. In terms of completion rates in the double-blind portion of the trial, results are the same. In terms of discontinuations for adverse events between drug and placebo, results are the same. Of those, not every country allowed us to have an open label extension trial. Of the countries where we were allowed to have the open label extension trial, about, I think, 92 to 93% of patients chose to go on to open label. Most of them are still with us now. In terms of demographics and baseline characteristics, basically randomization worked for gender, race, age, concomitant medications, and baseline seizure frequency with no statistical difference between any of them. As you know, the primary outcome variable in these trials is typically your percent reduction, your median percent reduction in seizure rate. In the current era, placebo tends to run around 20%, plus and minus a little bit. Typical effective anticonvulsants run around 35% in their response. Excellent drugs, from my point of view, sort of best-in-class drugs, I think Keppra, Vimpat as an example, tend to run around 40%. In comparison to placebo, normal, 21.5%. SKP, 3089, 55.6% reduction in seizures, significant at the p.0001 level. So the next question is, so how robust is it? How does it affect the different subtypes of seizures? Against simple partial motor seizures, we were able, with a mere 30 patients basically in each of the groups having those types of seizures, we were able to achieve statistical significance in part because the effect size is so huge, 76.3% versus 27.8%. In complex partial seizures, which are of course the dominant seizure that you see, because that's what the trial was intended to recruit, again, the results 55.6% versus 21.1%, significant at the p.001 level. In secondarily generalized tonic-clonic seizures, again, we achieved statistical significance with a 77% reduction in seizures. The second way you look at these drugs is by the European criteria. How many people had at least a 50% reduction in seizures? By that standard, we were statistically significant at the .0001 level with a 50.2% of patients having at least a 50% reduction. We started thinking about this. Actually, we were thinking about this before the trial concluded. We were thinking, what would be something compelling? What would be something dramatic? We concluded, well, a 90% reduction in seizures would definitely have to get people's attention. So that was one of the measures. Fully a third of the patients, statistically significant at a .0001 level, had a 90% or more reduction in seizures. But we were also thinking, what's the point of this? What's the real goal? I think we've heard it referred to again and again in this meeting. What do you want to do? You want to eliminate the seizures entirely so that people can go about living normal lives. The difference between one seizure and 0 seizures is the difference between being able to drive and run your life and being impaired. So we looked at the number of patients with 100% cessation of seizure during the maintenance period when they had achieved their full dosage. 27.5% of patients achieved that. Some of these patients came in with 15 seizures a month and went to 0. Often, on the first dose at the 50 milligram dose forward. Separate, again, statistically significant at the 0.0003 level. In terms of serious adverse events, it is unsurprising that for placebo, the serious adverse events were they had seizures, right? Status epilepticus, or a major attack of seizures. For the drug, we had one patient with a UTI, and another patient who on their very first dose got some redness in their palms and feet, were taken to the hospital. It was thought to be probably a hypersensitivity reaction, given steroids, sent home, did fine. In terms of adverse events, the adverse events are adverse events familiar to everyone who's ever treated epilepsy. Somnolence, dizziness, nausea, fatigue, right? They come on from the first dose, from 50 milligrams. They typically blow over, as these things tend to do in the course of a few weeks in most patients. I might note that it's interesting that in the normal healthy volunteers treated with this drug, we treat people to 500 milligrams without significant incidence of these adverse events. It's very clear that when someone's already on 3 medicines and then you pile on a 4th, you're going to get a higher rate of adverse events than you do from the drug alone. And indeed, in our open label trial, a number of the investigators started discontinuing other drugs because we allowed them to then pump our dose of drug up to 400 milligrams. Our conclusions: YKP-3809 is a new chemical entity, has multiple mechanisms of action, selected by, as I spoke about yesterday, pathophysiological screening. It's been assessed in an international, multi-center, double-blind, randomized, placebo-controlled trial as adjunctive therapy in partial complex epilepsy. I might note that we saw the same direction of results in every country in the world. It's not driven by just one country. Once a day, well tolerated, a significant improvement in every measure of seizures that we were able to think of before the trial concluded, including the European and US criteria. Most interestingly to us, notably present was a dramatic increases, 90% or more decreases in seizures, and 100% reduction of seizures during the maintenance period. Adverse events are AED adverse events. Not a lot to say there. We think this is a promising new therapeutics. You can make your own decision whether this begins to change the game. I would say, having registered or worked in programs that led to the registration of a lot of drugs in my life, you have to do it twice before you can truly believe. But at this point, we're pleased. Thank you very much. I appreciate your time and attention.
Dr. Katz, please.
Yeah, 2 questions. The best way to distinguish yourself from another drug is to compare yourself directly to another drug. So I'm wondering whether or not in your plan you intend to do a study looking at that. And the other thing is, and so that's the first question. Second question is, do you intend to study a higher dose?
Yes. We're deeply into our phase 2B trial. I decided to allow the dose to increase up to 400 milligrams, again, as tolerated, because I want to drive that % elimination of seizures up to the highest level possible. I mean, I'd like to see that get to 30 or 35 or 40%, because that makes such a huge difference. So we're trying a higher dose. I might note, though, that about 2/3 of the patients got up to the 200 milligram dose. About 1/3 finished out at 150 or 100 and a few at 50. And they all did well as well. So we're also checking a 100 milligram dose, because that may be sufficient for a lot of patients. In terms of comparator trials, yeah, if we're able to replicate this data, it's simply a question of who do we attack, right? Who is the drug, preferably the one with the best price, the one that's the most efficacious drug in the market? Who do we take as a comparator? This is almost a requirement for Europe, from my point of view, for pricing authorities. But I think it's also a useful thing to do, because from our point of view, differentiation is not just about being clinically more useful. Differentiation is something you have to demonstrate to win in the marketplace. We think that head-on trials are a great way for us to do that. Before we do that, though, we want to make sure that we know the right dose, the right titration, the right timing, all the minutia of clinical development that you have to get right in order to maximize the effect of your drug. But Absolutely, I completely support that idea.
Thank you. I don't think we ever heard Russ recommend a head-to-head trial when he was working for the FDA. Our next speaker is Dr. Joe D'Souza from UCB to provide us an update on brivaracetam.
Good afternoon. Thank you, Steve, Jackie, and members of the organizing committee for inviting me to speak and give an update on brivaracetam. Here my disclosure. Please note that brivaracetam is an investigational drug and not approved by regulatory authorities, including the FDA. This is a summary of the key non-clinical features of brivaracetam. And brivaracetam is a compound from the UCB drug discovery program led by Henrik Klitgaard, and targeting to optimize binding to the SV2A ligand. So you can see that brivaracetam has a high affinity and selectivity for SV2. And because of this high selectivity, it has little effect on other mechanisms, including activity on high voltage activated calcium channels and AMPA receptors. It has high lipophilicity, and due to that, it has high rapid blood-brain barrier uptake, and also a quick onset in animal models. In vitro, it has activity in drug-resistant spontaneous epileptiform activity, and very profound activity against the hypersensitivity in the high potassium, low calcium fluid model. In vivo, it causes potent and complete suppression of seizure activity in animal models of partial and generalized seizures. And at high doses is effective in the MES and PTZ models, which, as you know, were used for a long time to screen anti-epileptic drugs. Brivaracetam has a high therapeutic index at doses causing seizure protection and acute motor side effects. Here's a summary of the PK profile of brivaracetam. It has rapid absorption and complete absorption throughout the GI tract. It has dose proportional pharmacokinetics from 100 to 600 milligrams, and its absorption is not affected by food. Protein binding is less than 20%, and the volume-based distribution is that of total body water, so you don't get any sequestrating into tissues or major organs. Metabolism is primarily by non-cytochrome-dependent hydrolysis, and secondarily by oxidation mediated by CPCY2C19. 90% of the dose, 9% as parent drug, is excreted in the urine within 72 hours, and the half-life in healthy male adults is 8 hours. So far, there's no impairment of efficacy of the oral contraceptive at doses of Briv up to 400 milligrams. So this slide shows the 2 fixed dose completed studies, and this has been presented before. The efficacy of the completed 2 phase 3 double-blind fixed dose studies. Study N0153 achieved statistical significance for the primary efficacy variable, which was a percentage reduction over placebo in the baseline adjusted partial onset seizures per week over the 12-week period of the study. And so was the 50% responder rates and the median percent reduction. N01252 did not meet the primary efficacy variable at the 50 milligram dose. However, statistical significance at a nominal value of less than 0.05 was achieved at the 100 milligram dose, and also for the 50% and median percent reduction. This slide summarizes the most common adverse events, seen as common events reported in the database that we have so far from all 5 double-blind placebo-controlled trials. You can see that we have over 1,200 patients in the Briv group and over 400 patients in the placebo group. The most common adverse events were headache, somnolence, dizziness, and fatigue. And the incidences were very similar to placebo. And there were no incidences in the BRIV group that was twice as more often as the placebo group. This slide here summarizes, again, from the same database, all 5 double-blind placebo-controlled trials, the dropouts due to adverse events. And on the left are the 5 trials. The first 2 are the phase 2 trials. The second are the fixed dose phase 3 trials. And the third one is a flexible dose phase 3 trial. And on the right are the doses that were used in these studies. As you can see, in total, the dropouts due to adverse events was 3.9% for the placebo group and 5.3% for the brivaracetam group. So the clinical trial program so far, we have in our completed trials, we have 32 completed phase 1 trials and 8 trials in phase 2 and 3 in adults and children with partial onset seizures and generalized seizures. This accounts to an exposure of about 2,000 patients with about 6,000 patient years of experience. And some patients have, because of the long development program, have continued on BRIV for over 8 years. So I'd like to report today that we have just completed our phase 3 pivotal trial. This took us 3 years. We had to screen over 1,000 patients to get 760 patients worldwide. It was a randomized, double-blind, placebo-controlled, paragroup, multi-center study in focal onset and uncontrolled patients with focal seizures. So this is a 3-arm study comparing BRIV, 100 milligrams and 200 milligrams, fixed doses with no titration. It was powered for both the FDA and the EU efficacy variables, which is median % reduction and percentage responder rates. So this is the largest study completed so far in partial onset seizures. We randomized 678 patients in 27 countries in the US, EU, and rest of the world with 208 active sites. So the study has been, enrollment has been completed. We've just, the last patient, last visit has just completed the study. So the results will be available in the second half of this year. Thank you very much.
Any questions for Dr. D'Souza? Chris?
Hi, Dr. D'Souza. That was really pretty good. I just question about the mechanism of brivaracetam versus levetiracetam in terms of how it binds to the SV2A. I know she mentioned it's more lipophilic, but if you can go in more detail how it really differs from levetiracetam in terms of its primary mechanism.
Well, I wasn't able to show the data comparing Lev and Viv in this study, but you know, it does have a high affinity. It binds 10 times more strongly to the SV2A. It has high, compared to levetiracetam, it gets absorbed into the brain much quicker in rats, and now there'll be an abstract at AES to show a PET study showing the absorption, the quick absorption in Lev compared to brivaracetam in monkeys.
So the Tmax is only a half hour, so it's immediately absorbed.
It's almost immediately,
okay.
yes.
Okay. Great. Okay, well, very good. Thank you very much.
Thank you, Chris. Please.
Thank you for the talk, Dr. D'Souza. One quick question. What would you expect the target recommended dose to be for brivaracetam?
Well, we would recommend the dose to be 50 to 200 milligrams, depending on the results of the last study. Thank you.
Great. Thank you very much. Our next speaker is Dr. Jackie French to speak about Vertex Pharmaceuticals VX-765.
So I think most of you know, and if you don't, I'm here to tell you that this drug is currently not in development, but we're talking about it anyway, okay, for anybody who's interested. Because there's been a lot of discussion about inflammation and anti-inflammatory therapies over the course of the last couple of days, and this is the only drug that actually has been in the clinic that the mechanism presumably is anti-inflammatory. So as you have heard, animal and human data from resected tissue strongly implicates cytokines and inflammation, not only in the development of the epileptic focus, but also in the perpetuation of the epileptic focus, and we've heard several people talk about that. VX765 was developed by Vertex, which really was not a CNS company at all. They were developing it for psoriasis, and then, several members of the epilepsy community came up to them and said, would you consider doing some trials in epilepsy and seeing if it would work? And the drug is an interleukin-converting enzyme inhibitor, so it works by modulation of pro-inflammatory cytokines. Now, the person that was most interested in this drug from the get-go is Anna Maria Vanzani, who works at the Mario Negri Institute in Italy, and she had performed this particular animal experiment prior to us going to Vertex, and it actually uses the same model as we heard about yesterday that the NIH has now adopted as one of their interesting models, where you give hippocampal kainate, the animal goes into status, then there is a period where there are no seizures, and then eventually the animals develop stable epileptic discharges that look like mini subclinical seizures, and they're easily countable. So this is, as you heard, a very treatment-resistant model, and the animals don't respond to most typical anti-epileptic drugs, and this was a study. You can see the days at the bottom. It wasn't a long study where you count baseline seizure rate, and then VX765, the compound I'm about to talk about, was administered in these chronically seizing animals, and you saw a slightly delayed, but not too much, but by about a day and a half, response with a significant reduction in seizures that continued while VX765 was administered, and then at the end of the study, in the last few days, days 8, 9, and 10, VX765 was withdrawn and the seizures recurred. So it wasn't, at least in their hands, it wasn't disease modifying, but it was a very short experiment, and perhaps if it had been given for more days, it would have been disease modifying. But that clearly was a very exciting thing for us, looking at this data and thinking, could this be a drug that would be useful in people with chronic epilepsy? And so the first study, again, a company that was modestly interested in what their drug would do in the CNS, so we had a very small proof of concept study to see whether we were on the right track or not. This consisted of a screening period, then a 6-week baseline, and the one thing that we required for this study was patients with a very high seizure frequency, because at that time the company only had enough tox to cover 6 weeks of treatment, and therefore you had to have a lot of seizures in the baseline. So it was 1 seizure per week during at least 3 of the 6 weeks, and at least 6 total observable seizures, so 1 seizure a week overall during the baseline period. The other interesting thing about this study is that it allowed up to 4, not up to 3, but up to 4 background anti-epileptic drugs, because this wasn't the typical type of anti-epileptic drug that would tend to cause sleepiness, dizziness, and other things that would have a pharmacodynamic addition to the side effects of people's drugs. So that actually made recruitment quite easy. But as you can see, there were 48 people in the active arm and only 12 on placebo, so it was not powered to be a definitive study. Another thing that we wanted to know is whether there was any kind of disease-modifying effect, so we kept the drug going we kept the patients counting seizures for 6 weeks after we stopped the drug, just to see whether the seizures would go back to baseline or not. So they continued their seizure diaries. So let me talk about the safety first. It was very well tolerated. The adverse event rate was similar for the drug versus placebo. 1 subject on drug discontinued due to rash. And the serious adverse events were probably, they happened to be all on VX765, but I don't think we can blame VX765 for a hernia surgery. There was somebody who had some pain, and that seemed unrelated to drug, and 1 person had acute worsening of complex partial seizures. And that was, in fact, after the drug was withdrawn. So here are the results, so interpret them as you will. The first column shows you the typical results as we've been seeing from all the studies that have been presented, where the treatment period, there was an 8.6% reduction when placebo was subtracted. The 50% responder rate was 10% when placebo was subtracted, and the percent seizure-free was 12.5%. Oh, I'm going to talk about that in a second. Okay, so that was the treatment period with, again, placebo, and there was a very small placebo group subtracted. So that didn't look very encouraging. But we were kind of looking over, just eyeballing the data, and it seemed like the first few weeks that they were on the drug, they weren't doing that well, but as the time progressed, the people who were on drug and not the people who were on placebo seemed to do better and better as time went by. And we remembered that in the rats who had not been treated for very long, there also seemed to be a delay between when they first got the drug and when they got the results. So we thought, well, it's only 6 weeks, but let's just look at the last 2 weeks and let's look at the last 4 weeks. And then let's look at after the drug was discontinued to see if there's sort of a delayed response. Now, if you think about it, you think that if there's a placebo effect and it's psychological, after I tell you that the drug's been discontinued, you probably are not going to, have a placebo effect, I presume. But in fact, you could see that, again, the numbers look much better when you get to the last 2 weeks of treatment, and actually in a hybrid period that we put together. This is like post hoc of the last 2 weeks of treatment and the first 2 weeks of post-treatment, that actually the results look better. And you can see that, remember, these were people who were having at least a seizure a week. Most of them were having many more seizures than that. A lot of them actually had no seizures for the 4-week period of the last 2 weeks of treatment and the first 2 weeks of follow-up. So that was a little more encouraging, but it certainly was not proof of any sort, as you can see. But, we kind of had this feeling that we might be seeing a shift to the right of the effect of the drug, so a delayed effect that persisted into the post-treatment period, and then by the end of the post-treatment period, they actually went back to their typical 1 seizure a week or more. And that led us to design a second study. Now, I will say that I begged and pleaded with Vertex to do a small, definitive proof of concept study that would prove once and for all whether this drug would work or not. But for reasons that only people in this room who develop drugs might know, they decided that instead they were going to do the study to end all studies, which meant that there were 5, count them, 5 different arms to this study. So you can see there was placebo, 300 3 times a day, 600 3 times a day, 900 3 times a day, and 1,200 3 times a day. And each of these groups was going to have 100 patients in it, which would have been a very, very large study by anybody's standards. And unfortunately, after about 50 people or 60 people were enrolled in the study, the company decided, this is way too expensive a study. It's much too big a risk. We're not going to be doing this. Goodbye. Sorry, Vertex, if you're watching. But we appreciate everything that you did for us up until this point. We really do. As it turns out, as you can see, there were about 10 to 12 people per group, which, particularly if we're not talking about the lower doses being effective, is not sufficient patients per group to, again, have any definitive answer. So here's, this was presented at the AES last year. You can interpret it as you will. You can see the change in seizure frequency from baseline for the 300, 600, and particularly the 1,200 milligram group looked pretty good. And for some reason, the 900 milligram group looked bad. But I mean, with 10 people per group, and there were a couple of major outliers in that group in terms of very, very high seizure frequencies. it's not going to tell you very much. So what am I telling you? We still don't have the answer of whether an anti-inflammatory drug is effective for people with epilepsy. There's more data that we're going to be mining in this study. We wish we could have recruited some more people. And we also have many of these people remained on drug in a long-term extension for up to a year, and we're going to be looking at that data as well in the future. So the second study was terminated before a definitive answer could be obtained, and the question of whether an anti-inflammatory drug could be effective in treatment-resistant epilepsy remains open. Thank you.
Thank you. Do we have any questions? Steve, please.
So, Jackie, in a post analysis, I'm curious whether if you looked at the seizure frequency, say, in that 900 group versus the 1200 group, you know, to see whether maybe they had a lower seizure frequency, therefore less inflammation, or I guess what I'm getting at is the seizures presumably are releasing the cytokines through which, you know, 765 might be acting.
I do know that several people in the 900 milligram group actually had very high seizure frequencies. So that's not the answer. But, you know, we all know that neuroinflammation, you know, some people think MTLE, but in fact, if you look at the resected tissue of pretty much any kind of lesional epilepsy, you're going to see inflammation and probably non-lesional epilepsy as well. Whether some people, as with every therapy, are better candidates than others, we just don't know the answer. So we wish we had had more patients that we would be able to review, but this data will be reviewed and maybe we'll find a few answers. But it's all going to be very mined data at this point.
Nick, please.
Hi, Nick Poulos, University of Washington. Jackie, do you think there would be any advantage with this drug to taking a step back to the preclinical phase and maybe subjecting it to an ASP-like battery of differing animal models, maybe to find, you know, do some sort of dose ranging kinds of experiments and get an idea of, you know, where the efficacy in animal models might top out?
I mean, Steve should stand up again, but I mean, the only new animal model that would be interesting to put it in would be the murine encephalitis, but the acute animal models, MES, PTZ, 6 Hertz, there's absolutely no reason why it would work in those models.
Well, there are other animal models of, you know, spontaneous chronic seizures that generate, you know, clinical seizures that, you know, might be interesting to see.
You know, Anna Maria has used this molecule in some really interesting seizure models. So the one that just blows me away is that she used it in the Geyer's rat, and it reduced seizures in the Geyer's rat. No lesion, no, you know, KN8, nothing, no blood-brain barrier disruption. So that, to me, I think is the most interesting thing. So it is effective, and I'm not sure you're going to find a dose from looking at animal models that's going to be the human efficacious dose. But I think that this is a really interesting molecule, and hopefully it has not seen its last day. And I just want to say one other thing, and that is that the, you know, Vertex, thank you Vertex, have given us all of the data from these studies. They were done with the Epilepsy Study Consortium, and they have told us that we can have all of the data. And if anybody wants to help us look at that data, I would welcome any help that I can get. Thank you.
Thank you. Next speaker comes to us from Italy, Dr. Dorothée Giunetti, who will speak to us about pitolisant.
Yes, ladies and gentlemen, Chairman, thank you very much for inviting me to present to you the data of the antihistamine 3 receptor antagonist, pitolisant, and I will show you the data. Everything was financed by Bioprojet, a company in Paris, France. So the H3 receptor, it was in the 80s originally described as a presynaptic autoreceptor that inhibits the release of histamine in the brain. It also inhibits the release of other neurotransmitters. Here it is shown. It is found in human brain regions with high expression in prefrontal cortex, hippocampus, and hypothalamus. And it is very well found in the limbic system. So what about then the antagonists? The H1 and H2 receptor antagonists have been very successful therapeutic agents, and they were used in allergy and in gastric ulcers. So the H3 receptor antagonists are not yet on the market, but they are promised to be interesting for enhancing a variety of functions such as alertness, learning, memory, and pitolisant has been studied also. There are publications, you can see phase 1 and phase 2 studies, and it has shown to improve refractory daytime sleepiness in patients with comorbid neurological diseases. But why would epilepsy be interesting for as a target? So there are case reports of seizures after use of H1 and H2 receptor antagonists, both in elderly and in children. And a study has been done in children with febrile seizures, and they found a low histamine concentration. And of course, mechanistically, it would make sense if it modulates activity of neurotransmitters like dopamine, glutamate, and GABA, that it could and should probably work in epilepsy. So we're talking about the pitolisant, a non-imidazole-based inverse agonist antagonist. And it was studied in rat models, was effective in the GARS model, and specifically also effective in the kinase-induced epilepsy model and in the maximum electroshock. So a pretty wide range. Healthy volunteer data were gathered, of course, and up to 120 milligram, there was a Tmax between 1 and 3, and a clear dose-response relationship. So based on that, it was decided to perform a proof-of-concept study in epilepsy patients with 3 dosages, 20, 40, and 60 milligrams. So this has been published in Epilepsy and Behavior last year. And this is a collaborative study with my colleagues in France, from Paris, from Rouen, and from Marseille and Strasbourg. And that is Parrain, Janton, Masnou, and Edouard Hirsch. So we're talking about a phase 2A study with as main outcome, what is the goal? Suppression of photocally-induced EEG, epileptiform discharges in epilepsy patients. So here is an example of a patient. First, there is a placebo day 1 baseline, and photic stimulation, you see the lead down under, can be performed regularly at different time points, standardized time points. So first in a baseline day. Then these data are collected in a way that the patients don't get seizures. You determine threshold sensitivity in 3 different eye conditions, and you end up with then ranges which can be transformed into a standardized photoparoxysmal or photoconvulsive response. So in this way, you get a lot of EEG information. After the second day, in which you give a single oral dose of the drug, you have a placebo day 3 just to look whether the effect of the drug might outlast several hours, as was the case, for example, in levetiracetam. So these are the baseline characteristics of the trial participants. 14 patients were studied. There were the majority females, all but 1 had generalized tonic-clonic seizures. 5 were without co-medication. Spontaneous discharges were seen in 6 different subjects. Frontal epileptiform discharges are more over the occipital area, as well as 2 generalized. And here are the PPR ranges per eye condition, per patient. So here are the results. You can read this in detail in the publication. So the 3 dosages were tested. There was a clear dose-response relationship. Abolition of the PPRs was found more often at a higher dose, and also the reduction changes of, as is our standard, of at least 3, was also increased with increasing dosages. And which is a side information that also myoclonic jerks in patients who showed that clearly during photic stimulation were abolished. This is an example of EEG of one of the patients with the effect, same hour, day 1 versus day 2, so post-dose, and you see a clear difference. Time of onset was 2 hours post-dose, duration of effect 4 hours. And so graphically, these are the overall result. 9 of the 14 patients, so 64%, had either complete abolishment or significant suppression of generalized epileptiform discharges. And there was a dose dependency with strongest effect at highest dose. So to put a little bit things into perspective, I want to give you some information about this photoparoxysmal EEG response. You can find it at all ages, starting from age 2 till 60 years of age as well, more often seen in females. There is a clear relationship with seizures in daily life, so with epilepsy, and a variety of seizure types. It can be found in 40% in the easy-to-treat epilepsies, the idiopathic generalized epilepsies, and you can see it's hyperventilation as well as photic stimulation that can be used as a lowering of the threshold of these patients. Then you get an example, so both in absence seizures as in juvenile myoclonic epilepsy. This can be seen here are examples. Then also it can be found at basically the same percentage in patients with Dravet syndrome, and this is in the literature. And just recently we finished in Holland a study, and we found 43% in the Netherlands, apart from all other provocative factors that are well known in this type of patient. So this is an example of a patient with Dravet syndrome who showed a response. You find it in occipital epilepsies. This is a collection of patients otherwise completely normal with occipital seizures having visual aura, or basically these are seizures, symptoms, and also an example of the EEG. So that is the benign form versus the catastrophic form is the Lafora disease, where you can find it as well. And it's not so surprising that you can find it in the temporal lobe. So to finish my talk, you can even find it in patients with Alzheimer's disease. We found it just in Italy in 2 patients. So you can find it in all over. So I do hope this this pitolisant find its way to, like levetiracetam, and for the use of treatment in epilepsy patients. Thank you.
Thank you very much. Question, please.
Yes, so H3 antagonists are also known to have profound effects in terms of sleep architecture and arousal. So help me understand whether any of the subjects in your study reported changes in sleep quality, attention.
Actually, this wasn't studied. It is a single dose study. And of course, patients can show some side effects, and the side effect profile was not different from any other drug. But you're absolutely right. That needs to be done.
All right, thank you.
Good. Thank you very much. And now Dr. Ton Bund from Izumi Biosciences will talk to us about Alaquidar, if I'm pronouncing that right.
Good morning. Thank you for the invitation to present. It's an honor with such an illustrious company here. I'd like to start with a riddle to shake it up a little bit. That is, if a patient with epilepsy, perhaps a child with epilepsy, starts responding to their drug.
Just get a little closer to the microphone, please.
I'm sorry, are we not?
No, you are, but just get a little closer.
Get a little closer.
Shouldn't touch it.
Okay, where's the, is there a control as well for the slides? Oh, there it is. Okay. My apologies for the false start. Nobody wants to miss the riddle. That's a good sign. We'll do a little quiz afterwards. So how come that if a patient with epilepsy starts off responding very well to their treatment, that over time they develop resistance to the same drug? What's the issue here? What's the riddle? Any answers? Any takers? Okay, let's move forward. We've heard a lot of beautiful stories about marketed drugs, marketed diagnostic tools, interventional tools, different tools in development, and what we'd like to discuss with you today is something different, what we call an oral pharmacoenhancer. It's not a drug, it's given concomitantly with other drugs to make them better, to actually enhance the penetration into the epileptic lesions and to restore therapeutic drug levels so that now the patient can actually respond again. So we're developing an oral pharmacoenhancer, and we're targeting the 30 or 40% or so of patients with epilepsy that indeed stop responding to their drug. So what are some of the reasons? Well, first of all, what's been mentioned is the blood-brain barrier as a major impediment to drug distribution into the brains. And in fact, it's a barrier for 98% of all of the FDA-approved small molecule drugs. Happily, the anti-epileptic drugs are all selected on the basis for their ability to cross the blood-brain barrier. But then over time, the same gatekeepers that are part and parcel of the blood-brain barrier also are upregulated in the epileptic foci, and so they in fact cause drug resistance. What I'll share with you are 3 rat models that have been developed independently from Izumi that show that a combination of known anti-epileptic drugs with an ABC inhibitor can in fact restore therapeutic drug levels and responses in these rats. About Alaquidar, we know what the mechanism is. It's a potent, importantly equipotent, selective inhibitor of 2 gatekeepers or transporters, ABCB1, known as PGP or P-glycoprotein, and ABCG2, also known as BCRP or breast cancer resistance protein. And it's important to note that these are present in the blood-brain barrier. They can be upregulated, but they also function synergistically, so they're formidable gatekeepers. We'll share with you some of the pharmacokinetics that in fact you can give this together with anti-epileptic drugs. You can retain stable plasma concentrations of the drugs. You can maintain steady levels in the brain, but you really target the distribution to the epileptic lesions, which holds a lot of promise for a good safety profile. We'll share with you some of the pharmacodynamics, that in fact a number of ABC substrate drugs can now be targeted for increased therapeutic concentrations in these lesions. We'll go over the efficacy models. And in fact, we're starting with the Department of Defense on a funded project to enhance and create the next generation antidotes for poisoning with nerve agents. You may recall that in Syria last year, they used sarin to just wipe out entire villages. We're now working with the Department of Defense to create better antidotes to protect those victims. Previously, Alaquidar has been taken into clinical development in 4 phase 1 clinical studies. It showed good safety, but it had horrible pharmacokinetics, and so that's the angle for us to really improve upon. What's interesting is that there's a strong regulatory precedent in FDA-approved pharmacoenhancers that you may know from heart therapy that are used in patients with HIV/AIDS. In fact, those pharmacoenhancers have a different mode of action in that they interfere and they slow down the metabolism, particularly of protease inhibitor, and thereby they increase the area under the curve, the Cmax, and importantly, the Cmin. So now you get a nice buffer of the concentration at trough over the minimum effective concentration that's required for antiviral therapy. These products have been the basis of accelerated approvals with the FDA. They've become significant clinical and commercial successes, so I think it sustains the test of differentiation. And so we are developing, again, a pharmacoenhancer that has no inherent drug-like activity. So we like to partner with companies that have these drugs and that would like to develop it for this targeted patient population that have drug-resistant epilepsy. So maybe the most important part is actually the little visual on the bottom, where you see an anti-epileptic drug in blue that is present in the dark red plasma or blood compartment, and it travels across the blood-brain barrier. It passively diffuses into the brain. It's then recognized as a substrate by, in fact, two transporters. Here is only one depicted, PGP or P-glycoprotein. That's an energy-driven vacuum pump that readily vacuums out the drug away from the brain. It says, it doesn't belong here, I'm going to throw it out. And so, in effect, this pump is so effective that there is no therapeutic concentration, and if at all, it just whips in and out an insufficient mean residence time. So what we propose is that we can give the same anti-epileptic drug in combination with our pharmacoenhancer elaquidar. Now they travel across the blood-brain barrier together, but elaquidar has a much higher affinity for these transporters, and so it reversibly blocks the vacuum pump, and now we obtain the therapeutic concentration and the mean residence time to restore the efficacy in that patient with epilepsy. So here you see some data developed largely in the Netherlands, where you see that in patients with epilepsy, on the top left, you see the upregulation of, in fact, 3 transporters. What's important is that there's a very high baseline of BCRP or ABCG2 already in the human brain, and that there's indeed further upregulation. There's a dramatic upregulation in PGP from a very low baseline, so people like to talk about it. It's easy to measure. And then there's also an upregulated in MRP. On the bottom left, that you see in patients with brain cancer, that they much more readily upregulate BCRP, already from a very high baseline. And so those patients with brain tumors and epilepsy are highly drug resistant. What's very important on the right, most of the information about the functionality of these transporters shows, in this case, from genetic knockout models that don't have any of these vacuum pumps, that these 2 pumps work together synergistically. 1 + 1 = 4. Or, in fact, if you look at the case of imatinib or Gleevec, 1 + 1 = almost 13. So a very dramatic synergistic effect. On the next slide here, you see in 3 different rat models with different anti-epileptic drugs before and after that indeed we can restore efficacy. And now we get to, again, treat with an efficacious therapeutic level the same patient that previously was drug resistant. I should say these are rat studies, not patients. In the middle there, you see the pharmacokinetics, that indeed it is targeted distributions to the epileptic lesions, and there's a non-significant effect on the rest of the brain as well as the plasma levels. That's not been shown here. So here is the pharmacokinetics of 2 oral formulations that we developed that nicely obtained a concentration at trough in our target range between 400 to 600 nanograms per ml in a highly predictive rat model. Here we summarize that the improved PK and improved pharmacodynamics that I've shown you also translates into improved efficacy. On the top right, you see a model in adult glioblastoma in the mouse, where only the addition of elaquidar significantly improves survival. There's a similar publication in Press from Duke about a pediatric brainstem glioma model. In the middle, you see the basis for our contract now that we start with the Department of Defense, where in the case of a neurotoxin, there's improved neuroprotection by adding an ABC inhibitor and reducing significantly the convulsions. On the bottom right, we've already gone over the effect in drug-resistant epilepsy models. So in summary, we're ready to restart the IND with new anti-epileptic drugs, and hopefully together we can restore the responses that patients have seen previously on their anti-epileptic drugs. Thank you very much.
Thank you. In the interest of time, I think we'll move on, and Dr. Brunt will be available for questions afterwards. Our next speaker is Rick Stewart from Biennial Pharma to talk to us about fenfluramine for Dravet syndrome.
Well, thank you very much for inviting us to present. We're here today to talk about 23 years of data in the treatment of Dravet's patients from Belgium. So, as we've all heard over the last couple of days, the Dravet syndrome is a catastrophic form of epilepsy with very few treatment options. We have been using fenfluramine, as I said, for over 23 years, and it's the only drug that has actually demonstrated long-term seizure freedom in these patients. On average, we're looking at around about 6 years of seizure freedom with an average treatment period of over 13 years. The standard kind of response that we've been seeing in our last data point was that 92% of patients were either seizure-free or had a greater than 75% reduction in seizures. We've met with the FDA and the European agencies, and they've given us specific advice with respect to the trial design, and we expect to commence 2 phase 3 clinical trials towards the end of this year. We've got orphan designation granted in Europe and the US, and we're pending in Japan. And as with many other potential treatments for Dravet syndrome, we believe we've got potential for breakthrough status and other routes forward. So very briefly, we believe this is a transformational treatment. Our focus so far has been on seizure freedom. And we conducted a recent survey with the Dravet Syndrome Foundation, which identified that 3/4 of parents are looking for either seizure freedom or a greater than 75% reduction in seizures. And so far, no drug has actually shown that kind of response. The drug can potentially have significant benefits for patients, not only in terms of seizure freedom, the absence of status epilepticus can have a significant impact not only on the patient welfare, but also in terms of the pharmacoeconomic benefit to payers. And we believe that there is a potential for the reduction in cognitive impairment and the potential for a reduction in overall Dravet syndrome mortality. I'm going to cover here the history of the treatment with using fenfluramine. It first started in 1998, and the most recent data point that we have is in 2014. The first report was actually in a publication by Boll and Seizure back in 1996, which examined the response of 11 patients taking fenfluramine over a period of around about 8 years. This was subsequently followed up by a publication in Epilepsia by Lieven Lagai and Burton Kuhleman, which again identified the overall patient response. This was a group of 12 patients, and since then there have been public reports at the European Pediatric Neurology Society meeting in 2013, and most recently at the World Child Neurology Congress in Brazil. This is a uniform and consistent response that we're seeing over a very long period of time. But most recently, Brabant decided to have a more detailed examination of the data, and we conducted a longitudinal study by looking at each individual patient before treatment with low-dose fenfluramine, during every single assessment that they had over the period of treatment up until early 2014. And what we saw here was an even stronger response from the patients, particularly with respect to seizure freedom and the reduction in overall seizures. And what was important was it actually gave a very strong contrast between the Takayama publication in early 2014 and the response that we were seeing from our patient population. One of the important factors is, given that this is fenfluramine, there were concerns with respect to its previous use as a weight loss drug, and our detailed analysis over the whole duration of treatment identified that there were no significant cardiac effects, principally because we're using a substantially reduced dose of fenfluramine. What I'd like to do here is to give you a comparison of two patients from that longitudinal study. The patient on the left is our oldest patient. She was originally treated back in 1988, has now been on drug for 26 years. And what we've done here is to provide a common currency, is to have an annualized rate of seizures at the beginning. And then, as you can see over time, but certainly on the left-hand side, this patient has gone from approximately 12 convulsive seizures a year down to a very low level. In fact, her last seizure was about 9 months ago. There are always breakout seizures related to fevers, but this is a substantial change, a potentially transformative change in her life. The patient on the right, patient 2, is our most recent patient. She was first treated in 2011, is currently 3 years old, was first treated age 14 months. Prior to treatment by fenfluramine, she had 25 convulsive seizures in a 9-month period with 11 hospitalizations, and her most recent seizure was around about 5 months ago. So you can see from this analysis that low-dose fenfluramine is having a profound effect on the lives of these patients. So, in conclusion, what we see here over this long-term observational study is that we have an average seizure-free period of over 6 years, and that 92% of the patients within the study were either seizure-free or had a greater than 75% reduction in seizures. And this is a truly potentially transformative drug for Dravet's patients. Thank you very much.
Questions later? Yeah, please. Do we have any questions? Dr. Cloyd.
Do you envision that patients who might be candidates for fenfluramine would require some special workup, particularly a cardiovascular workup, in order to take the drug?
What we actually intend to do is to have a full EEG workup. We are designing a Dravet syndrome care program. So at each stage, we're anticipating there'll be an assessment every 6 months. So at each stage, they'll have an EEG. So yes, we're very carefully monitoring the overall cardiac effects. But so far, over 23 years, we haven't seen any.
And you may have mentioned, I didn't catch it, are you envisioning, you must be envisioning adequately controlled trials in the U.S.?
Absolutely.
Could you just say something about that?
Yeah, we're actually having mirror phase 3 studies. We're expecting to have one phase 3 study in Europe with a mirror study in the U.S., 40 patients in both trials.
To take place when?
We're anticipating towards the end of this year.
Thank you.
Thank you. Michelle?
Yes, 2 questions. One, could you comment on concomitant drugs that were used in the studies? And the second question is, with all this data, can you tell us what the outcomes are of the individuals who've been on the drug for so long? You alluded to an effect on development. Are these people developmentally better than other people that have not been on the drug?
I'll start on with the first one, because that's the easy one. Yeah, most of these patients are either on clobazam, valproate, topiramate, so it's a combination. This is an adjunctive therapy. So it's a broad range, and indeed in a couple of instances we've actually seen them, because it's European, using stiripentol. In terms of the cognitive impairment, we have no data to suggest that's the case. Our expectation is that if we're able to treat the children early enough, and our expectation would be really from 1 year upwards, we may just be able to have some impact on cognitive impairment. I mean, it's not really known whether the cognitive impairment is related to the continued hypoxia from the seizures, or is it a component of the disease progression.
Okay, thank you. John, please.
Just curious, are any understanding of the mechanism of action, given that it's a low dose that you're testing? Is this releasing serotonin, or do you have any thoughts on that?
It's not fully elucidated yet, but we believe it's a postsynaptic 5-HT1A agonist. But we're still doing additional work in that area.
Please.
Just a quick follow-up question to John's. Is there any reason to believe that this might be effective in other epilepsies besides Dravet?
That is a very good question, and that's one I admitted from the presentation. What we'd like to do is to focus on Dravet syndrome initially, but we believe there's a potential that it might actually work in Lennox-Gastaut and potentially Dose syndrome and other refractory epilepsies. But one step at a time. We've got some very, very good data here, and the conduct of the clinical trials is absolutely critical. But yes, we are going to explore that.
Great. Okay, well, thank you very much. And our last speaker for this morning's session is Paige Figge from the Realm of Caring Foundation to talk about Charlotte's Web, facts and future.
Hello. Let me just say thank you for having me here today. As a parent of a child with catastrophic epilepsy, it's an honor to stand in front of a room full of people who've addressed this in their professional lives and dedicated your lives to studying epilepsy and furthering drug treatment. My presentation is going to be a little bit different, a lot different than what you've heard today at this conference. I'm here to share a little bit of my experience with a brand new treatment option, one that has become a hot topic very, very quickly. It's often misunderstood, both by the patients and the medical communities. My daughter is Charlotte Figge. She is a Dravet child, as you can see. She has a uniquely severe case of Dravet syndrome. So when she was 5, we ran out of options, and we began treating her with a cannabis-based oil. And in Colorado, where we live, it's legal. And so we got her started. We lab tested it. The strain of plant that we use is extremely high in cannabidiol, extremely low in THC. It's actually classified as a hemp supplement. And it stopped her 300 weekly tonic-clonic seizures down to 0 from the first dose. That was 2.5 years ago. So she is drug-free, 2.5 years drug-free. She is no longer relies on her wheelchair, her feeding tube, oxygen. She is non-medical, and she is 99.9% seizure-free. So this is one anecdotal story. We kind of waited about 1.5 years to come out with the media about this, and we had 40, 50 patients at the time, and this was in August, and we were seeing these miraculous results from this oil, and they renamed the oil Charlotte's Web. So the strain of plant is Charlotte's Web. The oil is what we do is we have a specific milligram per milliliter infusion. It's in an olive oil infusion, and we have a dosing that's extremely similar to a pharmaceutical model. It's lab tested several times. We third-party lab test it to ensure the accuracy, and the Realm of Caring Foundation is our nonprofit that was built around this oil, this Charlotte's Web strain. And so we support and advocate and creates support for the parents that are actually migrating to Colorado as medical refugees to come for this treatment. As somebody who's seen a viable brand new treatment option in real life and in the faces that we work with in Colorado, it's become somewhat frustrating to watch as the body of evidence grows, but remains in the realm of anecdotal. We have several retrospective case studies some were compiled by people in this room on CBD extracts. We still recognize the need for new treatment options and to undergo the right processes and move this realm of evidence-based medicine as quickly as possible into clinical trials. We have this media attention, this hype surrounding this, and so we are faced with a bit of a dilemma, as you probably well know. The inquiries from families that the doctors and the scientists are getting, these are patients that are out of time, they're out of options. We realize this is a difficult situation to be in, to have a parent in front of you who desperately wants to try this treatment, but the medical community still does not have the tools that come from controlled trials. So for the realm of caring, this dynamic is equally as frustrating. The press surrounding this issue has typically focused on the success stories, the miraculous success stories, like Charlotte and some of the other patients that we have, many of the patients. This creates an unmanageable expectation. So the parents expect that 100% seizure control or 99% seizure control. They're moving their family across the country, from different countries often, to come try this. And it's, we really need to push forward with our clinical trials as we will do. So I think that patients come and they expect to find a 100% seizure control, but some actually do not respond at all. This is just a new seizure treatment. As you in this room understand very well, these families don't have time to wait for this due process of the FDA process when the treatment option is available right now and showing some validity and some level of efficacy. This is why our organization finds it imperative to continue to provide channels for access. At the same time, we push forward to quantify Charlotte's Web with the best possible clinical trials. The greatest hurdle to clinical trials in the United States is a scheduling of this substance. It's a Schedule 1 on the Controlled Substances Act. It's the plant is a Schedule 1, the cannabinoid is a Schedule 1, the CBD is non-psychoactive, and yet it's still a Schedule 1 substance. So fortunately, many other countries have invited us to come in and do our clinical trial in their country. So that's what we're doing for the immediate future. We're hopeful for the data that's going to be accumulated in the coming months from a blinded, placebo-controlled trial. It will begin in Uruguay, and it'll likely end up as a multi-center trial utilizing institutions in several other countries as well. The trial protocol is in its final stages. It's being developed by the combined efforts of Dr. Orn Davinsky, Dr. Edward Ma, the advisory help of Dr. Roger Porter, and the preclinical efforts of Steve White. So that's in a quick nutshell. I know we ran over today, but I just wanted to just come out and explain what is happening in the future with Charlotte's Web and what is happening now, and just kind of clear up some of the misconceptions. And I really want to thank you so much. The Epilepsy Foundation has been a really validating supporter for us behind this, for the parents who are using this and seeing results, real results. And so I just want to say thank you very much. I know this is kind of out of the typical norm for the presentation, but so thank you for having me.
Thank you. I'm sure there'll be some questions from the audience.
No.
No. Maybe it was atypical, but I think, speaking for the audience, it was wonderful, and we really are very grateful for you being here today. Elson, please.
Question. Thank you very much for sharing your personal story. It's so important for us to hear about it. My only question is that if you should get the results from Uruguay, did you say, the country, is it something that would be acceptable to FDA, or do we have to jump the hoops again? Certainly, it will serve as a sort of motivation to the public and to FDA, to government agencies to do such studies here in the U.S. too. But what would be the status of that study for the United States?
I think the protocol is just slightly different down there. We're going to use the same protocol that is being used here. It's just that we're using the whole plant instead of isolating certain compounds in the plant. So there's hundreds and hundreds of things in the plant, and that's why it's a little bit different. I don't know how that's going to be viewed by the FDA, to be honest.
Great. Dr. Katz?
Yeah, well, just to follow up again, I haven't been involved in any of these discussions. I don't know if you have been discussing this with the FDA. There's no rule that says the agency couldn't rely on a study or even an entire data package, an entire NDA that's performed outside the United States. So that's possible, and it's happened. Of course, there are issues of data integrity, and every time a study is done in a place that the agency doesn't really have a lot of experience with, you know, because they look very closely at the data and this sort of thing. But there's no a priori reason that couldn't be acceptable. I would say, though, based on your last comment, that the entity being studied is not exactly the same as the entity that you would presumably want to have marketed in the United States. That is likely to raise questions because there are many other compounds, obviously, in the plant than in just pure cannabidiol. So I would predict that that would be an issue. But the fact that it's done elsewhere doesn't have to be a big issue.
Oh, okay, thank you.
Well, thanks again. I want to thank all of our speakers. And one final question, please.
Been any negative side effects? Have any kids regressed on?
What we're seeing is, and you know, I'm not a doctor, I'm a mother. What we're seeing and our doctors are watching is a possible interaction with other seizure drugs that the children are on. So the CBD alone, we're not seeing side effect from that, which is equally as exciting to have a new seizure treatment that has no negative side effects and actually helps with sleep, behavior, and appetite, the three very common detrimental side effects of typical seizure drugs. But no, we're not seeing that. We do see some possible interactions, levels raised in the blood of some other drugs that are metabolized in the liver on the same enzyme.
Great. Well, I'm sorry we ran over, but it was well worth the wait. And thanks again, Paige. And
thank you.
Up.
Please, just as yesterday, we have exhibitors and poster presenters who worked really hard and would love for you guys to stop by and see what they did. So lunch with posters and exhibits.
Very smart.
Thank you.
Boo. I like that.
Or maybe I can have a leash to have you.
Yeah, you can reel them in.
1, 2, podium.
Oh, that one's kind of roll, but it doesn't roll.
I was pulling it.
RF1, check. RF2, check, check. Number 3. is RF6. Hey, and of course it sounds different. Whoa, check. 1, 2, 3, 4, 5, 6. Check, check. RF. 1, 2, 3, 4. Check. Five, 5. Check. RF. 5. 2, 2. Check, check. That's 5. Number 4, 4. Check. 4. 1, 2, 3, 4, 5. Check. Check.
designed to detect such emergencies and contact.
afternoon. If everybody can get settled, we'll begin our afternoon session. I'm going to borrow one thing. Review the title we've given to our August session coming up here on who is investing in epilepsy and CNS therapy research and innovation today, funding beyond angels, grants, and awards. We are fortunate to have as our moderator for our panel Casey Lynch, who is the Managing Director of Neuro Insights. She is also the author of their excellent annual Neuro Industry Report and of their monthly Neurotech Insights publication. She's also the CEO of Cordesign, wearing multiple hats. She has a MS in neuroscience, and I learned today that she actually trained in Dan Lowenstein's lab. So we're delighted to have Casey with us. On the panel, we're going to have Greg Simon, a longtime friend, innovator, and great person. He's the CEO of Polywog, with whom the Epilepsy Foundation has formed an important new partnership that you'll hear more about. Polywog is an innovative financial firm transforming the healthcare industry and helping to finance some of our smaller companies. He has a rich resume, but I will highlight his role as the founding president of Faster Cures, which is a center of the Milken Institute. We also have with us today Dr. Manuel Lopez, who is the vice president at Bay City Capital, specializes in CNS. He is also a scientific liaison for the Pritzker Neuropsychiatric Disorders Research Consortium. Also with us is Dr. Sigrid van Bladel, who is a venture partner at Aberdare Ventures. We have Dr. Adam Bristol, who is the vice president at Aquila Partners, an investor, an investment firm. He also serves as a startup mentor for the NIH Commercial Assistance Program and as a business advisor to the Michael J. Fox Therapeutic Development Initiative. And finally, we have Sam Wu, who is an MD, PhD, and managing director of MedImmune Ventures, had experience at SF Life Sciences before, with broad experience in biopharmaceuticals and platform companies, as well as past work at McKinsey & Company. We have two of our panelists. Sigrid also, I think, spent time earlier in her career at McKinsey. So with that, I will turn it over to Casey.
Thank you, Warren and Roger, for inviting me to moderate again. This is always a great conference. Just to set the stage, I thought I'd share a little bit of Neuro Insights data on venture investment from last year. We don't have the semi-annual numbers put together yet, but investment in neuroscience, including pharmaceuticals, devices, and cell therapy companies and diagnostics, was up last year 11.7% to $1.44 billion. That's really great compared to overall life science investing, which was down 2%. The gains were mostly due to the pharmaceutical sector, which was up 30%, alone over 103 deals. This is a lot due, obviously, to the IPO window being very open and active, as well as the M&A. In devices, the investment was down a little bit last year to $320 million, over 35 deals. That's a lot due to a really great year that happened in 2012 for neuro devices. There was so much investment that it was bound to come down a little bit. And finally, in neurodiagnostics, it's always usually sort of low and steady, $62 million invested over 14 deals. So with that, I'd like to ask each of the panelists to introduce themselves a little bit more, if there's anything they'd like to add to Warren's introduction about your fund, and also what's your interest in neuroscience and epilepsy in particular? Why are you here today?
Sure. I'll start. I'm Adam Bristol, and I'm with Aquila Capital. We're a life science investment fund here in San Francisco. My personal background is actually in neuroscience, and I did my PhD and postdoc in the endogenous cannabinoid system. I wasn't specifically studying epilepsy, but certainly the CB1 receptors in neuropathology and neurophysiology, specifically with the cerebellum. And my investment fund, we really cut across therapeutic areas. We do some things in CNS. We also do a lot in oncology and autoimmune and inflammation. We're very much sort of fundamental due diligence, long-term buy and hold investors, primarily in the public markets. And we can describe how the public markets are important even for entrepreneurs and private companies, because the robust public markets can set the stage for the capital markets and for private companies as well.
Hi, my name is Manuel Lopez Figueroa. I also have a PhD in neuroscience years ago. And I wear two hats. I wear a hat of venture philanthropy. I work for a foundation that is called the Pritzker Neuropsychiatric Disorders Research Foundation, which is funding a consortium of universities, including some in the Bay Area, like Stanford, but also UC Irvine, Cornell, Michigan, and Hudson Alpha. The goal is to identify novel therapies and diagnostics for psychiatric disorders, bipolar, major depression, and schizophrenia. Therefore, some of the comorbidities with epilepsy. And we have been very lucky that over the last 13 years of funding, we have identified novel targets for depression. And through the funding of the Pritzker family, we will be hopefully moving some of these towards the clinic this year. So it has been very enlightening in that regard. These are multidisciplinary groups from basic science all the way to the clinic. And I'm also wearing the hat of venture capital. I work for Bay City Capital, a company that has been around since '97. We have about 1.6 billion under management. We have invested in over 100 companies in the life sciences space, going from biopharmaceuticals, medical devices, diagnostics. And we cover the whole spectrum. We go from very early stage all the way to traditional VC investments, but also pipes. We invest in public markets, and we also do different kind of transactions. The CNS space is one that obviously is close to the heart, and about, I would say, at least 10 companies that we have invested. Some of them, we have people represented. Next Wave, some of the members of Next Wave are here. But also we have done investments in companies like Civitas recently, whereby the money that the Michael J. Fox had put into it put a stamp of approval, or a stamp of a knowledge of the quality of the technologies that we are investing in.
Great. My name is Sigrid van Bladel, and I'm a venture partner at Aberdare Ventures. And I'm maybe more of a generalist in that my PhD is only in molecular biology. So I'm not a brainiac to the degree you guys are.
I'm sure you are.
I joined Aberdare as a venture partner in 2011. Before that, I thought I had retired, but apparently not. But I had worked at NEA for 18 years as a partner. NEA is a very large firm. Aberdare is more of a small, specialized firm focusing on healthcare investing. And NEA is more generalist, very large. And at NEA, in those 18 years, made a number of investments in a variety of medical fields, but maybe most notably NeuroPace, which was a company I was on the board on for 11 years. And also XL Pharmaceuticals, which was the maker of the Diastat, which was the rectal benzodiazepine product for rescue medicine for seizures. And they also had the retigabine asset, which then that company got bought up by Valiant. We also invested in Alexa, which now has a product with the Staccato technology. And NEA also invested in Supernus, which has recently had the sustained release topiramate approved. My other investments were in cardiovascular field. I was on 15 plus boards over that time period, and some big returns and some pretty spectacular misses too, but that would take up too much time. So I'm from Belgium originally, came to the States to do an MBA. My PhD was at University of Ghent, MBA at Stanford, and then picked up by NEA.
Hi, I'm Greg Simon. Can you all see us over there, hear us over there? Don't know what you did wrong to go over there, but we'll let you back here.
Maybe it's us.
So, yeah, maybe it's us. So I'm always the answer to the Miller analogy test of which one of these people don't fit. since I have an American history degree from the University of Arkansas. But I don't mean to drop names. My interest in epilepsy was when I started working with Mike Milken and met Warren Lammert and many of the other brave and inspiring families that Warren introduced me to over the years. And when I started Faster Cures, I'm sitting alone by myself, having left my own business to do something Mike Milken thought was a great idea. And I said, how am I going to do this? And I realized I didn't have to do it. There were all kinds of people who were already doing it, changing the way we do research. I just had to find out who they were. And they were the people who were taking foundations into a new direction. And it was all about culture. When we had our first meeting with a number of foundations from a variety of diseases, no matter what the disease was, the biggest obstacle was not the science. It was the culture of the way we do science. So now at Polywog, I'm focused on the culture of the way we do finance. Because for the last 50 years, finance, private company finance in particular, venture finance, has been the playground of the very wealthy and people who had access to people who had access to people. So what I call the Barbra Streisand principle. And the problem with that is that you're not going to solve any of our problems in epilepsy, Parkinson's, Alzheimer's, et cetera, by finding a few people or a few funds that have millions of dollars to put into it. You're going to solve it by finding thousands of people, millions of people who have thousands of dollars to put into it because they realize that the risk of losing their money in the investment is less than the risk of keeping the disease the way it is now. So fortunately, otherwise I'd be out of a job, your government passed a law that makes the democratization of venture capital possible. And you couple that with social media and online investing that makes it very easy to get information to people. And the law that allows me to tell you that I'm raising money for a private company in public, which a year ago would have been illegal. So now you have the ability to communicate what you're doing in raising money in private companies, small companies. You have the ability to reach people through social media. And you have people who are willing to take a chance on, who've never had the opportunity to invest their money in the future of a disease that affects their lives because it was too expensive and too elitist for you to get into the funds that were making the big investments. And those funds are necessary, but they should not go away. But the future is going to be millions of people making choices and spending thousands of dollars each. And that's what Polywog helps facilitate.
Fascinating. So my name's Sam Wu. Very, very interesting, by the way.
Thank you.
I didn't know that's what you guys did. I always feel guilty since I live in Silicon Valley if I don't know what's going on, even though I'm not in the tech sector. But I'm actually not on the program. I'm crashing this panel at the invitation of, I guess it must be Warren. But I'm a managing director with MedImmune Ventures. We are the corporate venture capital arm of AstraZeneca. So if anything, I represent that end of the spectrum that's going to come up with the hundreds of millions of dollars to pay for the last push, right? The pivotal studies that get drugs to approval. I think Warren mentioned my background. I've been in venture capital for 12 years, only got into this current firm about 3.5 years ago. Have a broad experience base in terms of investing in both medical devices as well as therapeutics in almost every therapeutic area. In terms of what we do for AstraZeneca, we're kind of we actually invest with a focus on financial returns. We don't necessarily invest on strategy, but we are call it a window into early stage innovation for our corporate parent. And certainly, epilepsy, neuroscience is one of the 5 major therapeutic areas for AstraZeneca.
Great, thanks. So focusing on epilepsy, it's such a heterogeneous disorder. So we have sporadic forms, genetic forms, childhood, orphan, elderly. It seems like in terms of trial design, in terms of companies bringing drugs, a lot of people are focusing on orphan disorders now, which present some benefits. How do you view this segmentation of the patient population? I'm going to start with Sigrid.
Well, Part of the problem with doing target is really in a targeted way segmenting the population. It's difficult to do when you have a disease. It's easier to do when you have, say, a cancer where you can do molecular typing and you can really rule out certain molecular markers and really start honing your therapies to that. We don't really have the luxury of that in epilepsy, from what I understand, yet, or not enough to do that. So it's very difficult to do an intelligent triage and selection based on that. However, one area where you certainly can is looking at whether you have more generalized disease or a disease in foci. If you have an epilepsy that has one or two foci, say, in their hippocampal, then a patient like that could very much benefit from a more site-specific therapy, such as an implantable device, like the RNS, to really help them bypass the side effects of a lot of these medications, because a lot of these meds don't belong in your frontal lobe, particularly if you have a focal onset. You don't want to be messing with a person's alertness and with a patient's sense of balance and things like that. So triaging based on focal or not is certainly a very meaningful way, I believe.
Will, do you want to comment on, I mean, is a trial that's going to take all comers, is that going to be a problem, do you think, in the readouts? Or?
Let me follow up on the first question, because I think that actually there was an article that just was published by AstraZeneca, by members of AstraZeneca in Nature Biotech. It talks about the 5, actually 6 R's on drug discovery. And is Mani Pengalos, which is the head of CNS, is one of the last author in that paper. I will highly encourage you guys to read it.
He's actually the head of R&D. Head of R&D.
Yeah. So one of the R's is the right target population, and the target population can be defined genetically. And I think that in the case of epilepsy, I mean, we have seen today, but over the last 2 days of this conference, how we're starting to have a better understanding of the genetics. We have novel targets that allow us to identify novel populations, and therefore you can start targeting the population that you can include in your clinical trials, but hopefully also the ones who will respond to your particular treatment. And I think that helps on, in particular in CNS, where we know that there is a high placebo effect. Hopefully try to ameliorate that placebo effect by assuring that that's the population that is going to respond, but also tailoring treatments to avoid, in some cases, the unnecessary adverse events or the adverse events that come with certain treatments. So I think that we're moving in that direction, whether it's in epilepsy or even in other indications that are, obviously oncology is leading the trend, but I think that is now being applied to CNS.
In the public markets, genetic segmentation and orphan disorders is very hot right now. We're seeing a lot of activity, for example, in Duchenne muscular dystrophy, where the FDA seems to take action, and then even it seems like the patient groups may even be having an impact, and we can't quite tell what's happening yet. But what's your perception on the public markets of how this is playing out?
Yeah, no, absolutely. So I think there's probably a couple topics embedded there. I mean, the first is that the public markets are very fallible. They are catalyst-driven. They are momentum-driven. They are sentiment-driven. And so, but some of the fundamental concepts that the public markets have certainly latched onto are orphan drug designations because of the things that come along with it. There's a perception that you can have an expedited development pathway. There's a perception that you can have premium pricing, and pricing that's going to be resistant to certain genericization or pushback on pricing, and so you can sustain that dominant market position for a long time. Certainly orphan drug designation can provide that for you. And then I would also say, too, that there's a free associating, but in terms of Duchenne muscular dystrophy, it is a great example of where patient advocacy groups have played a very important role on multiple fronts. They have played a role in patient registries. They have provided funding in certain cases. They have been advocates in front of the FDA looking to enhance and hone the clinical trial design. What are the important patient-reported outcomes? I think, getting back to some of this patient stratification we were talking earlier, just in preparation for this meeting, I was kind of just thinking on a high level, in the asthma field, to the extent that I understand it, the asthma is defined by treatment failures. You are inhaled corticosteroids failure, then you fail this, then you fail that, and so that's kind of what defines the patient. It seems, from my, being pretty far removed, but epilepsy, it seems like it's a little bit like that. That is, often these patients are recalcitrant, and so they're almost defined by what they don't respond to. That's not really getting at the biological mechanism. And so when I looked at a few pivotal trials recently, the patient stratifications, at least in terms of post hoc predefined analyses, were on age, number of AEDs, seizure frequency at the time of enrollment. Again, not speaking to mechanism at all. And so I think that, so I'm kind of conflating two things, but and patient advocacy groups can have a huge impact in driving the clinical science, as well as marshaling resources towards improving clinical drug discovery and development success. But I think the translational biology's got to catch up too. The public markets will recognize all of this. They really will. But I think that if you look at other areas of drug development, like in oncology or in inflammation, where some of the mechanistic and objective clinical endpoints are there, sometimes they tend to latch onto those. But you can see tremendous successes if you have positive clinical results in CNS. And we could talk about a number of examples more recently. But the public markets will recognize it. But I think there's some inherent challenges in this area that need to be addressed.
Sam, you want to add something?
Yeah, I mean, from the corporate, and particularly from the pharmaceutical corporation perspective, right, there's quite a bit of focus, and I can definitely speak for AstraZeneca on this, on being able to identify subsets of populations that are more likely to respond to specific therapies. And this is across the board. I mean, going back to asthma, right, AstraZeneca probably has one of the leading pipelines, if not the leading pipeline in respiratory right now, and particularly in asthma. They have multiple compounds that are targeted for very specific patient subsets, not defined by what drugs they failed, but by very specific biomarkers, right? And in some cases, very specific to the antibodies that are hitting particular targets. I honestly see that this is the path forward, right? What we have to do with epilepsy is understand a lot better, what are the mechanisms, what are the underlying causes, and what biomarkers can we use that will help us differentiate patients in a way that's meaningful, that will really have a relationship to the drugs that we're developing to treat the disease.
The one thing I would just want to add while I'm thinking about it is that reformulations and sort of incremental improvements that may improve patient compliance or convenience, those have done really well in recent years too, and I can give you a whole basket of publicly traded companies which have done extremely well because of some type of product offering. So while I would love to have the grand vision of sort of better molecularly defined or biologically defined treatments, I think there's also room in the public markets will recognize some of these more incremental improvements, for lack of a better term.
And that's a great lead in. Let's talk about reformulations a little bit. And Greg, I'm not forgetting about your
No, that's okay.
Polywalk and crowdfunding, certainly important. How do you view, we heard Betsy Katz previously at the FDA stand up and talk about head-to-head comparisons and reformulations. While incremental improvements are going to face regulatory and reimbursement hurdles to prove that they're really better than what's out there. So, Manuel, how do you view investing in those kinds of opportunities?
I have a bias because we have done very well investing in companies that are reformulations. We just, the last investment was Civitas, which is one of the oldest drugs, L-DOPA, which the company is reformulating, but also putting in a device that allows for an early onset of action and to treat what is a significant problem on the Parkinson population, which is the off effect. Another exit that we're proud of is NextWave, which is also another reformulation for ADHD. And we have done several investments that we felt that the market opportunity, again, you have to look at what are you doing, what is it the market that you're trying to achieve, and what is the unmet need in order to do something that by others may be as something that is not innovative, but we felt that it was a right investment.
And NextWave was a liquid formulation of current ADD medicine that was bought for $500 million by Pfizer? So
700.
700.
Sorry. It's a couple of zeros.
Right, so that's huge. And they did have to get to approval before they were bought. So that's
well, yeah, it was a structured deal, but yeah.
Okay. Good to know. Sigrid.
But, and drug delivery was, particularly in the 80s and 90s, that was the golden ticket, right? You just reformulate, you patent extension, and a lot of money was made, and a lot of our firms, and Supernus was an NEA company. But I think in this day and age, you have to ask yourself, who's really benefiting here? Because if it's the pharma company or the investor benefiting, but the patient is only marginally benefiting, it's not sustainable. Our healthcare system we have such inflation in our spending. That's not where we should be making money. That's not where money should be made. It should be in making significant impact on outcomes or saving significantly on these huge expenses that are being made now. So Aberdare actually took a stance in their most recent fund against what we called premium priced, marginally better products. We just would not invest in them. That's because our healthcare system cannot support it. It's radical, but I fully embrace it myself.
I'm in complete agreement, and certainly, traditionally pharma companies prefer to see the novel mechanism and a big impact drug. As an investor, and as an early stage investor, I prefer to invest in something that I think will have a big impact and therefore a big return, as opposed to investing in something that will have a smaller impact, and I'll have to take all the way to approval before I can get to my exit.
It's funny, we often think, and I'd be curious from the other panelists, we tend to think risk for our drug development companies is kind of coming in 4 buckets. It's financial risk, it's clinical or technical risk, regulatory risk, and then commercial risk. And so on the reformulation or drug delivery side, you may think, and it's probably true in most cases, that the technical risk is going to be lower than a novel first-in-class agent. But then the commercial risk is probably going to be higher, and then maybe even getting higher in the future. There's no doubt that, especially if you're taking a 505(b)(2) strategy where maybe almost like a bioequivalence is really what's getting you approved, then it's convincing the payer, convincing the prescriber that this is something demonstrably different. I think when I look at companies like a NextWave and others, I think what's most impressive about NextWave, again, I don't know this as well as you do, Manuel, but the number of almost, again, I'm on thin ice here, but it seemed to me from the outsider's perspective that they did a lot of studies that maybe not necessarily needed for FDA approval, but were important almost kind of marketing studies, that are studies that could demonstrate its value versus other products on the market. Is that a fair assessment or no?
I think that all the elements that you mentioned before were put together in the analysis of what the opportunity was and what the requirements were, or what the questions were addressed by means of the studies. So for instance, I think that you make a very important point, which is the payer's perspective.
Sure.
I mean, you have to make a very good case that any drug that you put into development is going to be differentiating enough with a standard of care. And in many cases, standard of care with genericization have a great impact on the amount of money that you need to develop those technologies. And therefore, if you were a pharma company, you can do a lot of clinical trials to some extent to answer more questions. With the venture capital commitments, you may have to limit yourself to the questions that are critical. So I think we answer. Obviously, the product was approved, so I think we answered all those payers.
So, Greg, at Polywog, you have 2 epilepsy companies, as I understand it, on deck. One which would be available in 2 weeks?
Probably by the end of the month.
By the end of the month, and then the other to follow. What kind of diligence, what do companies need to, what requirements do they need to meet to be listed? What kind of diligence do you do in order to include a company on Polywog?
Right. So Polywog is not a venture fund. So let's start there. So if you're a venture fund, you're using other people's money. And so, companies are from Venus and money is from Mars. Think of it that way. Companies are aspirational, they have vision, and then they meet people who are very hard-nosed, who say, no, this won't work, this won't. So when a venture fund has to do that because they're using other people's money and they only have so much of it. So if they saw 5 epilepsy companies in a given area and they thought that they had to pick one, they'll pick one. Our goal is that if we see 5 epilepsy companies that all have promise, we will put them all on our platform and let the investors choose the ones they think are the best for them. So how do we choose? So we do 2 things. We look first, is this company dealing with the problem that there's a crowd that's interested in dealing with it? Yes, in the case of epilepsy. Now there are lots of other diseases where people are still dealing, especially in the neurosciences, where people are still testing peptides in rats, it's hard to get a crowd to want to follow that. That's what seed funding is for. It's what NIH funding is for. Second question we ask is a big question of, is this company investable? Are they doing something that would make a difference? Is there an addressable market? Is the money they're raising taking them to a milestone? Is the team appropriate? Do they have the right kind of board? Do they have the right kind of science? And has it been tried so many times we know it can't work? Or is this a crazy idea that might work? The problem isn't that we're funding too many companies. You know, we hear a lot of times venture funders particularly will say we're funding too many companies because their failure rate is enormous. Well, that's not how it works. If you've got a low success rate, you need to have more tries, right? If you're a baseball player and you're in a slump, you do not get better sitting on the bench. You want more at-bats. So what we're trying to do in the space where companies need $2 to $10 million, not the VC amounts where they need 20 to 50. We're trying to get people who have been funded by foundations primarily, foundations that have a rigorous scientific and business analysis process like the Epilepsy Foundation. We then ask them to help us identify the companies they have given money to that they think offer the best promise. As broker-dealers, we have to do our own diligence. We don't just take them and post them. And then we look at those questions that I just asked. And for instance, in the muscular dystrophy space, we've looked at several companies, and we might want to help fund them all. A friend of mine who has one said, but I don't want to be one of 5 companies on your site. I want to be the only muscular dystrophy company on your site. And I said, that's not how it works. That's not how it works. We're trying to find as many shots on goal that are worth taking, not trying to pick winners, because nobody knows the winners. Nobody knows the winners. So at the end of the day, we're trying to give more people an opportunity to see more companies that need to get to proof of concept to get through what has been so badly named and appropriately named the Valley of Death. Nobody wants to invest in the Valley of Death, so we're trying to build the bridge of life to go over the Valley of Death by giving people a chance. Because the companies that fail for lack of money, we have no idea if their idea would work. NIH gives $30 billion a year to 300,000 people. If only 1% have a great idea for a company, that's 3,000 companies a year. Last year, venture capital funded 474 biotherapeutic companies. And if you scratch them, they'll say, well, all the good companies got funded. No, all the companies they could afford to fund got funded. But they're using money from 3% of the 9 million accredited investors in America who are allowed to invest in private companies. We're focused on the other 97%.
Great. So I want to talk a little bit about devices, and then I'm going to go to audience questions. So get your questions ready. In the device area, we're finally seeing some signs of life from the device division at the FDA. NeuroPace got approval, yay. I noticed, however, that Medtronic was still giving sort of vague, we're still working with the FDA on their DBS device. So from an investor perspective, how do you view device investment currently? Unfortunately, for some reason, the devices aren't as hot on they're not doing the IPOs like the pharma area, and it's unfortunate, but that doesn't mean it's not a great time to invest for the IPO that could happen 5 years from now. So great.
An advantage of having started in the business in the last millennium, many years ago, is that comment keeps popping up. Yeah. Where are the medical device IPOs? Yet money is being made in medical devices. I mean, for a while even, why is healthcare even worth investing? I remember that very clearly in 2000. And I was the only person at NEA doing healthcare investing, including NeuroPace. And so these cycles of skepticism and nervousness, they happen all the time. What may be happening in devices is that these companies get acquired before they have before an IPO is really seriously contemplated. So there's a huge variety of potential acquirers. It's not just the big 3 or big 4. I mean, you have companies left and right, making some unexpected acquisitions, so, or, even device companies, like take Conceptus last year getting acquired by a pharmaceutical company. So I don't think, my perception is that investors aren't necessarily deterred by the lack of exit options in devices. I think there's maybe a trend towards being cautious just because of the time it is tending to take to get through the FDA, and the back and forth with the FDA can take years. And so it's really, as an investor, you have to think carefully about the timeline, the money that it's going to take, and add a big fudge factor in there. And then be very disciplined about how aggressively you invest, because you do not want to get too aggressive up front, run out of money, and then not have enough reserves. And then in round 5 or 6 of financing, because that's how these deals go right now, you may end up being wiped out by a new investor. So it shifts how you spend your coins, and you just kind of think very carefully, and you go in with your eyes wide open that reimbursement may take longer than you think, and the FDA might throw some ridiculous hurdles at you.
I may add that we have seen a trend in the last years where going through the EU, it actually makes more sense because of the regulatory agencies are less stringent. Payers are willing to pay in many cases, and you get traction in the market. And actually, if you have a company that has some traction in Europe, please come and talk to me, because we have an interest in particular medical device companies that follow in that trend. Nevertheless, I mean, as we know, the FDA continues to be hard in many regards, and payers also is not as trivial in the US as it could be in Europe. We are lucky to have an investment in a company like Nevro, which is a pain medical device, and going to your question about public markets, I mean, I think that there are other companies that may come up with the windows that we have seen in the last year and a half as medical device companies going public.
And this point that Sigrid brought up of the pharma companies, I know many who are definitely keeping an eye on the device area, especially the non-invasive devices, including GSK with their new fund. But MedImmune, you said you do device investments, and what's your?
So we have been in the device space for the last few years since I came on board. Actually, the rationale actually had to do more with our strategy than because the corporate parent had any interest in medical devices. Our approach was to be financial investors that would be good partners to other leading venture firms, and we were willing to do that by making good financial investments in almost any space where we had expertise. I think, to speak for AstraZeneca right now, they're very much focused on their pharmaceutical business. They're very much focused on their core therapeutic areas. So, I won't let anyone start believing that AstraZeneca is going to get into medical devices all of a sudden.
Good. So, audience.
Can I one follow up on this point though? Like, Seeger, I'm curious because I live in the therapeutics world. What's a blockbuster device do in terms of revenues? Because one thing that I always see what kind of buttresses the pharmaceutical side and all the IPOs is the lottery-like home run potential of a big drug. And I've often wondered if other areas in life sciences could necessarily match that. And so then that kind of changes your thinking about what the value proposition is and just what the,
Well, if you go, depending how far back you go in time,
Sure.
I mean, stents.
Oh, sure.
Billions and billions.
Yeah, that's a good point.
Pacemakers.
Yeah.
Frank was the CEO of Ventritex.
Sure. Yeah, that's a
But
good
then you have, and sometimes the data is buried because the company gets acquired, they're showing nice sales traction and they get gobbled up and you don't really know what the sales are and they're buried.
I've often thought of it, that the value creation in devices is slightly different than the trajectory of a drug development company. So drug development has a very long, protracted, expensive period of presumably risk reduction and value creation over clinical development. Similarly, devices have that too, but for them, sometimes it's an engineering problem. It's maybe more of an EPS story as they're behind the pavement, kind of post-approval. So the growth is a little more organic, where you can have explosive value creation in in any market, private or public, with a therapeutic, if you have compelling clinical data. And so I'm just wondering, I'm just kind of thinking it through, because I hadn't really thought about necessarily whether or not boom-bust cycles and IPO windows device would necessarily track or be covariant with the IPO windows for therapeutics companies.
They don't always,
They don't always,
I think the other question maybe to ask you, Adam, is public investors are willing to buy into a pre-approval, post-phase 2 drug.
Yeah.
They will not buy into a pre-approval. In fact, mostly they won't even buy into a pre-revenue stage.
Good point.
Medical device company. The question is why, right? And I know part of it, since I invested in this, I know part of the reason we don't, we look at it this way is because, well, we know that the acquirers don't act until you've got $20 or $30 million in revenue in the device space, which is very different from what happens in the pharma space.
Yeah, no, I think that's exactly right. I was going to say, why is that? Yeah, I think that's exactly right. I mean, clearly in terms of what are the ultimate customers looking for, I mean, clearly drug companies, device companies are developing products for patients, but very few, certainly in the therapeutics world, they're going to grow up and market their own drugs. I mean, some do, but generally their customers are the pharma companies. And, pharma is often looking for compelling assets at all stages of development. What really drives deal value in my mind is not necessarily stage of development, but is a competitive dynamic. It's something that could be really breakthrough about it. It may be very different in the device side, which then is reading through to the investor behavior as well in terms of what they're looking for.
And so therefore we don't see the IPOs because the companies aren't getting to that stage.
To the inflection point where they
have that
investor
IPO.
appetite. Exactly. Yeah.
Okay. Audience questions? Lauren, do you have a question?
Hide behind the pillar to ask.
So we can't throw anything at them.
I don't know if I can move the microphone. There we go. I just wanted to ask, make a further comment about our partnership with Polywog and ask a question about that. We, the Epilepsy Foundation, have actually made an investment in Polywog as a platform to try to help some of the smaller companies and projects that we funded. And we funded in total 60 now, as Rogers mentioned, of about the 120 new therapies in the pipeline, most of which are at very early stages. But for those that are in their early stages that have often received funding through grants, friends and family, generally less than $1 million in outside capital, we are finding it difficult for those companies often to get a bite from venture. Some of them do, of course. And we thought that this platform could help us move sort of at least into the clinic, in some cases into early proof of principle, at which point we might be able to secure funding from more traditional institutional sources. The Alpha One Foundation made a similar investment to try to develop the platform, and I know Greg is working with other disease organizations to try to bring them into the platform. But I'd just be curious for your feedback about how this could fit into the ecosystem of new company finance and venture fund investments.
So there are two different ways that it fits together. One, as I mentioned, is the raising money for individual companies on the platform with the ability to reach through the foundation to donors, to people who care, to people who visit the website, people who get the newsletter, people who talk to other people, none of which was possible literally last August, and now it is. So the ability of people who are in a group to communicate with each other, to let people know that there's an investment opportunity that's relatively modest compared to what investment used to be before the private companies were allowed to have more investors and still be private, is one way we do it. There's a second way I didn't mention, which is in addition to the private company fundraising, we are creating public venture funds. Private venture funds, you generally need $1 million to get into a private venture fund. But we're creating publicly traded, registered funds that will cost people $5,000 to invest in, that will invest in the verticals of healthcare that people care about. People don't care about big cap stocks or small cap biotech. What they care about is epilepsy, Parkinson's, Alzheimer's, etc. So our first fund, which is registered at the SEC, and we're currently putting the underwriters together, will be a $100 to $200 million fund to invest in regenerative medicine and stem cell therapy companies, both private and small public companies. And that has an independent manager. We pick the manager, we raise the money, and then they invest. And by law, they're required to only own so much of a company. So by law, they have to co-invest. So that already has led us to a lot of relationships with firms that would never have invested in some opportunities unless they could co-invest. And now we bring a lot more money to that space. The second fund will be in health information technology, and the third fund will be in neurodegenerative diseases, a wide swath of those. And there will be other funds in cardio and cancer, et cetera. But the point is, we have to be able to take these small companies and seed them to the next level, and do that in a way that we have 40 or 50 shots on goal, like in some of our funds. Now, you might ask, how is that really going to help? How is that really going to benefit epilepsy and benefit an investor? Well, 7 years ago, if I had told you I think this Facebook thing might be real, and they're going to have a huge platform, and there's no way they can build everything they have to put on that platform. They're going to have to buy some stuff. So if 7 years ago I had put a fund together like the one I just described, and we'd gone out and invested in 50 small companies that we thought Facebook would like, and one of them was Instagram, and one of them was WhatsApp, you'd be very happy today. Now, if I had told you 7 years ago that I was investing in a small company that allows you to text people with pictures and it's going to be worth $19 billion in 7 years, you would have thought I was crazy. But today, if I tell you I'm investing in 40 stem cell companies, and I only need a handful to make a difference, because pharma can't build all those companies, they have to buy those companies. And when a company makes a difference, they will get bought and they will make a big difference. The problem is how we spread it out so that we can get more things funded at the bottom level of the pyramid. The partnership with foundations gives us the benefit of your knowledge and gives you the benefit of our financial platform so that we can bring money to good ideas. That's what, at the end of the day, it's what it's all about. And not always a lot of money. It can be $1 million. It can be $500,000. Because that sometimes is all someone needs in conjunction with other money to get where they're going.
Warren, first of all, I would like to applaud you as a person, because what you have done here is very unique. I mean, you have been able to bring to the table over the years people from all the members of the ecosystem, starting from all kind of sources of funding, and that's not trivial. Venture capital, biotech companies, entrepreneurs, all the way to pharma. And to do that requires certain skills. And you can use, for example, Michael J. Fox as another foundation that is trying to follow a similar path. And clearly now the dialogue with pharmaceutical companies becomes critical, because the more advanced the programs get, they're going to be finally the ones that have the capital to bring them to that next stage. And unfortunately, we live in a environment right now where CNS have been de-emphasized. But to your point, and the point that you're trying to make, is the idea of bringing some of these technologies to the next inflection point, that when something that is clearly fulfilling the needs, whether it is a medical needs or whether otherwise, is going to be the right time for those companies to go to the next stage. So again, I would like to personally applaud you for your efforts.
I think the only answer is all shucks, Warren.
Karen, did you have a question over there?
Yeah. It's kind of a wrap-up question in the sense that time is tight, but also I'm curious, we have representation on this panel from venture capital, from corporate venture, from crowdfunding, from public equity investment. For each of you, from a therapeutic or device perspective, depending upon where you invest, what is the threshold? If you were to say to a young company, don't come to us until you can show us these things or you know this thing, is there a threshold that you would advise companies in preparing to raise money that they need to have hit, or they need to be able to describe that they understand?
Yeah, I'll start. I mean, that's a million-dollar question, and obviously every investment opportunity has its own vicissitudes and nuances, and so everyone kind of stands on its own. But in general, the things that we think about are clearly the people, and we want extraordinary people involved. That's just not just the scientific founders. That's the business executives who are enabling it and moving it forward and helping reduce the execution risk that goes along with any endeavor. But I think the things that I think about, are superiority versus some, an animal model or standard of care in a clinical trial where something, if you really light it up, you've got something special. We've seen a lot, animal models have their own challenges and their own benefits, but I think that, if you can really get to a level of conviction that no one's really lit up this animal model before or demonstrated these clinical effects in an early trial, I think that can be something, that's stage agnostic, but I think to really demonstrate differentiation and superiority, that's exciting, and I think that's really worth investigating further. But I know that's kind of a squishy high level, but every one will probably have its own technology validation pathway, and earlier on, if you can stack the deck against yourself and then really demonstrate something special and unique and not seen before or rarely seen before, that's really compelling for me.
We have time for a couple more quick answers.
Yep, sorry.
As I mentioned before, I mean, we cover the whole spectrum from seed to typical VC investments to all the way to public companies, regardless of the stage. I would say we are putting more emphasis now in early stage opportunities, innovation, before proof of concept, because we agree that there is a valley of death, and we would like to help fulfill the pipelines of pharmaceutical companies. And with the guidelines, we hope to reach that next inflection point.
I think at a firm like Aberdare, it's a little different than a larger firm, but I think a small firm has to be very careful not to get involved too early because of the risk that I laid out. You want to get a certain amount of ownership, but you want to have enough dry powder. And so often that math just doesn't work. So if you look at Aberdare 4, which is our most recent fund, the one exception we made to that was Clovis, but that was a CEO. Pat's been a moneymaker, and Paul's been involved with his companies for the longest time. That's now a huge publicly traded company, and that was an exception, but that's the people factor. So there are things that can still make you go, I know you're going to need $200 million, but I know you're going to be able to raise it too. And the public markets will fund you. And so those are all qualitative factors that can change the thinking.
We don't really do that. We tell people, if you have an idea, come see us. Because we find out that these guys have an idea, these guys have an idea they've never met, maybe even different diseases, same mechanism they're dealing with. So we have the luxury of being able to say, you know what, come see us, and if we can't help you, we'll tell you. But we get the other question. Why should we come to you? And our answer is very simple, because you have to.
So I'm sorry, Sam, we're out of time. I'm sorry, everyone who has questions. I just want one single answer from each of you. Where do you think epilepsy innovation therapies, the most innovative therapies, will be 5 to 10 years from now? What will we have available to these patients?
God, it's tough. I would not be surprised if in my lifetime, so it's going a little bit farther than 5 to 10, if some sort of gene therapy or something where it's going to be replacing, having a real impact in intracranial gene therapy. It's a risk.
I'm very excited by technologies like ultrasound or things that are not invasive. I think that there has been more advances in that space, but we'll have to see.
I think that and the work that's being done on inflammation, and tied to that, how we're understanding that there's so many of our diseases are tied to the health of our gut, particularly autoimmune diseases. And we're hearing these stories about fecal transplants working. I mean, this is weird stuff, but this is actually bacteria being brought in and out of balance and having huge impact, including, of course, in the gut. Fecal transplants are for IBD, but we're seeing now in autism some very interesting studies coming out. So we're connecting the gut and the brain. Is something big going to happen there? We don't know. I mean, it may be a crapshoot, but I find that very exciting. What?
Well, as an American history major,
what did I say?
Micro.
The history of innovation is it will come from outside the field, and it will be something you never imagined. So keep your eyes open and look around.
Great. Sam?
I'm going back to molecular mechanisms of disease. I think we'll learn a lot more over the next few years, and it's going to tremendous difference in our understanding of the disease and our ability to treat it, just as this approach has worked in many other diseases.
Great. Thank you all so much. Great session.
All right. What did I put the accent from, or did I use the wrong one?
No, the cat and all the...
Formally going to introduce the Shark Tank, but the Shark Tank is up next, and we'll be setting up that for a moment, so this will be exciting and fun, and say thanks again to our investors for a great discussion.
Everybody should stay where they are, because if you are in the audience for the next 2 hours, you will magically have $75,000 of venture capital in your pockets that you didn't have when you walked in. So keep your seats.
Okay, everyone. We could start getting everyone in place.
2 minutes we need to wire up the sharks.
Okay. Give it 2 minutes. 2 minutes to wire up. They need 2 minutes to wire everyone up. Just go ahead and start, huh? Because
Maybe they're getting unwired.
That's it's a transition. Just go ahead.
Yeah.
Okay. Everyone, we're going to go ahead and start the next session that we're very excited to kind of get going, and that is obviously the Shark Tank competition. I am Dr. Joseph Servin. I'm editor-in-chief of epilepsy.com, and I have the pleasure of getting everyone set up at least as a prelude to truly the highlight of this conference. How can you beat spending an afternoon helping other people give away their money? And this is the way that we are going to kind of move forward. But before we get into the actual Shark Tank competition, it's important to kind of go back a little bit in the history of the Shark Tank and to kind of revisit two of the previous winners of the Shark Tank to hear how's it going as a way of kind of getting us started. And so our first person that I'd like to bring up is Mr. Charles Anderson. He was our 2012 Shark Tank winner for SAMI. And I'm going to have him come up and kind of guide us as to what's gone on with SAMI over the past 2 years.
All right, thanks. Hello, everyone. I'm going to start out with a video that we produced for our Indiegogo campaign that you've been hearing about. So why don't we start that? So this will give you a quick, here we go. Yep.
This is a quick demo of the SAMI Sleep Activity Monitor. The SAMI camera is installed on a wall opposite the bed. The iPod is located in your room. When the SAMI app is launched, it automatically connects to the camera and displays a live video stream. The camera has a microphone that is easily turned on and off. The camera is very sensitive to motion. In this setup display, the red shows the motion that the green meter is measuring. SAMI will ignore short movements such as rolling over. If motion continues, an alarm sounds. The display switches to live view and the microphone is turned on. All motion is recorded. SAMI makes it easy to quickly locate and view recordings of interest. Recordings made during an alarm are marked in red. These recordings are saved until you delete them and are easily shared with your doctor. SAMI is highly configurable. You may choose to use the alarm or just use SAMI for making recordings. When using the alarm, you may schedule the on and off times. There are also settings for alarm and microphone volume, for brightness, for motion detection thresholds, and region. This has been a very brief description of the SAMI Sleep Activity Monitor. For more information, please visit our website at www.samialert.com. Thanks for watching.
So why do we do video monitoring for seizure detection? Video monitoring has a number of nice features. SAMI looks for movement over the entire body, and it's not just looking for repetitive motion, it looks for any motion. In our case, for example, our son's seizures often would look more like sleepwalking. SAMI is easy to live with. You install it on the wall opposite the bed, and there's no sensors that you have to remember to hook up or patches to put on. It just sits there and monitors for you. It reduces parental stress. Somebody pointed out earlier that by looking, a parent can very quickly determine whether something is wrong or not. So we provide that capability. And you can just check in whenever you want. And finally, the video recordings are very useful. We're actually surprised by the response on that. The video recordings give you a way of showing a doctor what is actually going on. It shows you the early onset of the seizure, which may not be observed, especially with nighttime seizures. And it gives you very accurate seizure duration information. I know when I was asked about duration of seizures, I couldn't give you anywhere close to an accurate number for that. If you're dealing with it, you have no sense of time. So SAMI obviously does not work for seizures with no movement. And the movement must be fairly continuous. It doesn't have to be large, but it does need to continue. Movement can be fairly small. The camera is sensitive. The alarm doesn't work very well with active sleepers or kids that sleep with a pet in the bed with them, although we found in many cases still the recordings are useful. And there are privacy concerns, especially with older children. And there's privacy concerns coming up recently about webcams being hacked into. SAMI does not connect to the internet, so that's not a big risk, but it is a concern. And finally, SAMI is not a medical device, so insurance reimbursement can be difficult. So progress. 2 years on. We've shipped now over 90 units to people. We've been doing app updates every 2 months, roughly. These are just pushed out to the iTunes App Store, and anyone with a camera can download and install it. We've started design of a 3rd generation camera. The main thing that we're trying to do is make it less expensive to manufacture and ramp up the volume. And then another big update on Tuesday, the Patent Office granted our patent on the technology, so that just issued. Thanks. Next steps, we're going to continue to work on it. One of the things I want to do is make it easier to share the recordings. And we're starting to look at connections to other sensors. If there's somebody here that wants to talk to me about adding video recording to their sensor, I'd be interested in talking to you about it. So, really want to thank the Epilepsy Foundation for their ongoing support. The Shark Tank competition win was important, but they've continued to support us. We've teamed up on this Indiegogo campaign. If you'd like to learn more about it, just go to Indiegogo.com and do a search for SAMI, S-A-M-I. Again, I really appreciate the efforts of everyone here and the attitude of urgency, which is really important. Thanks.
I know that Mr. Phil Gattone has an announcement that he'd like to make, and if you could stay right here, it'd be great.
Charles, I just want to let you know that with the Indiegogo campaign, the goal was $90,000, and if you check it out, you'll see that Charles and Cynthia just got past the halfway mark. There's 7 days to go still in the campaign, and I want to give you an announcement and tell you some good news that due to the generosity of an anonymous donor, the Epilepsy Foundation is going to be able to provide a matching gift up to $25,000 for the next $25,000 raised, which will put you guys over the mark. So we are really grateful for this anonymous donor and the donation made, and we just really want to incentivize everybody here to help push Charles and Cynthia over that finish line. So thank you very much, Charles, and congratulations.
Now let's hear from the 2013 Shark Tank winner to hear about the progress that's been made since last year in Miami when his name was announced. And that is Utkarsh Demirci, and that was for the project Bioacoustic MIMS in Medicine and Lab. So Utkarsh, a pleasure.
Thank you. It's a pleasure to be back here again. Our goal has been to really develop a device that can measure from a finger prick volume. How do I switch to the next slide, by the way? Is there a button here that I need to use?
Right there, the green one.
All right. I'm supposed to be a technology developer and still yet having problems with it. But the idea is to be able to develop a finger prick device that you can use easily to be able to detect the levels of anti-epileptic drugs with devices that you can actually use and dispose in a microfluidic chip with a simple and inexpensive user. I need to see this slide. Oh, you have to? All right. So the idea has been always, how can we develop these chips and reduce the cost and issues related to developing technologies to be able to reduce the cost and make these available in a patient's bag or at the bedside so that these measurements can be done in a timely manner and you don't have to lose the time between actually seeing a side effect to the blood collection and going to the hospital to get clinical tests. And another area that such devices can be really useful could be when you have to study multiple drugs and You have to actually see the drug-drug interactions, and there is always an issue with adherence of these patients when you are testing new drugs. So you would be able to actually use a cheap, a few dollar test to be able to monitor from a drop of blood or from a drop of saliva or other type of fluid, because our technology can handle multiple types of fluids to understand the different levels of drugs in patient circulation. And this can be actually pretty much multiplexed on the same surface. Our device is extremely simple and extremely sensitive and specific. It uses particles that are gold nanoparticles that are self-assembled to a surface, that are the yellow parts that you see with the right molecules on top, where you can just take this plastic surface and attach to it, apply directly whole blood or saliva or other types of fluids so that you can capture the drug levels, and it requires just a single wash step. And over this year, we have been actually able to change this system so it can become a reader that is about $500 and a chip that costs a few bucks only to be able to monitor these levels using really a simple portable gadget that you see at the right bottom of the slide, which is just using the insides of the device that we have designed in our lab. And what we are looking at is pretty much the color change on the surface, which can give you actually data within the clinically relevant ranges of from few micrograms per ml to even higher levels. If you would like to actually look at the square there, that is our some serum-based testing that we have been optimizing this device for with the support from American Epilepsy Foundation within the past year, validating that these surfaces, these plastic surfaces that we used to use in old days when you had transparencies, where I would have less problem pointing out to the right slides, so those transparencies are now becoming your chips and devices to actually detect. In this case, we started with carbamazepine levels from serum, and it works with blood and other samples as well in a broad range. I would like to kind of point out that this technology can have on the same chip multiple types of drugs. We are actually going forward with patient samples. Once we have actually designed the system already and done these spiked experiments, we are moving forward to actual experiments with patient blood and saliva. And we want to actually, in addition to experiments with determining the drug levels, we want to do other functional assays that are important. This is an advantage of working with blood, being able to look at liver function and other measurements on the same chip in just a few minutes from the same finger prick blood sample of 10 to 20 microliters. In terms of commercialization, we had done a provisional within the past year around this application, and it's moving forward, and we have been talking to drug development companies and pharmaceutical areas in terms of getting this product out with Steve Zachter. I would like to take the opportunity to again thank the American Epilepsy Foundation as we move forward into a new year with this project, and it also allowed us to win a big competition in Brigham and Women's Hospital at the Harvard Medical School called the Bright Futures, which was around the same idea, and we are continuing our work. Hopefully next year, if we are here again, we will have more patient results and liver functionality assay results from actual patient samples. Thanks very much.
So now we'll get right to it, and Jackie is our moderator for this session, and we'll turn it over to her.
So this is what you've all been waiting for, I hope. This is the third live annual Shark Tank, and I want you all to think that you're the studio audience, and we have some rules of the road. We're going to be resetting the stage here a little bit, and the sharks are going to come up. So this is how everything's going to work. We have had a large pool of applicants. I guess they're the fish in the pool that the sharks are going to fish from. So we started out with 58 applications from all over the world. We had a call for applications, out of which we fished 6 finalists. And we picked these finalists because we thought that they had ideas, and these are any number of people, you know, from people who just have ideas in their own home, thinking about their own problems, but they know how to make them happen, to very high technology. So we've invited 6 people to come and present very briefly in front of you. So sharks, wherever you are, you get 5 minutes of presentation, 5 minutes, to convince us that your proposal, your idea is the one that deserves to win, that you're going to be standing here next year and telling us what kind of progress you've made. Now, this year we're going to run it a little differently. We have assembled an incredibly long-toothed set of sharks. Actually, yeah, sharks do have teeth. And they represent... Sorry, Marty. They represent, and they'll tell you about themselves, but they represent all different types of people who might have some reason to have expertise that can add to what we know about these proposals and contemplate these proposals. And I will tell you all, too, very importantly, that the sharks have been given access to a little more fleshed out proposal than you all have access to. That will allow them to ask specific questions. And for that reason, we don't want you guys to make up your mind about anything until you've seen all of the sharks and you've seen all of their, all of the applicants, you've seen all of their presentations, and you've heard our sharks ask all their questions. Now, after that all happens, that's when the fun begins, because we actually have $200,000 that we are going to be giving away this afternoon on the spot. Now, that $200,000 will be broken up so that each of our sharks gets $25,000 in their pocket. I just made it happen. Feel in your pocket, and it's right there. Now, we have decided that $25,000 is probably not enough for any of these ideas to really get the boost we want them to have. And so we have decided there will be no loan sharks here. And we don't like loan sharks anyway. So therefore, if a shark is going to give away money, he has to get somebody else to believe in that idea too. So the minimum amount that we're giving away is $50,000. So fish over there, that means that if you hear some shark say that they like your idea, don't get too excited yet, because they have to get another person to like your idea too. So the minimum amount we're giving away is $50,000, all right? The sharks are going to decide how they're going to apportion the money that they have in their pockets. And then after that happens, the fish over here are going to get another 15 seconds to tell the audience why they should vote for them. And then the audience now has $75,000 in your pocket. And all of you should be online. Make sure you're online. And we're going to get some information, and you are going to be able to vote. And you will see in real time how the votes are going on a screen in front of you. And then your vote is going to decide that $75,000. So I hope you're all ready. I hope you're going to be paying really close attention. How many people have the app downloaded? Okay, most of you do have the app downloaded. If you haven't downloaded it, you have another hour while this is going on, while the presentations are going on, to download that app. So get online, download the app, because then and only then will you have an input. And the wonderful thing is that the people in this room represent our whole community. They represent patients, they represent parents, they represent doctors, they represent researchers, technologists, the private sector. This is you. And so all of you are going to decide which of these ideas is worth $75,000. Okay? Are you ready? Okay. Sharks, take your places. Okay. Are we missing a shark? Oh, no, there's only 5. That's right. There's an extra chair. For me, but okay. All right, you guys move down then. Did you want to? They want to take this thing away. All right. Yes, that's the next thing. So the first thing we're going to do here, I'm going to move myself into the middle, sharks. after they take this away, is that, oh, they're pulling it back. Okay. So each of you guys is going to start out. We're going to start over here and tell us who you are and why you particularly are good to be a shark this year.
Is this?
Yep.
All right, it's on. My name is Andy Smith. I think I was put on here because I have epilepsy. So I'm kind of the young face that also represents epilepsy. I currently work at Credit Suisse as an investment banker. However, I've been involved in the epilepsy world for quite a while. I worked in the National Youth Council for a while. And then before that, I was working with the Epilepsy Foundation for a little bit as well when I was down at University of Virginia. Up in New York, I'm also trying to get involved. I'm trying to help start up a Circle for Epilepsy, which is a nonprofit, and just trying to help research. So glad to be here. It's great to see all the faces and the support. Hopefully I can help.
And I'm Milton, so is this on? It's not, huh? Yeah. So, oh, it is. Thank you. So I'm from Rochester, Minnesota. I'm with the epilepsy program at the Mayo Clinic. I'm currently the president of the American Epilepsy Society. I think I got invited here because this is my 35th anniversary of working with people with epilepsy. I just reflected on it when I wish I really have Andy's age and credentials being with the venture firm and Credit Suisse. But I think that's the reason why I'm here.
I guess I'm a bit of an odd man out. This is the first time professionally that I have interacted with epilepsy as a syndrome, professionally, I guess we could just say. I actually am in the ag business. I work for a company that focuses attention on orchards. So we grow almonds and pistachios and you know, try to better the world with healthy foods. I have a unique position in the company in that I actually run a small biotech within this ag company to understand how our phytochemicals, how the consumables that we take in, the oranges and the almonds and pistachios and pomegranates that we eat, how they may help us. I do have 2 people in my family who do suffer from epilepsy. So hopefully I can bring a bit of a professional thought that is outside of the realm of the therapeutic area, but also a personal touch to the thought process.
So is it almond or almond?
Depends on if you are a grower or a buyer.
Okay. But he's being modest because he also brought us Get Cracking and
yes,
and the Cutie.
And yes, Cuties, and Fiji Water, and Teleflora is also part of our organization. Yes.
Now they're impressed.
Now they're impressed.
My name is Marty Morell. I started out my life as an epileptologist, not quite 35 years ago. I'm more sharp-toothed than long-toothed, Jackie, all of us are. But, and then after working at Stanford and at Columbia, I met Frank Fisher, and 10 years ago, well, actually 16 years ago, started working on the NeuroPace project 10 years ago, pretty much full-time, although my current Stanford boss, Bob Fisher, has allowed me to maintain my activity as a clinician. So I've spent a number of years now learning about how difficult it is to deliver brand new therapies to patients. And that is not only the funding, but the difficulty of designing clinical trials, not knowing until after it's done whether you've picked right or wrong, and then the considerable regulatory hurdles. I'm now in the process of enjoying learning about commercializing a product after 16 years. So anyway, that's my perspective.
My name is Mark Cuban. I own the Dallas Cowboys. Mavericks. In fact, that's not true. My name is Jim Abrams. I run a not-for-profit foundation called the Charlie Foundation, which we started 20 years ago when my son Charlie's epilepsy was cured by a ketogenic diet. After a number of more traditional epilepsy treatment options had failed. And I became aware at the time that that Charlie's was but one of millions of kids who preceded him prior to finding diet therapy. And so we started the Charlie Foundation in an effort to get the word out that diet therapy can be a very effective therapy for kids with difficult to control seizures.
Thank you. All right. So now it is my pleasure to invite the first 5-minute fish, who will be John Davis, who will be telling us about shower power. Where is John Davis? There we go.
Hello, my name is John Davis. I'm here to present a safety device called Shower Power, and I've got a short video. Sorry.
Oh, they probably just unplugged it. Is it going to work or not, this one?
Hello?
You can use mine.
Tethered here. I'll stay close. Hi, my name is John Davis. I'm a inventor of the Shower Power. It's a safety device for showering, and there's a short video that we'd like to present in advance of my presentation.
The bathroom occurred in the bathtub and shower. Over 80% of these injuries were related to falls. Showering safety continues to be a growing concern. Shower Power is a shower safety monitoring system intended to provide protection, peace of mind, and showering independence. It was envisioned by Jonathan Davis, who has a background in chemistry and medical device development. In February 2014, John's youngest daughter, Claire, was diagnosed with junior myoclonic epilepsy. The inspiration for his design was developed as a way to safeguard his daughter after a seizure event occurred while she was showering.
I love my dad, and I am very proud he used his creative talents and ingenuity to develop a concept that will not only help me, but families everywhere facing the same concerns that we have with showering safety.
The Shower Power monitoring system consists of 5 interworking components for showering confidence: adjustable proximity sensor, shower water shutoff valve, phone base, relay unit, and a wall alarm. Shower Power, providing your loved ones with protection, peace of mind, and showering independence. Shower Power.
Well, there's the short video that we have. Let's get to the presentation. I think this is working right. This is the right button. There we go. Okay. Again, the Shower Power was a concept when my daughter was diagnosed with epilepsy basically 4 months ago. Our biggest concern was shower safety, and she did have a seizure in the bathroom area. And so what we wanted to do was provide independence for her, safety, peace of mind as a caregiver, and also for her as a patient of epilepsy. We've got a few statistics from New York Times, the CDC, and also from the Kansas City Star. And they're presented in graphical form here. Again, this chart shows the age group across the bottom and the rates of treatment in the emergency room on the y-axis. Again, these are rates per 100,000 people. As you see, the incidence in injury occurs as people typically get older. And again, in 2008, the rate was 22 million people went to the emergency room for falls and injuries in the bathroom. So there's a concern there. Broken down by sex, 2/3 are typically women, and the cause of injury is basically a trip or a fall. And again, this chart shows the activities that are most dangerous in the restroom, the bathroom, and bathing is up to almost 30% of those injuries. Some of them are also leaving and entering the tub area as well. So again, the shower is quite a dangerous place overall. And again, this chart shows essentially 70% of those trips and falls were in the bathroom area. And our experience as a parent was that, again, the first seizure from our daughter happened in the restroom, in the bathroom. And we didn't have an awareness at the time that she was having this seizure. And we wanted to provide her some privacy and autonomy. We were typically scared when she was in the shower listening for any incident that might happen in there. And we wanted to also provide in the future safety for her as she went off to college and gained her independence in the future. So again, the shower power is really a proximity sensing device that is looking for the upright posture in the shower. And if that does not occur, an event is triggered within these components. So essentially there's again, at this development stage, there's a time limit if a person were to go below the sensing device, and that would indicate that there's something not quite right. Again, this event would coordinate amongst the components. The water would shut off through a shut-off valve. This event would also trigger a phone contact. You could have a pre-programmed contact, 911, a neighbor. And also there's a proximity alarm nearby that caregivers could be alerted that there was an issue. Here's a pictorial of a typical showering situation, and shows all the components in the shower there. And if there were to be a seizure in the shower in this case, the components would be combined together to provide safety for not only the patient but the caregivers as well. And we'll go through these individual components here shortly. Again, the first component is the proximity sensor. It is envisioned as a mountable component in the shower. You can adjust the, essentially it's it can sense the level of the person in the shower. It's adjustable for their height. At this point, we envision it about hip height, so if they were to reach down or shave their legs, it would still be indicated they were in the shower normally. Again, we have the shut-off valve. Again, all these components are battery-powered, so there wouldn't be any concern of electricity in the shower. And they're all connected through a wireless connection. You can have Bluetooth is the typically known, but there's some other solutions out there, Zigbee, and there's some other off-the-shelf solutions for communication amongst devices. There's the phone base. In this case, we have an iPhone in the cradle here. This would sit in the bathroom area. This is the signal relay that relays amongst all the other components and make sure that signal of the event is triggered amongst all the interconnected devices. And then finally, the event alarm that might be in the hallway outside the bathroom that could alert the caregiver. And again, I have a short timeline here. Typically, this timeline comes from my experience with the medical device development. I work for a company. We have diabetes analyzers, and I help develop a product from napkin sketch through the FDA. And so there's typical device stages that we want to go through, essentially go, no-go steps to make sure that the design has been completed and the patents have been submitted for. At this time, I've submitted for a provisional patent, and so that's ongoing at this point with the patent office. So there's typically 5 stages to the program: developing the device, getting it through testing, manufacturing, some licensing agreements in stage 4, and then finally the 5th stage is, maybe that's the valley of death, which is the FDA. And again, this is an unknown territory. Certainly with device development that I experienced with our diabetes device, there was a lot of back and forth with the FDA. So it really is kind of an unknown question at that point. And again, I'm with a company. I'm the senior systems architect for the diabetes device that we have, and I led that device from napkin sketch again through the commercial development. And I've got a research team that helped me with that device as independent contractors, and they've agreed to come on board with me as well. So again, in short, we want to provide safety, independence, peace of mind for the patient and the caregiver, and the shower power components again.
So we are open for questions from the sharks.
Do you think you're going to have to go through FDA? Could this be a non-regulated device?
It certainly could, and I believe the SAMI device is clearly paved the way for not having to go through that route. I'm sure he may be envisioning the FDA route, but I do believe there's a possibility that it could just be an off-the-shelf component that people wouldn't have to go through the FDA device development process.
So you might have a product that did not require FDA approval
Yes.
first, and then consider your regulatory path.
Yes, that could be possible.
Then if I were a shareholder, I would imagine your market is bigger than in epilepsy.
Yes. Yes.
Right? We were saying about this nursing homes, for example,
Yes.
or in my home in a few years, maybe. And and you may not necessarily need FDA approval for that. I don't know. I have a question, though.
Yes, sir.
So you have described an idea. I really like the idea, but you didn't tell us about the method of sensing. Is it because you don't want other people to know? Or?
No. At this point, again, I've only had about 4 months to give this whole concept birth, and so we're working with the engineers on
so
how
you
to...
don't even know what your sensor is going to be?
No, at this point, it might be optical or ultrasonic to again, sense motion in the showering area.
Okay. And by the way, one last area, and I don't mean to chastise you,
Yes.
but I did check with one of our leaders here, and we don't use the word epileptic.
Okay.
Yes, in your paper.
Okay.
But we commonly make that mistake.
Okay.
Thank you.
Anybody else?
Yeah, do you know if it's so you don't know yet whether there be any interaction with a vagus nerve stimulator? If somebody with epilepsy, are you familiar with implants?
I've read a little bit about
yeah.
that.
So would there be a reaction or an interaction?
I don't believe so. Again, being a Bluetooth wireless device, I would imagine that those would not have any interaction with implantable devices. So that's a good concern, and we could be sure we're not having any interaction.
How much are you anticipating a unit would cost?
Well, at this point, again, it's an early stage, but I would imagine maybe the package for roughly $200 might be a good starting point, would make it an affordable option for patients.
And you gave us some stats on the number of accidents that do happen.
Yes.
If you extrapolated from that and you said, okay, that would give me a market of X in terms of just people, forget about dollars.
Right.
But you can answer dollars too.
Okay.
How big are we talking about? How big a...
Well, again, the latest statistics that I read on the epilepsy website was that there was possibly 2 million epilepsy patients, and certainly there is a potential, again, in the aging market and in other diseases which might bring more than the 2 million patients. And certainly, again, aging and also the youth market as well might boost the numbers past that point.
So, Dr. Morell, I was asked to ask you to comment about your experience, and maybe Dr. So as well, about patients and accidents in the shower.
Yeah, and this actually gets to a question I have. So I have had 2 patients in over 20 years, I gave it away, who fell in the shower and blocked the drain, one because they closed it, and the other just because their body covered it, that one died and one had a drowning injury with pretty significant permanent brain injury. But my question is, you showed the statistics for accidents, bathing accidents. That's a pretty broad category. So I see this happening. I see somebody dropping. They're still going to have the head trauma. What is it that, what do you imagine that you're going to do for somebody who has a fall in the shower as far as preserving their safety?
I think it's twofold. Certainly one is shutting off the water and preventing the drowning potential. Again, that was, as a parent, our first concern. Again, also is the proximity sensor through the phone and also through the wall alert in the general vicinity for caregivers to come to their aid. So again, I don't believe it will prevent the event. It'll just make it a safer event.
Will the interface allow for any other information to flow through the, I guess, phone application besides just the alarm?
At this point, I'm not sure, but certainly that could be built into the app and have some more statistics put into that call through the iPhone app.
So, have you developed a beta app Have you developed anything yet past the concept?
No, I haven't at this point, but I do have my engineers are, again, we developed this laboratory analyzer with, essentially with circuit boards from the ground up for this laboratory analyzer, so we have experience in software and engineering from the electrical and mechanical and device and design development stages.
So from a flowchart perspective, you've identified your buckets, you just
That's
haven't
correct.
run it.
That's correct, yes.
All right, I'm going to ask you for my microphone back. And thank you very much. So any of you, who want to see a shower power a couple of years from now, think about that when you're making your vote. The next person I'm going to invite up here is Nirav Sheth, who is going to talk to us about the BioStamp wearable sensor.
Thank you, Dr. French. I'm a vegetarian. I don't know if it's going to help me well. I don't think so.
They would have to be vegetarians.
Well, now the question is, do you bleed?
I do, and that'll probably show. So my name is Rav Sheth. I'm with a company called MC10. We're a technology startup in Cambridge, Massachusetts, whose mission is about bringing conformal electronics to challenges in life. We have brought some interesting applications and solutions to consumer healthcare and to performance athletics and fitness, but it has always been a passion of ours and a mission of ours to go and try and apply our technologies into healthcare. So it was a pretty exciting opportunity for us when we came across the Shark Tank event here at the Pipeline meeting, and thank you also for the panel for selecting us to be here. So since we are new to epilepsy, we did probably what most companies would do who are smart to begin with, which is go ask people who actually might know what the unmet needs are in the space. And so if we could cue the video, this is what we heard.
I think it's hard for something to make a transition better. I wish doctors knew more about how specific medications react to specific bodies.
With epilepsy, you really need to know the effect of the medication on the patient.
The doctors, to properly or fully diagnose the situation, needed more accurate information, and that's when I was admitted to a hospital. And at that point, it was still monitored by my parents. In the hospital, what they could do is just be next to me, and at night they could tell I was having a seizure, and at that point they would press a red button. So that would immediately go into the history of that night, and that would more closely identify when I had seizures and how long they were. My mother made this book. Well, this just has when I had seizures and when I didn't, and also medication I was taking at one time, when it changed, by how much it changed. You know, was I taking 100 milligrams, 200 milligrams? Was I taking it just in the AM or the PM or in the afternoon as well? At that point, she was very much relying on me telling her about my seizures, and to be quite honest, I did lie a lot about that, just because if I told my parents exactly what was happening at certain times, I knew that they would be more upset about it than I was.
Our frustration was in getting her to take her medication. The side effect of the particular drug she was taking was weight gain. She wasn't taking her medicine. She was either spitting it out or going into the other room and flushing it down the toilet. When she got older and got to be an adult, it was much harder to monitor because you, as an adult, you couldn't stand there anymore and say to her, okay, take your medicine.
I needed more than just my resilience. I needed a doctor's opinion of how the medication was affecting me. I guess if there was some type of device that I could carry on me that an automated device that could just capture every episode that I had, if it could put a timestamp on it. And if that would detect every seizure and report back to much more accurate data to my doctors and to my family. If something like this could record my seizures accurately, it would certainly take the place of people, my parents, my friends, monitoring me, and it would certainly save them a lot of time and, you know, give my parents much more sleep.
What we're doing here in MC10 would be very useful to me in monitoring Christy's seizure activity and how her body responds to different medications.
So that was kind of abrupt. Okay. I'm here somewhere. Point it in every direction.
Pointing at front is probably best.
Can we just go to the next slide? Okay, we'll stop there. So I lied. I'm actually not a vegetarian. But I was trying to get away with something, which is something that we actually found in several interviews we did with adolescents as well as parents of adolescents with respect to epilepsy. And so when it came to what could MC10 do about the situation, we were pretty excited. We focus on conformal technology, conformal sensing, and the device that Pat was holding up in his hands is the same thing I'm holding up here. It's just orange for visuals, but there's actually a nice one here with electronics in it that I'm going to pass around to the sharks. And so to just build the points, I think, about what MC10 could do to help persons with epilepsy, it's about passive wearability, or passive sensing and wearability. So if you look at the pictures on the screen, you're looking at pictures of adolescents, teens, folks who have a desire to live their own life and do not want to be reminded that they have a condition. And so if they want to live their lives, how do they continue to monitor what they want to monitor but still go on living? And so the idea of having a wearable skin patch that could be positioned in multiple locations, it could be on the arm, it could be on the torso, the leg, that makes a big impact. We could have multiple ones. We heard yesterday in the pipeline meeting about the need for multiple modalities. We have the opportunity to do that as well with these patches. And the other thing that's sort of not shown is the fact that these are all folks for whom communication can be a barrier. The accuracy of communication, having the right language to record something, that may affect teens who may lie, but it also affects the folks who have the seizures and the other events that happen at nighttime, for which there is no recording. So the other things that we can offer. Okay, so what you're seeing here is a picture of the diary. The fact that you have the ability to sense things electronically takes what is subjective information, which is difficult to stay consistent, right? Even in the transition from just childhood to adulthood, and a diary that was originally kept by a mother and now being recorded in by the patient herself or himself, or someone who goes away to college and now even changing physicians. Changing from a subjective, both recording method over to an objective one from a sensor-based technology adds value. We have also, while we've been focusing on hardware and technology, been focusing on software. So MC10 actually comes at epilepsy from a small history now in movement disorders, where the idea of tremors and the idea of diaries has already been prominent, where in fact billion-dollar drugs have been approved by the FDA partially and sometimes significantly based on data that comes from improvements in patient diaries. So replacing those diaries and that subjectivity with information that can go on an app, the communication to a smartphone that's recording not just the objective data from our sensors, but in addition, data that may come from the patient themselves or other inputs as well is critical, because we know that epilepsy, and if you didn't know before, or if I didn't know before these last couple of days, I certainly know now, it's as much of a lifestyle impact as it is a clinical one. The communication piece is critical. This gentleman here may be long-toothed or sharp-toothed, I'm not sure, but he may be a physician, he may be a father, he may be the patient himself, but the idea is that if we can take the information, turn it into something that is usable and trackable, which is clearly possible with the cloud and with our own apps as well. We can transmit it to whoever it needs to be transmitted to. So what is the guts of the device? This slide actually shows to everyone, I think, basically what we're talking about. So this is the device that's, I don't know if it transitioned over. Good, as long as everyone had a chance to see it. It's a silicone encapsulated patch that's, thanks, Dr. French, I'll just hold it up. So it actually is very flexible, and this is something that has the ability to measure. It has a 3-axis accelerometer in it, similar to what you might find in your smartphones, in addition to having a battery, a Bluetooth radio antenna, as well as the ability to measure biopotentials with electrodes as well. Sorry, thank you. So all those things are possible with the device. And finally, taking a step back from the individual or the patient back to sort of just therapy in general and the efficacy of care. The reality is if we have this sort of data, and we know that there are data issues that have to be resolved as we've discussed over the last couple of days, but the access to that data, as is shown, I think, by these two articles that we found pretty interesting, and I think have both quotes and perhaps authorship from folks up here on the podium. At least, Dr. French, I recognize your name multiple times. The fact is, having better information does help drive the advance of diagnosis and therapy of conditions, and hence research, and and improve the search for a better care. So for a better cure. So those are all things that we believe are enabled by our technology. So what we need, why we're here, why I'm here, is we need partnership. We don't come at this from a deep knowledge of epilepsy. What is clearly present in this room, both in the companies, the companies who are startups, the researchers, there have been funders up here as well, has been the fact that we need to all share that information. The Epilepsy Foundation has already been quite kind enough to give us this podium to speak from, but the physicians who are shown there, in addition to the ones that you see here, Dr. Dorsey is a pioneer in telemedicine and Parkinson's, and someone that we've been fortunate to have a relationship with here at MC10, in addition to identifying some of the folks who have been really pivotal in bringing more patient engagement. And PCORI, which has come up as well, which does not have actually, when you do a search of their database, that many epilepsy-focused grants given out compared to some other disease states. Those are all things that we actually think that we would want to change, and with your help, we could. So the plan over a certain timeline, and I'm sure our dollars and timelines will be different through each presentation, but our idea is to show a proof of concept for potentially a particular kind of seizure or a group of seizures. It might be tonic-clonic, it may be partial, focal, what have you. We want to go find out what are the right sensors, therefore, to put in our hardware and to detect what and over what time period. That's where we would really need help. Over a period of time, as you can see from the prototype, we could create prototypes that would then be able to be placed on humans to just prove out certain ideas. Targeted validation, we believe, would take some time if we want to validate it from the standpoint of creating information that would be compelling enough to physicians to be able to work with their own patients to recommend something. We believe that would be necessary, so we've identified a length of time to do that, as well as a significant dollar investment as well. And finally, if you really want broader adoption and real scale, electronics is pretty funny. That's the price really comes down when you're able to achieve scale, and we believe having the ability to sell millions of these things does bring consumer-type price points into play. And so this would be something that would be really important, but would obviously take some time as well. So that's essentially our pitch, and I think if we think about what our mission is and the journey we would like to start with all of your help, I think it's very similar to the journey that Pat describes on the next video.
If I can help, a boy who's 6 and whose family is up at night and thinking about seizures and surgery just like my family was, and if I could make his or her life better, then I want to be a part of that.
So thank you to the foundation for allowing us this opportunity. If Pat looks familiar at all, it's because he ought to. He is actually on the CDC website. We were incredibly fortunate for him to actually be a friend of a friend. So great thanks from us to both Pat and John, who were there on the video. And questions?
Thank you very much. And I do have to take a couple of points away because you couldn't find Andy's picture on Facebook.
The silent face of capital.
All right, the floor is open for questions.
Yeah, I was, granted your video was interrupted, but I had trouble figuring out whether you're trying to track drug reactions or seizures.
That's actually a great question. I think we walked in probably somewhat naively with an idea that we could do a great many things. Actually, after listening to some of the ideas yesterday around the correlations of temperature, EMG, and other aspects to certain types of seizures, we believe we may actually have a shot at some of the seizures because we do have the algorithm capability. But that said, I think that's why that first step is so important. I think in speaking to both patients and clinicians, we would probably be able to narrow in on a specific target a lot easier than we have been able to thus far.
Yeah, Marty.
So I share Jim's confusion. I don't know what product you're talking about, and I'm not sure that you guys have spent a lot of time looking at what else is going on in the community, because we've been talking about seizure diaries and external wearable seizure sensors for a long time. So can you tell us what you offer beyond what we already have or what we've heard today we'll probably have in the near future?
That's a great question. So I think there's actually two major things going on. One is from a simple monitoring standpoint, we have the ability to obviously monitor on multiple parts of the body. So to the extent that you can get information from just the wrist, which was a prominent location that was mentioned yesterday, that's great. But the reality is there are things that happen elsewhere in the body that are important to specific types of seizures. So for example, to the extent that we would want to measure something from the calf or the arm or the torso, if we want an EKG response, those are the kinds of things that we could offer.
But you don't have this device yet, and you can't tell us that you could actually do these things. Is that right?
So actually, because of our recent experiences in movement disorders, we know that we can actually write algorithms to detect certain kinds of tremors. So that is a level of proof. It is not the full point, clearly. We've actually also done feasibility with EKG and with temperature as well.
Yeah,
no, go right ahead.
Oh, go ahead.
So talking about scalability, as I'm sitting here and I have my fuel band on my wrist, is what you're talking about hardware or software? If it's hardware, then it becomes more highly specialized. If it's software, it would seem that it could be baked in, theoretically, into wearables that could be sold to almost anybody. You just pick the right app or the right software, and that would drive down your costs significantly. So could you speak to me a little bit about software versus hardware and how cost structure would take place?
So there's two very different skill sets going on. I think one is just about identifying what the right sensing modalities are and the right locations in the body. And so to the extent that you haven't locked that yet, you don't have a hardware solution yet. Once that is locked, and I've given you sort of one application of it or one prototype of that that's here now that we're showing, once you have that locked in, then you can get that to scale, and then that drives those costs down. On the software side, what we find is that there's actually a series of different types of algorithms that can be written. If you simply want gait, that's great, but if you want fall detection, that's another one. And those are incredibly portable and have very little marginal cost. So I think those are good points. I think it all comes back to what is the most effective application of this technology to patients now, and that's where we would start. We could do both.
I think Elson and then Andy.
Okay, I'll just make a quick question. And the situation is that, as far as I know, there is not a reliable set of parameters yet to say there is a positive predictive value of, let's say, 92%. If you see this set coming up, this patient had a seizure. So you are developing a very, very advanced type of device when the parameters have not been validated. So there seems to be a mismatch. Not that you shouldn't go your way. I mean, this is important. But is that a sort of valid concern?
I actually would turn the concern into an incredible positive for persons who have epilepsy, which is a lot of what we've driven in here. As I mentioned at the very beginning, we've actually started in performance athletics and fitness, as well as in consumer health. So a lot of the scale of what we're investing in and what we're trying to develop actually does come at this from a different input or impetus. So you're right, this is pretty involved and very evolved as well, but it wasn't originally intended for the epilepsy population per se. It was intended for other pretty large-scale applications including, for example, movement disorders, as I mentioned, as I showed the Parkinson's app earlier. So as a result, what we would love to do at MC10 is partner with the folks here to find specific applications of something like this that actually you don't actually have to develop from scratch.
Thank you.
We don't.
Could you better explain the reporting process? So is the information flowing directly to the doctor, or is it going to the parents? Or I mean, I guess it depends on who your target is, a child or an adult. But.
No, that's great. I mean, the ideas of data, who owns it, where it goes, are actually, I think, continually evolving in the healthcare space, and if actually that's also true in the consumer space as well. So we have the ability to send this to the cloud. It's possible. In fact, we had a startup yesterday who came up here, and they've already built all that infrastructure. There are pharmaceutical companies, consumer companies who are all actually prominently announcing their ability to do that. So for us to be able to do that is clearly possible. I think what ends up happening is, do we want to use this at the very beginning for a specific clinical research trial for a particular drug? In which case, the way in which that data may be managed for that application may look very different than something that's in-home use.
So you plan on doing it in-home initially, or?
Our end goal, frankly, if I didn't say it, I'd like to say it again. I mean, we're a very patient-centric company. We're a people-driven company. So our goal is not to develop technologies for this like this that just simply get used in limited ways.
Right.
It may be easier to get into clinical trials sooner, but our end goal is to have something like this available for everybody.
Right. Okay.
One last question.
Real quick question. How long-lived is this piece of hardware, one? And then two, do you need to have your smartphone with you in order to constantly be uploading information, or does it have some type of built-in memory?
So yes, it does have memory, and so really that comes back to the application. You have the ability to map, I think it's 20 to 30 feet on Bluetooth Low Energy,
okay.
Or 20, 30 meters, sorry, forget which one it was. But so that's the distance. It doesn't have to be right next to you.
Okay.
And then in terms of the storing and what you want to transmit, you have the ability to stream constantly. That simply drives greater battery needs. You also have the ability to sort of store some memory and then transmit it over time or certain times of the day, which obviously uses a lot less energy.
Okay.
Thank you very much. All right, so. If you would like to see the BioStamp wearable sensor come to the marketplace, you will have a chance to place your vote. So next, Sandy Helmers is going to come and talk about WebEase, Enhanced Youth Epilepsy Management Behaviors.
Thank you. And it's a pleasure to be here, and actually an honor. So I would like to talk to you about something that we have to switch gears for, and that is, this is not a seizure detection. This is not an assay for blood levels. This is not a therapy. This is actually a toolkit for self-management, and it's for youth with epilepsy. And with that, how many of you have teens? I have bunches of them right now. And you know the lives that these kids lead, the lives that you lead because of their lives, the stress, the sleep problems, the coping with friends, the daily activities, parents with carpools and organizing schedules. Layer on top of that medication adherence. Layer on top of that the possibility of seizures. And you have a disaster waiting to happen sometimes, right? So what I want to talk about is epilepsy self-management. So what the heck is it? Because I find that a lot of people don't really understand this. So what it is being able to allow someone to learn to better control their epilepsy. That means adherence. That means managing sleep. That means managing stress and whatever else. Better control the comorbidities that go along with epilepsy. Mood. What do I do with my friends? Do they know? What happens if I have a seizure in front of my friends? What the heck am I going to do? The stress and the anxiety. Well, self-management for youth exists, but it exists for asthma, cancer, diabetes, surprise, not for epilepsy. So what we want to do is take a product that we already have, or a toolkit that we already have, called WebEase, that is created for adults, by adults, and actually adapt that for kids that are in the teenage years for use to better self-manage to actually help in the transition of care. So let's talk about that. So what the heck is WebEase? WebEase is a self-management tool that was created by adults for adults with epilepsy. It's evidence-based. We've already done a handful of randomized controlled trials that show that self-management, self-efficacy, and actual medication adherence actually do improve with this tool. It's free. That's a novel thing. It's internet-based, so access issues become less of a problem. It's interactive. And also, as I said, created by adults for adults with epilepsy. So it's very patient-centric. What we want to do is take and adapt, I don't know why this keeps doing this, adapt this for 12 to 17-year-olds to create WebEasy. Now, we envision to have a couple of units that will address medication adherence and also coping skills. This program, the WebEase itself, actually tracks seizures, tracks medication adherence, so do you take your medication correctly and how often, those sorts of things. It tracks stress levels, your sleep quality, and also your seizures, which are inputted by the person with epilepsy. We'll take all of that and put it into a program for kids, but make it more likable by the kids, more interesting, because all these kids, the teens, what's integral in their life? Technology, tech games, internet, Instagram, blah, 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 blah. So we're going to actually have to use that for the advantage of being able to get this to the kids. This is just a mock-up of what it could potentially look like, because what we're going to need to do is do focus groups, and this is my public health hat, focus groups to actually talk to teens, what they feel would be important for them to actually use this and to continue to use this. And what you can see on here, it may be a little difficult, but this would be like the landing page. It would be interactive. So you could potentially have an avatar talk to them and take them through things. You have a home button. You also have the different modules or the units on there so they can access it from this page. You can do My Diary, enter your medications, identify what kind of seizure you have, also how well am I doing. Then way up in the top on the right-hand side, you see this points. This is value-based, we could call it gaming points or value-based point system that would reward them for using this. And they could earn points to use, let's say, I've never played it, but one of my kids does. I want a tool on Minecraft, so let me go get it. Spend those points. Or if they're not into Minecraft, but they would like something else, could they use those points to actually then cash those in? And this is one thing that we'll have to do is look at partnerships with gaming industries, those sorts of things, because there's no way in a short period of time and with limited funding to be able to do that. Plus, we just don't have the resources. And it takes a lot of time and effort to do that. But that would be the landing page. So, in essence, fulfilling the epilepsy needs of youth and parents, because parents would be involved in this because they have to give permission to use the internet. We could push text notices to say, your child has done a great job with this, and this is where they're moving and actually start dialogues about their epilepsy. It would engage them. It's personal. This data is stored, but it's all HIPAA compliant. Independence, learning, all of those things all translate into youth epilepsy self-management. Now what I'd like to do is end with this video.
This program is brought to you by Emory University.
So it doesn't dictate your life. I see a lot of kids in my practice, and they all have epilepsy.
Dr. Sandra Helmers has spent the majority of her career figuring out ways her young patients and their parents can better manage seizures caused by epilepsy.
You want to have the children, number one,
take ownership of their disease, and that is sometimes difficult. Concerns that we had were, are you always taking your medicines? You want that independence, so we're not always hovering over her.
David Loring says coping with the stress caused by trying to keep his daughter's epilepsy under control has been a lifelong struggle.
It has effects throughout the family, and that's one of the things that we're constantly dealing with, because now we have a kind of a sick child who's in charge. We're worried about seizures if we go to Disney World.
Xavier Austin has had epilepsy since he was 2.
Yeah, I was bullied when I was little.
Although he hasn't had a seizure in a while and is well adjusted now, he still remembers a time when the condition isolated him from his peers.
I guess I acted like I had a problem, and I guess epilepsy was the problem.
It's making all of this up right now.
Many kids with epilepsy struggle to fit in socially, and the problem is often compounded once they reach middle school.
Middle school was a nightmare. I think it's a nightmare for a lot of kids.
They're at a vulnerable stage where you need to give them tools to feel confident that their epilepsy they can manage, also confident that they can have good peer interactions, so you build that confidence in steps.
Dr. Helmers and other experts at Emory University are working on an easier way to make those steps more appealing to teens with epilepsy by modifying a web-based training tool called WebEase.
WebEase was initially created for adults, and we actually have validated and proven that your self-efficacy, your ability to do the self-management, and not only for a short period, for a longer period, works. We've shown that in the adult population. What we're doing now is we want to adapt this to the 12 to 17-year-old age group.
To provide them with skills about how to better, for example, take their medication, or to better manage their sleep, or to better manage their stress. So that's I think the core of WebEase would actually show you.
Dr. Helmers and her team are currently surveying young people with epilepsy to determine specific ways the interface might be made more enticing to a younger audience.
It's work, but it's going to be fun. It is going to keep their attention, because what we want to do is we want them to engage. They can actually choose a goal that will fit them, that will be attainable, and then go back, check out, and see how they did. If they need some more practice with that, they can do that. If they have successfully met that goal, then they can set another one.
Fun and games aside, the youth version of WebEase is not intended to be a permanent crutch, but a tool that will empower the child with epilepsy to learn coping skills that can last a lifetime.
Well, I think anything that's fun and engaging, kids are going to be drawn to.
They're going to feel more safe around themselves, around others, and just have a sense of, I have this under control.
And if you can empower that child and the family to actually take control of that, you have a different family, a different kid.
The preceding program is copyrighted by Emory University.
So I just want to say thank you, and just to leave a couple of words, that this is empowering in a really important group for transition of care, and also for parental confidence that their child can do this.
Okay. Comments from the Sharks?
This is all, it's great, and we all need all the help we can get with adolescents, whether they're our patients or our children. But Sandy, I have 2 questions. One is, you have WebEase, so you need to tell us whether it's been working and whether people want it and whether they use it. Granted, this is for adolescents, but are the adults using this? And the second is, it seems to me there's a lot of stuff out there for teens about disease self-management, teens with epilepsy, maybe in bits and pieces. So what have you looked at what else is out there, and what more would you be able to offer?
So it was created a number of years ago for adults, and adults still use this. It is actually available at webease.org, and the uptake is there and continues. As far as for adolescents and chronic disease management, as I said, asthma is a big one. I'm most familiar with that. most of the self-management at this point in time is face-to-face, or it's getting the parents to enable the child to do the self-management. It's not e-health, which I think is a big advantage, and it's free. And it will engage with, on a platform that they all use now. And so I think that's what this actually brings to the table. And again, you can't take a chronic disease management for asthma and I think translate it to epilepsy, because epilepsy I think is very special.
I'm going to give a shout out to Andy here, who once upon a time was an adolescent. So.
Yeah, still am.
Closer than we are.
I guess one question I have is, how many questions does it take in order to do the reporting process? Knowing that our generation is a little more ADD, and I sometimes can't even fill out some forms because they're a little too long. I guess if I was to have a seizure and then wanted to report it, if it was more than 5 or 6 questions, it would be extremely tough for me to want to use this application, because I'd say it's not worth it.
You're absolutely right, and that's one thing that we need to do to adapt the adult version, because it is text heavy and it is a pain.
Will you still be getting enough information?
Yes, we will. Basically, that's all you need to do, and just it's not included in this, but we have a mobile app also that is going to follow the web base, because a lot of kids have smartphones, and it's not just going to be for iOS, it'll be for the Android also. And they'll be able to upload that information just like that.
And then are you capable of remembering answers so that they can then?
Yes, yes. Okay. And it will, again, be stored short term, but we can always store it longer term for on the cloud. But again, and this is one thing, Emory and I think a lot of us are, it's HIPAA compliant and totally confidential.
Yeah. Okay, and then another question, in terms of, I guess, moving over to the mobile gaming and trying to have that as a partnership, one concern I have with that is that, do you think people will start to abuse it for the points? I know that, again, they become very addictive. You'll see people playing games 24/7. Is there any concern that people will start reporting seizures just so they can get points?
That's possible, and we have no control over that. But again, with parental control of this, they'll be there in the background, because they first of all have to give permission to allow them to go on the internet. Okay. Second of all, with the parental sort of oversight that we'll be able to sort of help with as far as pushing texts and things like that, that might be a stopgap for that.
Yeah, just a,
that's a good question.
Point of concern though.
Mm-hmm. Yeah. Okay.
Thank you.
One more question.
Oh. It's okay.
Okay.
No.
So, we've just heard a couple presentations, and these presentations were about products that were capturing in an automated fashion data. Yours is the opposite direction. Can you speak to us a little bit about movement of data and accuracy of, self-reporting?
Well, that's a good question, because we've struggled with that. And we do not marry some of these automated seizure detectors, but this is more than just a seizure detection and medication monitoring. This is a self-management tool. It's a toolkit to let people learn about self-management, why it's important, and to set goals. So it's like if you wanted to lose 20 pounds and you had to change your diet and change your activity, you just can't do it just like that. You do it in steps. Or you wanted to quit smoking. First of all, you have to figure out, well, yeah, I would like to lose weight or quit smoking. Then how do you do it? And this program actually goes through with this motivational interviewing, set stages, and then feed information as far as the level that you're at. Are you willing to, do you want to hear about this, or you're not ready yet? And give you the tool to say, okay, I understand, this is a goal I can set, and do it. And then you check in and say, I've done that goal, and you're starting to learn. So it's a learning thing, and not just a detection.
Last question. I'll use the word analog inappropriately, okay? How quickly can you get to your avatar stage with it not being stalled in this analog version where it's just maybe clicking a button or typing a text, where you can actually have some dynamic interplay that will really engage the child into wanting to perform as necessary with the program such that you get the feedback necessary to.
That's a good question, and that, what we're doing is we're working with our IT department, and that, you know, we have the group, we have the self-management experts, we have the Emory IT folks who've worked on the previous version and the mobile app, and what we need to do is then change that interface, and that's what they're doing.
This is 3 months, 6 months, 6 year?
That's going to probably take about 6 months. We do a month to 2 months of focus groups, probably about a month, and then we're going to do about 6 to 7 months of software adaptation based on the focus groups input. Those are teams. And then that takes us to about 9 to 10 months. Then we're going to roll it out.
All right. Thank you very much.
Thank you.
All right. Next, I invite Oren Knopfmacher and Professor Bob Fisher returning to the stage to talk about an in-home and inexpensive anti-epileptic drug monitoring device using saliva.
Thank you. Oh, Jackie, you said fish. I thought you said Fisher. That's why I came up again. No, all kidding aside, I'm up here as a partisan because I'm a co-founder with Dr. Knopfmacher about the technology to inexpensively and portably be able to measure every day your anti-epileptic drug levels and other drugs as well, as we will hear. The company is called Avails Medical. To avail yourself of an opportunity, but if you take a moment and scramble the words, you may come up with some other relevant bodily fluid in the name. So the point that I made, marketing is everything. The point that I made yesterday stands, which is the biggest problem that many of us clinicians, including me, have in caring for patients is inadequate information. And furthermore, it's really critical, I think, to empower the patient and the family to have information to take care of his or her own health. And this will be a breakthrough technology that will let this happen in a way it has not happened before. In my view, the best seizure is one that's avoided. And right now, if you talk about serum levels in a medical meeting, even though I know there is excitement about it, and we just heard about another technology to do it as well, often eyes start rolling, and they should start rolling because we are terrible at understanding what levels of these drugs mean and using them in a proper way. The usual approach is to have a patient have a seizure, call the doctor and say, I just had a seizure. We say, gosh, I'm sorry. I'm going to add you on Friday. And of course, because they had the seizure, they just got scared to death, and they just take a bunch of their seizure medicine, which they may or may not have been taking before, and they have a level 2 days later in clinic that has nothing to do with the level that they had when they were doing the medicine. And by the way, why are we doing this after the seizure occurred or after someone has become toxic? So the Avails approach is to have people measure their level every day by spitting on a disposable, very cheap strip made from a type of transistor that you'll hear about in just a moment, sticking the dry end of the strip into a very portable reader, and then having a readout immediately of what their level is. It's also possible, although we've not enabled it yet, to link that to web-based advice programs. So our prototype was to pick the medication that's most used in the epilepsy clinics right now. That 25.6% that's up there is for levetiracetam, Keppra, but we could also do it for lamotrigine, topiramate, carbamazepine, valproic acid, or a variety of other medications. We use salivary levels not only because you don't have to stick your finger, but because they better reflect what gets into the brain. They're unbound. And when I was a young whippersnapper in the epilepsy business, many a time I would send saliva to the lab for measurements. We're translating that into the home environment. So the advantage is getting ahead of the seizure toxicity curve. I won't read the whole left side of the slide for you, but again, what we are doing is we are allowing people to take control of their own health and to stay healthy and to avoid problems. Now let me turn over for the rest of our 5 minutes to my colleague, a PhD physicist trained at Stanford, Dr. Oren Nafmacher, with the propitious initials OK, who's worked for 10 years with these field effect transistors, using them to make all kinds of very precise measurements. And I'm very pleased to have been able to recruit him into applying this technology to the medical problem with epilepsy as its first proof in principle. Oren, please take over, and this is the forward button.
Thanks, Bob. Well, with every technology which is available, there must be also a way for a great market opportunity. So our technology, which you're going to show in a second,
talk to the microphone, please.
Everybody hears me, yeah. Our technology, which we make available to anyone, basically, is cheap. We use disposable strips, which you can buy and then just dispose after use. Our technology works in any body fluid, so we can use saliva, as we just heard. We can even use it in blood, in urine, or other body fluid which are available. And not only that, once we made our technology available for all the epilepsy medication which are around, we can also scale it to any other vertical. So you can imagine using it for people who have heart diseases, people who need to take blood thinner. You know, there's a lot of potential for technology out there. So basically what we have developed is a prototype where you have like a simple handheld device. It's portable. It's easy to use. It gives you the information you need. You have on the other side disposable strips, as you can see in there. What you do is, or what we do on our end, we functionalize them. We make them highly specific to the medication you want to use. So every strip which you have is specifically to the medication you're using. And what happens is you take that in your mouth, you spit on it, you put it into the reader. The reader gives you the information, and as a physicist, this is the information you can read as you see on the graph. So we measure basically some electrical properties, and this changes with the concentration. Now, you as an end user will definitely not see that. That's for me as a physicist to play a bit around, you know, and see some stuff happening. But for you as the end user, you will see a number. If you're in the therapeutic range, yes, no. And if you're not, this might be even give you a signal to the physician, so you can, or to a caregiver who can help give you assistance in order to get back to the therapeutic range. So this is what happens if we have a highly specific sensor. This is what happens if we don't make the sensor selective. You can see that it doesn't respond. So this is how we differentiate between the different medications by making them selective. Now, we heard from the winner from last year that they're also targeting doing similar things, but optically. So the problem with optical devices, as you already pointed out, is that you can't like miniaturize that as we have here, or using this, because optical systems are very hard to control, and you need a lot of knowledge and highly skilled properties in order to be able to use them. So in order to make technology happen, we need a great team. And we are all Stanford alums, consisting of physicians, physicists, biochemists. We have also a regulatory expert on board. And if you have seen, the majority are basically females. So we not only are from Stanford can make this technology happen, we also have a great supporting team of advisors and investors. And with your help, we'll be able to improve the quality of life for epilepsy patients.
And it seems like last year's winner is now at Stanford, too.
Thank you. We're open for questions. Yes, Jim.
I have a couple questions. Is this for monotherapy?
Oh, sorry?
What if somebody's on 4 drugs?
So we can develop different strips for different drugs, or you can even put different its sensitivity, selectivity is on one chip. This is possible.
And you can detect levels of
individual ones.
My second question is, what if somebody is in ketosis and also taking medications? Do the strips factor in ketosis?
Yes, so it's a good question. For those of you who are not in medical, ketosis would be an acidotic state with ketone bodies. So the specificity of these strips is extremely high. So it's not going to be influenced in general by ketone bodies. There are calibration procedures that can happen right on the chip that take into account acidity and other physical factors. But ketosis, although we've not tested it in ketosis, I'd be extremely surprised if ketosis affected the readout. See, this is a field effect transistor, which passes current through it, a gating current that is influenced by the charges in the vicinity of the transistor. And so if you put different charges in its vicinity, it will change its current. And by using something like an antibody to make it specific to one drug, then only when that particular drug binds will that charging current change and give you a different readout. So you can be very specific.
Marty.
So what effect does spit concentration have? And the second question I have is, have you done work showing that these levels correlate with what you'd get by standard serum testing?
Next, we can show that the one prototype is sensitive to levetiracetam levels at different concentrations. The next stage, which we have not done yet, is to develop the use protocol, which would tell the patient how to use it, just like they would for, say, glucose testing in diabetes. So, for example, you wouldn't want to do this right after you swallowed a Keppra pill, because the concentration would be high. You wouldn't want to do it probably right after you'd consumed certain foods, or if your mouth was too dry to spit. So we have, believe it or not, at Stanford, you may know this, Marty, because you're there is a saliva center, and we are collaborating with the saliva center in order to develop the protocols. But a future stage will be protocol development.
No idea. Now I can refer my patients to the saliva center.
Yes, you can.
He's also on our advisory board, so.
Dr. Saliva.
Are there other questions? Yeah.
So how difficult is it to develop the antibodies? I suppose that's the distinguishing thing for testing each drug, or you have to make a judgment. Yeah.
So we're collaborating with a partner actually in the East Bay. They have specific antibodies for their assays, and they give it to us, and with them together we can not only like put their antibodies on our chips, but also develop new ones.
They're the same ones who supply the Stanford clinical laboratories with the antibodies to use in their big machines. And we have as a co-founder a PhD chemist also who helps us specifically with that.
How long does it take to actually execute a measurement?
So the measurement is very fast. So basically once you put it in, it's an electrical component, it changes immediately. It might take, so the actual signal might take to change a couple of seconds. To get the readout, we talk about maybe a couple of minutes.
Not like waiting for a pregnancy test.
No, not waiting for a pregnancy test.
Let me ask one quick question. Has this technology been used in the industry for purification, that is to say, test levels, purifications, pollutants and things like that?
We are getting there. So technology, so I'm in this field since 10 years, and it just has been commercialized in the last 5 years.
Okay.
So there are applications coming up for things as...
Well, let me know, because that's where I'm going to put my money, too. Thank you.
I use it in environmental sensing, environmental ion detection, any kinds of sorts of things you can do with this technology.
That's my question. Yeah. Thank you.
So I'm going to use the moderator's privilege to say there's a difference between sensing and getting an accurate level, and are you going to go for an actual medical device, and how close is the FDA going to hold you in terms of getting it the same as in blood?
Jackie, we're going to go for a 510K. We're going to do a clinical trial which will compare the levels measured from this to free, to unbound, and to total drug levels in the serum in patients. We're going to show a proportional equivalence to the measurements in blood.
And I'm just going to throw a pitch your way because I had a patient in the ICU that I wanted unbound phenytoin levels.
Yes.
And I waited 2 weeks, at which point I presumed that the unbound phenytoin levels probably changed.
Yes.
And, and the ones that I had ordered were no good to me anymore.
Well, with this, you'll wait 2 minutes.
Let me ask Bob another question, because you said you're going to do a clinical trial to demonstrate the proportionality. But have you done testing yourselves already so that you know that, that it is proportional?
We've just shown, go ahead.
There's a lot of study in the past on research papers. A lot of research papers in the past have shown there's, for Keppra specifically, a one-to-one correlation between blood level and saliva levels. Now, the technology works, of course, if you want to do it in saliva, works only if you have good correlations. But we are talking about 1-to-1, 1-to-2, 1-to-3, something like that, wherever we know. So we know for most of the epilepsy drugs, they have a very good correlation. So this is why, actually, we started with Keppra. And the next thing is to show now, even for the 510K.
But my understanding from your proposal is that you have a way to measure Keppra right now, levetiracetam right now. So what's the correlation between what you are able to measure from saliva with levetiracetam and serum levetiracetam?
This is where we're raising right now money in order to do the next step, which is what you're saying.
So you haven't done that yet?
We haven't done that
We
yet.
haven't done it.
We haven't showed it in water environment that we can detect it specifically with a high selectivity, sensitivity. The next step is now to do that in saliva correlation and get an FDA approval.
Okay. So I've been told we're running long. Thank you very much.
Okay.
Appreciate it. I also have one other comment, and that is that I hear that people are already voting. You cannot vote before we tell you to vote because your vote will not matter. We're resetting the vote when we start the counter. So hold those votes. You may think you already know the answer. You don't already know the answer. Hold those votes till the end. Okay, so for the next presentation, we're going to turn again to seizure detection, and this one is using a smartphone to detect seizures with Cesario. Ahmed Helmi. I want you to know you are not the first one to bring a kid to try and turn our heartstrings. This has been tried before.
He's bringing me.
I see.
So, can we show the video, please? Thank you.
Hi, I'm Ahmed Helmi.
And I'm Amir Helmi.
And we want everybody to have happy health.
Or to be health happy.
That's why we're creating
Health Appy,
a company to develop mobile health applications to improve patients' lives. Our first product is Cesario, and that's what we're here to present.
Cesario is a mobile application that detects seizures and sends automatic messages to families or caregivers in case of an emergency to get help.
This is a typical scenario where Cesario can make a difference. In our daily lives, we leave our homes and family and become subject to various accidents where we need assistance, such as falling or having dangerous seizures. Cesario was designed to detect such emergencies and contact family and caregivers with information crucial to providing help as soon as possible. Through Cesario, you can enter your emergency contacts, emails, or telephone numbers for SMS and save them. When an emergency is detected, an alert is triggered.
Emergency detected. Pick up phone and disarm Cesario alert. Pick up phone and disarm Cesario alert. Sending alert. Sending alert.
And if not disarmed within a preset time, a message is sent to your contacts containing important information on your condition or location so help can be provided. Cesario was introduced by Amir 2 years ago as a science project. Driven by a passion to help others using the latest technology, he took Cesario to school, region, and state fairs, and we hope to go international. Since then, Cesario underwent numerous refinements and significant improvements. Developed by a diverse team of energetic, enthusiastic youth, experienced mentors and experts, the latest version of Cesario was born. Did I mention enthusiastic?
The Cesario team wishes everybody to be health happy.
Well, thank you. As you saw, can we go to the slides, please? So I'm Ahmed Helmi, and this is my co-developer, partner, and my son, Amir. And as you saw, we're here to present Cesario, a mobile app for automated detection and management of seizures. So what is Cesario? It's a mobile app to aid epileptic patients and their families improve their lives, especially in times of emergencies such as seizures or falls. So we have many different features in Cesario. The three main features we want to emphasize at this point is automatic detection of emergency scenarios using only smartphones without the need for any extra hardware. So it's a software, it's an app that goes on your smartphone. It provides also easy and immediate communication between patients and their loved ones and caregivers with crucial information when needed. It's also recording sensed data for further analysis automatically. And well, we can talk about other features later on. Amir will tell us about that. So why are we developing Cesario? We believe that there is a compelling need for several things in this community, including improved self-efficacy and self-management. This has been talked about before. Also to bring a peace of mind to the families of the patients, that's very important. To also reduce the response time during emergencies, and to enhance the connectivity between the patients and the caregivers and the doctors, especially for dangerous seizures that include convulsions and so on, and falls, different types of falls. There are existing approaches for epilepsy-related technologies. Many we've seen here, and we've reviewed several of them. Many, and most of them actually include custom hardware and devices. And some of them are narrow in scope, such as only for sleeping and so on. They generally have limited scaling, and they're usually expensive. So we take a different approach. We try to use everyday technology and use smartphones. We looked at apps out there that are related to epilepsy. Most are simple diaries and reminders that have very limited capabilities. So we firmly believe that Cesario can indeed fill this important gap. And we utilize amazing, unprecedented technologies that we have in our smartphones today, including sensing, computation, storage, communication, and user interface, and also by providing the epilepsy community with a suite of tools to have background sensing efficiently for emergency detection and notification. So this is enabled by an elaborate architecture of software design of 5 main different components. From the bottom up, they are the sensing and detection subsystem that includes the algorithms that are actually the heart of the Cesario. They also run in the background efficiently, even when the phone is in your pocket and the screen is turned off. And when emergency is detected, the data flows upwards into the alert and the storage subsystems. And if the alert is not disarmed, it goes to the notification subsystems and the message is sent to the emergency contacts that are set up. In the middle of all of that, the user is in full control, so they have configuration of what to sense, whom to send the information to, and when to send it. And there are a lot of other features that Amir will tell us about.
So the first main feature that Cesario has to offer is the ability is the ability to detect seizures and distinguish harmful seizures and falls from every normal day activities and send automatic alerts to pre-identified contacts. As you can see here that the seizure tracking options have been turned on, meaning seizures will be detected, and on this screenshot, emergency is suspected. In the rare case of a false positive, you can disarm the alert, otherwise a message will be sent. Second main feature that Cesario offers is an emergency button. With the click of a button, you have the ability to send automatic emergency messages to pre-identified contacts. Another important feature that Cesario provides is the ability to store data for later analysis. Here you can see in our home screen, you can view seizure follower activity history by days, weeks, and months. We also store the data to a separate file for more elaborate viewing by doctors or experts. So where is Cesario today and where are we going tomorrow? Well, as of now, we have major functionality implemented, and we've tested the app with many different scenarios, including simulated seizures while consulting with experts. Initial tests have been very promising. While we've detected 95% of seizures and we have very little false positives, we're now supporting Android operating system, but hope to very soon expand to other operating systems. We want to enhance the algorithm to adapt to specific patients, because not all patients are the same, and we want to introduce a breadcrumbs feature that sends periodic notifications to pre-identified contacts to be able to keep families reassured. Of course, we want to expand testing with patients, and what's nice about our product is that it's an app, it's software. With the click of a button, it has the ability to expand on a global level, and that also makes it allows it to adapt to other hardware such as smart wearables. We want to thank the Epilepsy Foundation and community for being here, and here's our website where you can download the app, or you can scan the QR code to give us feedback or to be part of the Cesario project. Contact us at one of these emails. Thank you.
First, I have to say, very nicely done, and succinct. All right, open for questions.
Ahmed,
Who's
thank you.
going to beat up on the kid first?
Yes. Well, thank you for devoting your energies and your skills and intelligence to epilepsy. Really appreciate that. I have one question. You said that this has been tested with simulated seizures. Mm-hmm. And these are doctors who act out seizures, or what do you mean by that?
Well, this was first a science fair project, so I was not allowed to use real patients. But we collaborated.
Sure.
We collaborated with experts and doctors in the area, and they helped us simulate seizures so we could try to reenact out and see how efficient the app really was.
Mm-hmm. Do you think that's enough to sort of say that or what do you think? What do you plan to do?
That's part of the reason we're here. We're trying to extend our handout so we can get people to help us test out this app so we can improve more lives.
Yeah. Thank you.
Just add to that, because there's a very important point that the data is actually driving these algorithms. As Amir said, we consulted with neurologists in the field, and we saw videos and so on, and we simulated those seizures. We also had an 8-month-long trial of distinguishing normal to reduce false alarms and so on. But also there is the fall detection part, which we haven't elaborated on. There is the free fall, where the phone falls from your hand, but also there's the fall where somebody just falls. And we are also testing those out. And as Amir said, we're reaching out to this wonderful community, to these wonderful people. Hopefully they download the app, and some of them, if they want to volunteer their data, that would help definitely drive our algorithms. And we may even create an anonymous library that the whole community can benefit from.
One other quick question. Are there smartphones where you can wear into the shower?
There are. Yeah, go ahead.
There are smartphone cases that are waterproof, and there are new smartphones that are waterproof.
Shower power. Watch out.
Jackie?
Yeah.
So, no, I have two things. First of all, I'd like to offer you a job. I don't know about funding this project, but let me talk to you later. But second, so I don't wear my phone all the time. I carry it in the purse, and my son carries it in his back pocket, and I have another person at home who wears it in the front pocket. So how does this all work? Because you're tied to an accelerometer in a device that is not attached to the body. It's not even a wristwatch. So how is this going to happen?
We tested the app in many different spots in the body: back pocket, front pocket, holster. And we haven't done handbag yet, but we should look into that. But since this is software, we can adapt to new outcoming technologies, as Google announced this year, and Android Wear is coming out for smart wearables, so we can adapt our program to that.
Okay, and I just want to let you know, we have a very good retirement program.
I just have one question. How old are you, really?
I'm 13.
How old?
13.
13.
Can I just add to that? Certainly, if you have the movement captured by the smartphone that can work with an accelerometer, if you have it nearby, we can potentially tap into microphone or cameras or other things that can sense from afar. But yes, for now, as the app stands, yes, you have to wear it or be in the pocket to detect.
So with wrist smartphones, how would that work?
Oh,
Aren't some of the new smartphones are going to be on the wrist, correct?
Sure. So the nice thing about this is that it's algorithmic based and software which can be ported to the wearables that you have, like smart watches. I think you had the rest exactly. So if we perfect these algorithms with the data from the community, we can certainly port them to these wearables so they can work with anything like a smartwatch, Google Glass. I think even the earpods right now, they're doing sometimes EEG or something like that, so that can also work.
One more question, then we'll move on.
Well, actually, I have two, if that's okay.
Quick.
Yeah, they'll be quick. I want to make sure I understood this correctly. So you're saying that the algorithms will evolve so that they become personalized to each individual, and so ultimately they can track the historical data and basically measure each seizure onto the next?
Right now, we've tested the app with different thresholds. So if you cross the threshold, we can identify it as a seizure. Now, with data that we're going to collect, hopefully through this community, we will see each patient, and how they will, the algorithm will adapt. So say you have weaker seizures than other patients. The algorithm will lower the threshold based on that. So let's say you have a seizure and the threshold was too high, so we didn't detect your seizure. You can go in and manually record saying, okay, you had a seizure, and then the algorithm will automatically get lowered. Or if the seizure was really, really low, that you're getting too many false positives, you can manually put that in, and the algorithm will automatically
or if you say just,
raise the threshold.
you could say you had a seizure, it will remember the movements. The next time you have a seizure, it will be very similar movements, and it will say, all right, and it continues to remember that. So then even if you don't record, or if it's a fall, you just drop your phone, it will know that it will know it's not a seizure.
Correct.
Okay.
So there are several aspects to that, I'm sorry. So one thing is that you have the ability right now to change the intensity and the different parameters, hopefully in a very user-friendly way. So you can adjust the algorithms according to you manually. But if you don't want to do that, we can also have these learning algorithms that will adapt to you depending on the feedback that you get and the false positives and the false negatives and so on, and adjust not only the thresholds but also other parameters.
Okay. And then the last, sorry, one quick. You said that you might be transferring the information to doctors. Have you budgeted in the idea that you might have to get HIPAA registered or any sort of protection?
So part of the budget, I think, that we proposed was trying to cover that. Right now, it's all under the control of the user themselves, and so you have the data stored on your phone. It's not shared with anyone else.
So you're not going to put it in the cloud. It's going to be...
Right now, it's not.
When you go to a doctor, the doctor can view the files.
Okay.
Yeah, so you can either wait until you go to the doctor or wait, you can email it to them if you so choose, but that's configurable by the user.
Thank you so much. Okay. If you would like to be seizure happy, you will vote for this particular proposal. We have one last fish, Michael Lundell, Ketonics Breath Ketone Analyzer.
Boom. It works. Okay, can you? Wrong. That's the wrong movie. That's not the correct movie.
That's the wrong movie. Can someone somebody put on the right movie.
But
It's a nice
yeah.
movie.
Yeah, it is. If we get time left, I can take that.
There will be no time left. I
Nah.
can guarantee you that.
Okay. So you can put on the presentation. Is that it? Okay. So, hi, I'm Michel Lundell, and I'm CEO of a company called M-Object Oriented Software Engineering. We're a Swedish-based company located on the beautiful west coast of Sweden, which normally looks like this. A bit like here. For the last decade, we supplied IT expertise to big pharma in Sweden, a small company called AstraZeneca. I don't know, have you heard it? But today I will talk about a new product that we developed. It's called Genomics, and it all started 2004 with me having a seizure. A lot of tests were done, and one of the tests revealed that I had a possible brain tumor. The dream of having kids and see them grow up was lost. And many scans later, a specialist group concluded that it is an unidentified brain object, which I could live with for many years, and great. So I got a UBO and a brain. Not everybody got a documented brain. So I was happy, and I put it up in my LinkedIn profile. Fast forward to 2012, business as usual, everything was fine, and boom, I got another seizure. And the neurologist, he told me, you got epilepsy, and here's the drug. Take it and get used to it. So I took it, and one morning, 1 year later, my wife said, you changed in a bad way. So my personality changed. I was bad-tempered and stuff like that. So I looked for options. And doing that, I found the ketogenic diet, which could be an option to the drug. And I spoke to my neurologist, and he spoke with his colleagues that been doing studies of this, and he agreed it could be an option to the drug. So the switch from drug to the diet was planned in January this year. And before we did that, I had to prove that I could manage this diet. And the diet is a high-fat, low-carb, moderate protein diet. And how hard could that be? It gets my body into ketosis, which produces the ketones for my brain. And how would I know? So in the beginning, I was using urine and blood samples to see if I was in ketosis, which I found was very expensive and messy. So isn't there a better way to measure if your body is in ketosis? I made some research and found that acetone in breath could indicate ketosis, will indicate ketosis. So where do I buy a device that I can use for measuring this? I couldn't. There was none available. So being me, I set out to make my own. So I milled, I drilled, I cut, I measured and I tested, and finally in August 2013, I got a device that worked very well. I used it all the time, and people just wondered, what are you doing? And I explained to them, and they thought this was a good idea. You had to get it out somehow. So before I get into that, I want to explain how it works. You plug it in, and it initializes, it calibrates to the current conditions, like temperature, humidity, pressure, stuff like that. And when it gets steady blue, you exhale into it until you don't have any more air in your lungs. Then you view the result. And you can do this as many times as you want. It's very simple. So that's how the device works. You blow into it and you see if your body is in ketosis or not, or how much. So to explain how to use it, to apply it in real life, we take an example. Oh, wrong button. Lunch with a friend. Before you go to lunch at your friend's favorite diner, you measure a nice level of ketones. At the diner, you choose the most ketogenic meal you can find on the menu. It could be hard. After the nice lunch, back at the office, you measure again, and you notice that your ketosis has decreased. So what do? You adjust the ketogenic ratio of the next meal, and later on you can see that you restored your ketosis again. Lessons learned from this example is that even if you try to avoid the obvious carbs in that meal, there was some hidden, maybe in the sauce, maybe in the glaze on the steak or whatever. So don't eat there again. Will it work for others? It worked well for me. I correlated this to urine strips and blood measurements. It's not the same blood and acetone ketones, but anyway, it indicates ketosis enough. So I made some more devices, and I let experts or leading figures in the diets of paleo, low carb, high fat, and diets like that, try them. And they embraced the device and said it could help many to manage their diets for different reasons. One of them is, in Sweden at least, quite famous, and he said it's good for any number of measurements. It seems to work pretty well. And it's better than urine strips, but not quite as good as the blood measurements. And of course, it's not a blood measurement. It doesn't measure or indicate the same substance. And then from the US, there's a Jimmy Moore. He couldn't wait to get to share this with the rest of the world, he said. And he didn't wait. He actually showed it on an obesity conference in March this year, without asking me anyway. Anyway, they loved the device, and I decided to publish this to the world to help others that need to manage their low carb, high fat diets. So I put up a simple website, and I made an iPhone app, which is a ketogenic calculator, and let the app point to the website. And now the first adopters, they put out reviews, which were really, really positive. I was surprised. And now, since January, we have 600 users. And I'm getting feedback every day, and they are very happy with the device. It's a fantastic feeling to read this. So who's using it? And who would benefit from using a diet like that and a device like this? Well, we found that it's a lot of research in the effects of the ketogenic diet. It's not only for epilepsy. It's other medical conditions too, like diabetes, obesity, and other conditions. I think it could enhance the the quality of life for many to use a diet like this and adjust their medication. It's also in sport. Cutting-edge athletes are using ketogenic diet to get access to the body fat, using that as fuel. In sports like triathlon, ultramarathon, marathon, biking, running, you name it. It's really cutting edge in sports now to actually leave the carbohydrates behind and get your body to use the fat. And of course, we all want to lose the love handles for the beach season. And if you want to lose your fat, you have to use it. And when the body is turning the fat into energy, it creates ketones, which Genomics will indicate. So I'm now using diet, no drugs anymore. I manage my ketosis by using this device. I enjoy the side effects like weight loss, like energy level, like endurance. And I love the feedback from all the Genomics users we have now. And it's been a pleasure to talk to you, and thank you for listening.
Thank you very much. You realize you're going up against a really smart 13-year-old kid. Just saying. And I think we've given you a curveball here. It's kind of like, I don't know the right analogy, but like somebody on the Shark Tank pitching to Mark Cuban with a sports idea, because we have Jim Abrams here who runs the Charlie Foundation. So Jim, I'm going to give you the first word. So
my question is, it measures acetone. Does it measure other ketone bodies like beta-hydroxybutyrate?
No, it doesn't.
Oh, sorry, go ahead.
No, it doesn't. It comes from the same source, acetoacetate, but it doesn't measure the actually beta-hydroxybutyrate, no. And that, we say BHOB, that's simpler. That could be used as energy, which acetone isn't. So the level of the blood ketones and the state of ketosis could differ, because if you need energy, the concentration of the BHOBs are going down, and the ketosis is increasing because it detects that you need more energy. Mm-hmm. So if you measure your blood ketones, it doesn't say as much as this device does about the ketosis process.
I have a few questions. What are you actually sensing? We haven't heard too much about the technology.
It is sensing gas, acetone gas.
Strictly acetone?
No, there is other things.
Is it related to breathalyzers or alcohol sensors?
Yes, sort of. Yeah. You shouldn't drink wine and expect.
No, but would it measure ethanol as well as acetones?
Yes.
Okay, so how do you know how much of it is the acetone, how much of it is the?
I would say that if you have a glass of wine and then you measure your ketones, that is out of context, because it's we have that in our manuals that, well, you shouldn't drink.
Okay, and just because I know we're running out of time, this is qualitative, it's not quantitative, so you have high, medium, and low, is that correct?
We have two versions. One is low, medium, high, and one is actually 30 in a 10 divided. Each low is 10 different readings, medium is 10 different readings, and high is 10 different readings too, so you know where in the scale you are. Because if you blow 2 times and you get green and yellow, then you know you're in the borderline. But now you can see one yellow is very close to
okay, and any IP on this?
IP?
Intellectual property?
We just registered the name in Sweden.
So that's a little different than the technology, as you know.
Yeah. And the technology It's common.
We
cannot take any patent on it because my friend, who is a patent engineer, he told me that, okay, do you have the money to sue anybody that tries to take your thing? And I said no, and he said, well, publish it.
Well, if you're if you're talking about developing a company, though, and we heard from the VCs, you need some leadership and management. You need some proprietary information, which it sounds like you don't have. And I think you also need a focus. So you've talked to us about epilepsy, diabetes, obesity, and I don't know that there's necessarily a clear regulatory strategy either, right? You don't know if this functions the same in one person to the next. You say that it doesn't correlate perfectly with blood levels. So I would suggest that picking a disease state and deciding what your regulatory strategy would be a helpful step, not to monopolize the time.
Yeah. We have a strategy of developing specific devices, tapping into just focusing on, for example, diabetes use, because there are a lot of users that are having diabetes that use this, and there is always different quirks to each application, of course, but to use it to see if your diet is working, that's sort of general, actually.
So of your 600 users, how many have epilepsy?
That I don't know.
Okay. Are these folks sharing their data with you?
Yes, some of them are. I encourage them. As soon as somebody is taking contact with me, I always encourage them to know why do you use it, what is your interest, and stuff like that.
Okay.
This side of it, I'm going to ask a question. Do you have any data with blood levels and with your device to show that your device and the blood level or the urine strip, or how many of them do you have?
We have done a lot of tests with, in the beginning, urine strips, and then we continued with the blood measurements, and then we noticed that the blood measurements and the acetone in breath, that could differ because the blood ketones are getting used as energy. So even if your process is working fully to produce ketones, the blood ketones could actually go down because you're using the energy.
Right, so
the acetone is not used.
But I guess the answer is they don't exactly correlate. Okay, so we don't know which one really is the truth.
I can say how I use it. I use it as a compass. But I will say it's. I use it as a compass. If I keep that ketosis high, I know I will have enough ketones. Whatever the blood says, I will have it in a few hours. If, for example, if I've been out running, the blood ketones will go down. But I know in a few hours the body has, created enough to get it back, or even higher. So you have to think a little bit. But
okay, very good. Yeah.
Is there another question down there? Or?
Yeah, I don't see any from that side, so it's all yours.
So you don't know whether the people who, of the 600 people, any of them are on anti-epileptic medications also?
I haven't specifically asked that. I haven't known.
So you wouldn't know, for instance, like Depakote, people who are on an anti-epileptic drug called Depakote will get false positive readings for ketones in their urine. Do you know that you wouldn't be able to answer whether that would be true in their breath also?
Any other questions? Thank you very much. So you can feel the excitement building. We're getting very close. Don't vote yet, because before you vote, I know it's a little late, but there is a reception waiting for you, and I think that there are some kinds of ketone bodies, or at least alcohol, in that reception. But before we have the vote, we are going to see what our sharks think. We're going to ask them who they're going to vote for now. And remember, what did I say before? No loan sharks. Can you say it with me? No loan sharks. So if any one of them is the only one that votes, then we're going to have a discussion up here to figure out what the final answer is going to be. So by the time we finish here, you're going to know how the sharks have voted and how that $125,000 is going to go. And then each one of you be prepared. I want one sentence why you guys should vote for me after we find out where the $125,000 is going. Okay? So we'll start actually on this end.
Yeah. So I think I'm actually going to go with the saliva-based solution. I think it's a very breadth of impact. And I also know from growing up that testing can be a pain as you're growing up and your levels are constantly changing, metabolism's constantly changing. And if that solution can be applicable and impactful, it's something that I see as being a very strong solution in that area. But I'll stop taking the floor.
Okay, we got $25,000 on saliva. Dr. So.
Yes, it's a very tough decision for me between saliva or the biostamp, but I'm going to biostamp. And I think it has great potential, and I think the technology is there, and it will take perhaps a few more years, and they really need to push. But I wish I have a little bit. Can you give me a little bit more money so I can give it to Avails? I like the name.
Okay, so remember, you guys, wherever you are, you don't have the money yet. You don't have the money yet. We have to see whether there's a match. Okay, coming over to this side.
Well, I'm going to have to say that I was struggling with between the same two. I really like the idea of the biostamp because I see it as a platform that can continue to develop to help different types of people with different types of problems, and it can be used efficiently and effectively as technology will continue to develop. But I think I'm going to have to put my money towards the saliva study as well, because I think that that's going to be able to provide.
We have a winner. We have a winner. There's 50,000 unless we have some changes. Okay, I can't actually say yet.
So first of all, there were a lot of really wonderful proposals, but Jackie told me to be mean. And so
you weren't supposed to tell them that, Marty.
I don't know if I can be mean, but maybe I can have tough love. And I just want to make a couple of comments. First of all, I think that for many of these projects, it's great ideas, but we don't have that much money, and it needs to go towards epilepsy goals. And some of these, I don't think homework has been done yet. So I would say that's with the MC10 product. So yeah, the technology is cool, but it's all promise, and there's not a substance there. And I would have felt a lot better if they had come to us knowing what is already available to patients and what is being developed. I wasn't even clear that the unmet need, when I say unmet need, what is unmet in the area of wearable technology and seizure detection. You're not even ready for focus groups yet. You need to find out where people have been spending their money, where the Epilepsy Therapy Project and Epilepsy Foundation have spent their money. And putting that old picture of me up there, that didn't help. Although it is 10 years old. So. And I think that the saliva testing, I think Avails is fabulous, and I did appreciate that proposal. It was very focused, it was well thought out. It's early and there's a lot of risk, but I felt there was homework being done there. The catonics, I think that's really early too, and again, it needs to think about what the focus is. I would have liked some more information about the technology. It's not helpful if we give people information that's misleading, and I don't want to support a project until we know that it's actually have more information about whether it's going to be accurate enough to be safe to use in patients with epilepsy. The iPhone or the Android accelerometer, that's really incredible. And I would, I am so torn, but I'm going to stun everybody, and it's only because Avails was funded And I'm going to try to give $25,000 to shower power. And the reason is that I have so many parents and young patients that the kid is on one side of the bathroom door and the parents are on the other and listening. Now that's a tough situation, and I believe this is doable. And you guys didn't see the proposal. It's really well lined out with milestones, go, no go, and it's doable and it's quick. And frankly, it's not the most techno sophisticated, although it's pretty good, but it's a simple, solid solution to a real problem. So that's where my vote's going, or my 25,000, I guess.
For right now.
For right now.
Okay, there's some money on the line here.
Right. So my heart was rooting for a key tonic, and I would hope that you continue your research and refine it and become more specific with it. Today, I mean, for years, all we ever heard about with a ketogenic diet was epilepsy treatment. Today, if you Google ketogenic diet, the first thing that comes up is cancer. So there's going to be a huge application for testing urine through your breath in the future. So I hope you don't give up. And then I guess where I leaned strongest after that was the Web Easy. And I'll tell you why.
Oh my goodness.
I know.
It's getting exciting now.
and here's the reason. Dr. So and I were talking about this earlier today, too. My experience with the epilepsy population, years ago there was a study in the Los Angeles area, and there were 40 people who answered with epilepsy who answered a questionnaire about their epilepsy. And they were from all walks of life, all different backgrounds. And the one thing everybody had in common in this study was anger. They were angry that they had epilepsy. They were angry they had no control. They were angry about the drugs. They were angry about they couldn't drive. They were angry they had 100% of the respondents in the survey talked about their anger. And what I really loved about Web Easy is that it allows the patient, a kid, a teenage kid, to start to take control. So he's not, the word empowerment was mentioned several times during the presentation. And I think that's a very powerful aspect of the Web Easy thing, that I'm not, my life isn't totally out of my control. I can actually have some influence on my health and my well-being. And I think, then I'll shut up, but I know from teenagers who are on the ketogenic diet, and it is a difficult regimen to stick to, and it is very restrictive. But for the kids who stick with it, teenagers, there is that sense of, wow, look at me, I've taken control. I'm actually pulling this off. My epilepsy is better because of me and what I've done. And I think that's a huge area of epilepsy that we need to be thinking about.
Okay, so we now have, I'm just keeping score here, we have a match for the saliva, and we have 3 lone sharks. And repeat after me. No lone sharks. Okay, who's going to convince somebody, or do you want to change your vote, Dr. So?
Before I'm going to change my vote, and I know that Dr. Morell is not going to come along with me. She had already said enough bad things about biostamp. So I'm going to ask Mr. Abrams if he would come along.
I just say that it's not ready for this for us right now.
I'm kidding. So maybe you call me Dr. So I'll have to call you Mr. Abrams. Can you come along with me? And if you don't, I have a tough decision to make.
He wants you to change your vote to his vote.
I understand.
I take that as a no. Whenever I invite a girl out to a party, I understand.
So I'm going to, before you do that, I'm going to ask these guys over here, like, you guys don't immediately want to change over to anything.
Well, I just think we have a wrist seizure detector that's marketed and that works really well. We have a lot of stuff that's much more mature. So maybe it's because I'm from the Valley, but you can show flexible things with little microelectronics and say, and it could do this, and it could do that, and it could do this, it could do that. There were lots of coulds. There weren't any does.
Okay, I've made a decision.
Yes. And?
I'm going to give my 25 to avails.
Okay.
Final. That's my second choice.
Yeah, that was my second choice also.
So let me talk. Let's talk.
Okay.
Because otherwise she's going to say, you're dead to me.
Kiss and makeup.
No, I think the WebEasy is great, and I absolutely agree. The goal is laudable. All of these goals are laudable. Yeah. But the question is, what's going to be the most quickly realized, and which group can actually succeed in what they've set out to do? I am concerned that the product WebEase, I would have liked a lot more information about whether people are using that. This probably is me. I didn't even know it existed. And that's probably my ignorance, but none of my patients have talked to me about it. So I think it would have been very helpful in the presentation to understand how they were building on that strength. Secondly, they're talking about making something exciting for teenagers, but I didn't hear necessarily that there were members of the team. There's certainly great public health people, no question about it, our colleagues that I respect and admire, but where are they going to get that wow, that Hollywood factor? I didn't hear how that was going to happen. And I know that my 17-year-old has attention span of 3 seconds. So it's going to have to be really flashy. What we saw, it was a prototype, but it wasn't that flashy. So that's my only concern. I'd really like to hear more information about the team that was going to make this. Well, you're from Hollywood.
So are you going to give your money to Shower Power? Or are you going to make Dr. Morell?
I hate to, so how many salivas do we have?
We have 3 salivas.
And 2 shower powers.
Are you giving to Shower Power?
Yeah.
Yay! Okay, but it's not over yet. We have 2 winners. We could have 3, or we could remain with 2. I want all of the groups to stand up and give me 1 sentence in order, wait, where's my order, about why the audience should give money to them. So we'll start with Shower Power. You already have some money? Where are you? You already have some money in the bank? Why, tell me, would we give you more money? What would you do with more money?
With additional support, I believe we could push the product to market even faster and make sure the focus was on safety for not only epileptic patients, but also for other groups.
But you have to start learning how to say epilepsy patients.
Sorry.
If we're going to give you any money. Okay. All right. BioStamp. You had a vote. You lost the vote. Where are you?
One especially potent shark as well. So thanks for the votes. And there was a parent yesterday on a panel who said they don't see devices being the same if they're being used for research purposes and monitoring purposes. There was a parent actually on a panel who said that as opposed to a physician. And we actually have the ability to do both, because by making something smaller, we can actually get more sensors on or make them more wearable, either way. So from that potential and really being patient-centric, I ask for your money.
Sandy.
Okay, so we have the technology, we have the team, which includes teens, to give us what we need to do to create this technology tool to empower the teens to take control and manage their epilepsy for now and into the future. And with your funding, we can do that.
And I think you're going to have a hard road to hoe, but, Dr. Fisher, no, not you, Sandy. Why do you want more money?
Me?
Yeah.
We don't have a whole lot of money. We're just starting up. We don't have enough to do what we need to do, for example, for the clinical trial. So that's one of my 3 points, that this is going to make a difference. We don't have big backing. We're just beating the bushes around Silicon Valley now. Secondly, supporting this idea leverages physics technology, which is already developed in another field and is really ready to be brought into the medical world, the medical community. I guess I have 4 points. The third is that...
Wait, everybody else just had one sentence. I'm sorry.
Okay.
You need to sit down.
I'll sit down. All right, I heard people
and as the
saying more than
person
one.
who already got money, I'm sorry, Bob. I love you, but... I said one sentence. It was a long sentence. Okay, I think you're making a big mistake here. Send the kid. But go ahead.
He
go
told me
ahead.
to come up here. He thinks that you're not taking us seriously because he's here. I think you're making a big mistake.
No.
You should
no,
be
I don't
taking
think
us
so.
very seriously. We're very dedicated behind this. We're going to take it all the way. We're going to put all the resources. We're willing to team up with people. And I'm from University of Florida. I was with Southern California before, and we know people, neurologists there. My son is fantastic. I'm very proud of him. But this, I think we can make this in the hands of millions, literally millions of patients with a click of a button. You cannot do this with any other technology. That's why you need to back us up. Thank you.
Don't stop.
And, okay, ketonics. Last one. Oh, you're behind the pillar. Okay. Go ahead.
Okay. We need the money to market this product that is not a paper product. It's a proven product that works. And even if it's not specifically for people having epilepsy, it works for them too. So.
Thank you.
Thank you.
All right. Okay, you all have as much information as you're ever going to have. And it is time to vote. And now, David, are you going to come up and tell us exactly how to vote? And be very careful where you touch, because you get one vote and one vote only, and you don't get to take it back. So once you've hit it, that is your vote. I'm just telling you.
Hello. So first, you have to download the application.
Hopefully they've done that already.
Yes. And then, Brian, if you can show the other slide, you have to log into the, what's it called? There's a Wi-Fi spot, a specific one for
This is
the Hyatt.
The Hyatt meeting,
Hyatt
and
meeting.
this is the Wi-Fi code.
For Hyatt meeting. And then you enter this code here, 16645-1030.
It's for the audience.
Then you get access to the Wi-Fi, and then you can use your application. Next slide.
Next slide.
Next slide, Brian. Then on your home page, below the exhibit hall, you have a you will click on Shark Tank poll. This will open up a web page where you will have all of the finalists for today, and you will select you can select only one, and you can only vote once. It is not open yet. Give me just a sec. I need to go back. It's open. It's open. Okay, good.
Okay, is everybody ready? You have 90 seconds is what I understand. And in 1 or 2 of those seconds, we are going to see the live voting as it happens. I can't stand it.
That's a big gap. Yeah, it's
Now, I understand there are 51 seconds left, so if anybody wants to come up and do a dance or something, I think that those... A man walked into a bar. Yeah. No, focus on your voting. And we are apparently going to cut it off before the very end so that we can announce the winner. Oh, I see some movement still. 10 seconds left. Those who haven't voted, please vote. And now we are going to show you in just a couple of seconds the winner or winners. We know we already have 2 winners of the 2014 Shark Tank competition. All of you were great. I just want to tell you, many kids have tried, but none have succeeded until now.
Well, you know what? I just also want to say, I suspect you'll be able to get into any college you want.
One final announcement. Is Andy Cole in the room? Well, with Andy, we're going to work with the American Epilepsy Society to provide scholarships for postdocs to attend this conference next year. We're hoping to provide 4 scholarships at $2,400 apiece. And I'm going to support that. I'm looking for others to join me in funding that for next year's conference. So please see me if you want to help make that happen. Thank you. Phil.
All right. Ladies and gentlemen, as we break for our dinner, as the conference ends, I just want to say that the foundation is privileged to have hosted this amazing gathering. I want to take 30 seconds, if you'll indulge me. Yes, we are going to have checks coming up and check presentations if you want to see that and maybe even get an autograph or something from our winners. But before we do that, I just want to say thank you. You are our agents of change. You are the group that is going to make the difference for families for years to come. And I'm just so proud to have been here and experienced this with you all. Warren Lammert and I know that for too long we've operated in silos, which can delay the timely development and implementation of urgently needed therapies. Coming together allows us to facilitate partnerships that bring together expertise and funding support to advance therapies. It's exactly why the Epilepsy Foundation has dedicated itself to sponsoring this unique conference. And speaking of partnerships, I have the pleasure of announcing that pending our board approval, the Epilepsy Foundation is entering into a partnership with an epilepsy biotech company in telemedics led by Jim Jacoby and Daniel Fisher and Jeff Skolnick, along with Pfizer, led by Dr. Frida Lewis Hall, Ruth McKernan, and the Pfizer team, to develop a precision, personalized drug delivery platform for epilepsy. The first phase of the project will involve gene sequencing, computational modeling, personalized zebrafish lines, and iPSCs from 20 patients with Dravet syndrome, and will screen thousands of novel compounds. Thank you, Jim, Daniel, Ruth, and Frida, for your ongoing commitment for people living with epilepsy. We have also been fortunate to have been asked to partner with Epilepsy and Behavior, the highly distinguished, incredible journal led by Editor-in-Chief Steve Schachter, along with Bruce Herman. And Steve, I want to thank you so much for your leadership and your partnership in bringing the Epilepsy Foundation mission forward to a community that is so vital to the success of our mission. So thank you, Steve and Jim and Daniel, for these special partnerships with the Epilepsy Foundation. As fathers, inspired by children living with the serious daily consequences of seizures and side effects and risks of epilepsy, Warren and I share a passionate sense of urgency to make a difference for our kids and for every individual and family living with occurring seizures and their too often devastating impacts. 1 in 3 of those diagnosed with epilepsy have ongoing seizures despite all available therapies, and at least 50% of people with epilepsy live with unacceptable side effects from medications. Sudden unexplained death in epilepsy, or SUDEP, and other causes of mortality from epilepsy continue to take too many lives. Our conference is a source of inspiration and hope, as well as an opportunity for the broad community to celebrate and support the work of researchers and innovators working together to make available new therapies and bring an end to seizures, their side effects, and lives lost to epilepsy. Many of you have thanked the Epilepsy Foundation in your remarks and the Epilepsy Therapy Project for the support you've received. On behalf of my board chair, Warren Lambert, our entire board of directors, and leaders from all over the country representing the Epilepsy Foundation through our affiliate network, we thank you for your steadfast commitment to individuals and families and your creativity as together we accelerate hope for an end to seizures, side effects, and lives lost to epilepsy. Thank you. And here we have some checks. So who are we going to call up to the stage? Oh, okay, well. We're going to sign these checks and then we're going to hand them over. So, okay, Oren. Okay, can you come to the stage, please? Oh, Warren can sign. Warren, you sign. You go for it. We'll both sign. We'll both sign. Here. We'll both sign. Oh, great.
Sorry, I don't take credit cards.
At next year's conference, we will be paying in Bitcoins.
Congratulations, you guys. It's wonderful. I like to see the thanks expression when you walk in with this. You want to take it with you?
No. We have only one. We need two.
Oh, you're going to erase it?
Yes.
Oh, my gosh. You're going to erase it.
I only had the money for